including AHA Young Scientist Award and Ramalingaswamy Fellowship. We warmly welcome Dr. Manish Jain to chair this session. Welcome, sir. Each presenters will have 7 minutes for the presentation and 3 minutes for discussion. Let us maintain a supportive environment and appreciate the scientific efforts behind every presentation. Now I would like to call Shreya Saxena from Saraswati Institute of Medical Science, Greater Noida. She is not here. Next speaker is Shiv Kumar Saini from CSIR IHBT Palampur. Next is Diksha from AIMS Delhi. Please come on the stage.
Good morning everyone. So the title of my presentation is Preliminary Study for Determining the Anti-Cancer Activity of Beta-acin against Human Lung Cancer Cell Line. I am presenting the interim results. So lung cancer is the leading cause of cancer-related mortality. Despite the introduction of targeted chemotherapy, the 5-year survival has only reached to approximately 20%. And the tedious and costly process of new drug development has shifted the attention from plant to the natural plant-derived compounds, which often possess diverse bioactive properties. Hello? Hello? So beta-acin is a natural saponin which is derived from the seeds of Esculum hypocastanum, that is horse chestnut, and has shown anti-cancer activity against various cancer cell lines like osteosarcoma, breast and ovarian cancer. So the aim of the study is to determine the anti-cancer activity of beta-acin against human lung adenocarcinoma cell line, that is A549. In this, the objectives are to screen the anti-cancer activity of beta-acin using MTT, and thereafter to confirm the anti-cancer activity of beta-acin. Annexin 5 propadium iodine staining and Hodges staining was performed. And later on, the mechanistic properties were determined using the cell cycle and reactive oxygen and nitrogen species, and also the effect of beta-acin on autophagy induction was determined using phosphorylated ULK1. So initially, beta-acin activity was screened against human lung cancer cell line A549 using MTT assay. And thereafter, according to the IC50 dose, 3 doses were selected, and in those 3 and 2 doses were selected, and thereafter the apoptosis activity was confirmed using annexin 5 propadium iodide staining and Hodges staining. And the further mechanistic pathway was determined using cell cycle analysis, reactive oxygen species, nitrogen species, and autophagy protein, PULK1, ATG5, and LC3AB. The ones in the red are still going, so I will not be presenting those results here. So initially, the MTT assay was performed at 24-hour, 48-hour, and 72-hour. With increase in the duration of incubation of drug with the cell line, there is increase in the IC50, but there's a slight increase at 72-hour. And in annexin-5 propydium iodide staining, in all the markers like necrosis, early apoptosis, late apoptosis, the drug is significantly causing decrease in the population of live cells, along with inducing apoptosis and necrosis in all the groups, that is 20 micromolar beta-acin and 25 micromolar beta-acin. In Hodges staining also, it is a marker for determining the nuclear fragmentation, as it is observed that there is increased in the intensity of Hodges staining observed in all the treatment groups as compared to the control group. And it is significantly causing nuclear damage at both the concentrations, that is 20 micromolar and 25 micromolar. And this drug is also inducing a cell cycle arrest in the normal control population with the 60% of cells in the G1 stage. It is causing increase in the arrest of G1 cells in the G1 phase of the cell cycle at 2 doses, 20 micromolar and 25 micromolar. And thereafter, the autophagy marker, that is PULK1, was determined, and in this marker no difference was observed between the control and the treated group, but we are still doing the other markers of autophagy. And one more, nitric oxide. In this, there is significant decrease in the nitric oxide release in both the treatment groups as compared to the control group. And the work is still ongoing, and will be determining the ROS and proteins of autophagy pathway, and also we will be doing the acridine orange staining. So as per the results, beta-acin has showed anti-cancer activity against the human lung cancer cell line, but further confirmatory studies will be required for its mechanistic property. Thank you. Beta-acin is a plant-based supplement derived from Asculum hippocastanum, that is horse chestnut. Like, acin is a complex mixture out of which alpha-acin and beta-acin is there. So I am taking the beta-acin because beta-acin in the previous studies also has shown higher anti-cancer activity as compared to the alpha-acin. Osteosarcoma, breast cancer, ovarian cancer. Yes, sir. A549 human lung adenocarcinoma cell line. Sir, toxicity is like, so that I have not determined. No, sir. Sir, this is the first time in lung cancer cell line. Sir, right now I'm doing the remaining assays, and then we will do the animal studies. Thank you.
प्रेजेंटर को बुला लो, फिर इंट्रोड्यूस कर दो।
It is our privilege to introduce our chair, Dr. Nirmal Singh, Professor of Pharmacology, former head of the Department of Pharmaceutical Science and Drug Research at Punjab University, Patiala. With over 27 years of a distinguished academic and research experience, Dr. Singh has made major contribution to myocardial and cerebral ischemia, reperfusion injury, dementia, learning and memory, and neuropathy. He has authored over 260 research papers, boasts an impressive H-index of 61 with more than 17,800 citations, and has guided numerous M.Pharm and PhD scholars. His scholarly books, patent, and multiple awards reflect his impactful scientific leadership. We are honored to have him as a chair for this session. Welcome, sir. Now, it is my privilege to introduce our other chair, Professor Sangeeta Yadav, an iconic name in pediatrics and adolescent health, currently serving as professor at HIMSR and HAHC. She has previously been professor of excellence. This is not Sangeeta Yadav. Sangeeta Yadav. Okay, welcome, ma'am. Please, please, ma'am. Please, ma'am. Please come on this.
I'm Dr. Sangeeta Yadav, and I'm Associate Professor in Department of Pharmacology at Dr. Babasaheb Ambedkar Medical College, Delhi. And
It is my honor to introduce our one more chair for this session, Professor Avijit Hajra, from Professor of Pharmacology at IPGMER and SSKM Hospital, Kolkata, a distinguished clinical pharmacologist. His expertise span clinical trials, drug safety, biostatics, and medical education. With an academic journey beginning with his MBBS in 1992 and MD in pharmacology in 1997, he has since contributed extensively through teaching, research, guidance, and clinical trial leadership. He has also served the Indian Pharmacological Society as Secretary, Clinical Pharmacology. We are privileged to have him as a chair for this session. Thank you, sir, and welcome, sir. Now I would like to call our next presenter, Vinay Tyagi from Central University of Punjab, Bhatinda.
Good morning, everyone. Good morning, everyone. My name is Vinay Tyagi today. Hello. Good morning, everyone. So the title of my presentation is investigating the neuroprotective potential of remote ischemic preconditioning in sepsis-associated encephalopathy in mice. So this is a table of content. So let's begin with the introduction. What is sepsis? Sepsis is a life-threatening condition that commonly affects critically ill patients admitted to intensive care units and is the leading cause of death. So it is a result from the body's overwhelming response to an infection, which leads to the multiple organ dysfunction, leads to the including kidney, liver, heart, and brain. And if we talk about its prevalence, it globally affects 49 million people, and in India alone, 11 million cases. So one of the most common complication in sepsis is sepsis-associated encephalopathy. Around 30 to 70% of ICU patients caused increased mortality and long-term consciousness and cognitive deficits. Long-standing hypotension and systemic inflammation leading to the blood-brain barrier disruption, mitochondrial dysfunction, oxidative stress, and neuronal apoptosis, which leads to the sepsis-associated encephalopathy. And these changes are often associated with elevated biomarkers of neural injury, including NSE and ascended beta. Currently, there is no specific treatment for SAE, and except some antibiotics, which has their own limitations and side effects. So recently, a non-invasive technique called remote ischemic preconditioning that shown promising results, and it has been shown to reduce inflammation, oxidative stress, and apoptosis in various. Hello. Hello. RAPC has been shown to reduce. So RAPC has been shown to reduce inflammation, oxidative stress, and apoptosis in various conditions, including stroke. So studies have also shown that RAPC protects sepsis-induced organ damage such as liver, kidney, and heart. So coming to the research gap, so RAPC has shown protection in heart, kidney, and brain. And hello. And RAPC is known to exert its effects via TRP1 channel for its cardio protection. Additionally, TRP1 is also known for its cardio and neuroprotective properties. However, there is no literature is available for the RAPC protection in sepsis-associated encephalopathy via TRP1 medication. So our hypothesis is we used cecal ligation puncture model to induce sepsis-associated encephalopathy. This procedure induced the release of PAMs and DAMs into the bloodstream, which activate the TLR4 pathway on immune and neuronal cells, and which further activates the NF-kappa B, which is a transcriptional factor for the producing pro-inflammatory cytokines. And these pro-inflammatory cytokines further leads to the neuronal inflammation, oxidative stress, and neuronal apoptosis, which is the hallmark features of sepsis-associated encephalopathy. So we hypothesize that the cycles of ischemia and reperfusion, 5 minutes each, on hind limb, which protect the distant organ, so which further promote the further activate the TRPM channel and increase the level of CGRP and phosphorylate and activate the AMPK pathway, which is further activate the autophagy, which inhibit the apoptosis. So the aim of the study is to investigate the neuroprotective potential of remote ischemic preconditioning in sepsis-associated encephalopathy in mice, and the objective of the study is to investigate the effect of RAPC on oxidative, inflammatory, histopathological, and apoptotic parameters in CLP-induced associated encephalopathy in mouse model. So our experimental design is, the protocol was conducted for 14 days. So RAPC therapy provided 24 hours before CLP surgery, which include ischemia and reperfusion cycles, 5 minutes each, and the pharmacological intervention were administered 30 minutes before CLP. On day 1, we performed surgical procedure called cecal ligation puncture, and on day 7, we performed open field test, followed by NORT on day 8 and day 9, and further we conducted the Morris Waterman test for day 10 to day 14. After these tests, we sacrificed the animals and collected the blood and brain tissue samples for further biochemical estimation and histopathological studies. Coming to the methodology, so as I mentioned earlier, we induce a cecal ligation puncture method to induce sepsis-associated encephalopathy in 6 albino mice, and along with the check the mechanism behind the remote ischemic preconditioning, we used capsaicin, CGRP 837, and compound C. For this, we use 46 animals. For sham control, we use 6 animals, and further CLP, remote ischemic preconditioning, and further interventions, we used 8 animals in each group. Further, in this study, we evaluated various parameters. For behavioral study, we used open field test, NORT, and Morris-Waterman test, and evaluation of brain edema and nasal staining for histopathological studies, and biochemical markers of neuronal injury, oxidative stress, neuroinflammation, and apoptotic markers. Coming to the results part, we performed the OFT, and it was found that there is a significant increase in the level of distance travels, number of rearing, number of tossing, and latency of a central area in RIPC group as compared to the CLP. And after the intervention of various pharmacological drugs, it was found that the protective effect of RIPCs is reversed by these interventions. Similar results were found in NORT test, and same on the Morris-Waterman test, in which we suggest that RIPC has a significant role in recognition, spatial memory, and these cascade, TRP1, CGRP, and AMPK is involved in RIPC protection pathway. Further, we check the neural injury markers and brain water contact, in which we show that in CLP group, there is a significant increase in the level of brain water content, ascended beta and NSC as compared to the sham control group. And after the treatment with RIPC, there is a significant decrease in the level of these parameters. Continue. Okay. So in conclusion, we found that there is a the study indicates that RIPC exerts neuropathic effects in a mouse model CLP-induced sepsis-associated encephalopathy by mitigating oxidative stress, neuroinflammation, and various biochemical markers. So, thank you, sir. Sir, RIPC is basically a remote ischemic conditioning, which is pre, post, and post. Pre, per. Yes, sir. Sir, I don't know. Sir, RIPC is basically a normal term is preconditioning before surgery or before disease. Ischemia and
Before the disease.
CLP, yes, sir.
Then why I'm asking only what is the difference between preconditioning and RIPC? Okay. So you have carried out 3 different interventions.
Yes, sir.
You have used only single dose.
No, sir, there is a different dose.
There are 3 interventions, na? Different 3 interventions.
Yes, sir.
Sir, one is capsazepine. What is this?
CGRP compounds.
Okay. Another one is CGRP 837. This is CGRP agonist, antagonist, and then compound C. This
compound is AMPK inhibitor.
Okay. So you want to target 3 different.
Path. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes, sir. Literature. Yes, sir. Control. Sir, that's why we perform the behavioral parameters to check it is affect the brain or not. Yes, sir. Using for the sir, we perform only incision without CLP. Yes, sir. that's why we perform the OFT, NORT, and Moody's water mixture to check it is any changes after the CLP that they affect the spatial memory and recognition memory. That's why we check it out. After that, after confirmation of behavioral parameters, we further move to the next parameters. Spatial memory, yes, sir. Uh, Sir, I know, but
Shreya Saxena from Saraswati Institute of Medical Science, Greater Noida.
Uh, Hello. Good morning to one and all present here. My name is Dr. Shreya Saxena. I am a postgraduate resident in MD Pharmacology from Saraswati Institute of Medical Sciences. Today I'm going to present on AI-driven advancements in receptor pharmacology. So AI has been covering all the sectors today, and now today I'll be talking about drug discovery and design. So the main thing is AlphaFold. In AlphaFold, we can put an entire amino acid sequencing, and it will give us viable proteins in that sequencing the active structures and the activities of those particular proteins. Then we talk about AI-enhanced molecular docking. We all know about molecular docking. With the help of AI, we can make it much more efficiently. Then we do QSAR modeling and molecular descriptions. QSAR modeling is qualitative structure-activity relationship, where we can track the active sites of a receptor with the active sites of a ligand, and then we can understand the relationship between the two. And multiparametric optimization is where we see the permeability and the solubility of different drugs across the membranes, and hence we can increase the activity of certain compounds. Virtual screening and de novo drug discoveries, we have a large database of healthcare systems, and then we can screen the target sites out, and then we can do drug discoveries on specific new targets that are being discovered through the help of AI. Here are case studies on the drugs that are actually been working. DDRN1 kinase inhibitor. DDRN1 kinase was identified to be one of the leading cause of inflammation, cancer, fibrosis. And with the help of AI, this was discovered. And within 21 days of discovery, we have came up with DDRN1 kinase inhibitors. Then SARS-CoV-2 main protease. During the pandemic, there was a search for existing database and what would work at the crucial movement. So we came up with baricitinib, a drug that was used in rheumatoid arthritis earlier to be very effective as an anti-inflammatory during the pandemic, and it was used in the treatment of COVID at the given time. Then we identify CARS G12C to be one of the main mutants that would cause cancer. We identified this particular gene, and through AI we modulated it so that we could inhibit the mutation of this particular gene. Hence, it has been going on in a lot of cancer researches right now. Then we do AI in clinical drug development and safety. We have been very good with predicting ADRs through AI. And we can also predict adverse drug events. There is a software called BioBird. This would help calculate the renal toxicity of different drugs. And we all know Vigibase, that is used to that is used as a database for processing ADRs today. And we can also predict toxicologies of different drugs across the healthcare system. Multi-drug source integration is very important, as existing electronic health records, social media forums, public health records, and bioliterature that is present right now, we collaborate that. A real world problem was opioid crisis in US. We have all heard about this. So where opioid was given as a pain relief management, it would activate two different pathways. That would be one, a G protein coupled pathway, and beta arrestin pathway. Beta arrestin pathway was the main cause of addiction in US. So we have modulated drugs through AI that would only activate the G protein pathway, hence giving therapeutic effects and not any addiction effects. Artificial intelligence in AI in clinical trials and precision medicine. In clinical trials, we would recruit patients in different categories. Like, I'll give you an example. In Alzheimer's, patient changes their nouns to pronouns very early. And our smartphones and our smartwatches can track that. So we would recruit these patients into trials early on so that we can give a better Alzheimer medications at the earlier stage, and we would have much more therapeutic effects then. And we could always predict trial design optimization. We could predict the outcome of a trial before we go in. So our sample size decreases to a lot of extent. AI precision medicine, we can do precise dosing involving the comorbidities, geographical areas, age, weight, and other drug interactions. It has been studied in warfarin in a lot of patients, and it has given much more effective outcomes. Oncological effect, we would identify the target sites of cancerous drugs, and we would give targeted therapies to those particular areas which we know are more prone to mutation, and then these therapies would lead to a much precise and much better outcomes in patients. Pharmacoeconomics, we have genetic variant detection, optimal drug selection, event precision dosing, and clinical efficacy. We involve all these parameters and we inhibit like trial and error method. AI-driven decision making. AI-driven decision making is we identify what is integral in these cases, and then we work on that through regulatory bodies. AI has a great economic impact. The drug research that would take 10 to 15 years is now done in 5 to 8 years. The cost of making drug goes down a lot. And China is a great example of that. They do not have any domestic drug produced in 2017, but where in 2024 they came up with 48 plus drugs that has been used for with the use of AI and development in pipelines. The challenges are still there. There is modern biasness. AI cannot equate all the factors that are there. There is a black box problem where the clinicians don't have an explanation of where the AI is finding its results from. And there is data quality. We do not have that much of input. Right now, they're not from rural areas in different parts. And regulatory gaps is there. Path forward is where we have a lot of success in R&Ds and clinical trials, but we would still, as clinicians and AI researchers, would have to integrate to make a better and safer world for our patients. Thank you so much. So I am just a postgraduate resident. This is my first year, so there is a lot to learn. And this is an emerging field, and a lot of work has been going in this. There has not been any evidence of this coming into clinical trials yet, but it has been proposed. So I have not particularly worked in it yet. Sir, I just got to know about this research that it was a lecture yesterday by a sir. He was telling me that we can detect through AI, we fill a few parameters, like a few symptoms. Say for anemia, it has been going on in Chandigarh. So there is a questionnaire-based program where you identify symptoms of anemia, and you put, you do it as a survey, and you ask the public to fill that survey, and then through that we can see how it is epidemic in a particular area or not. If that is, we can track it through. Sir, I personally think that that was a way to have more preclinical recruitments in that particular field. And we can identify patients early on and then work on it. Sir, I've mentioned one alpha fold, which is very important. We do see protein sequencing, and then we put that protein sequence in alpha fold, we get a structurally similar protein. And if we want to change an analog in that protein, we try to do that. It gives us active sites of different proteins, and then we can work on those. And then there are a lot of softwares that has been developed on the basis of toxicity. So there is BioBot and there is DeepDoc. These softwares would predict the toxicity of different drugs. So while doing a clinical trial, it will be much more efficient for us to be working on that, knowing that this is the level of toxicity. So we would not endanger a lot of patients. Ma'am, the major challenge, most important that I feel like is the gap in data. So even for AI to work, we have to feed him data to get a result. There is a lot of lack of clinical infrastructure back in rural areas. We do not have computerized data in rural areas yet in India as well, and in countries like Africa. So it will be a problem to feed AI that data. Then secondly, it is a new concept, and for clinicians to accept it would take some time. So that I think is also a big problem that has been emerging with the help of AI. Yeah, thank you.
Thank you, Dr. Shreya. Next speaker is Shiv Kumar Saini from CSIR IHBT Palampur.
Good morning, everyone. I am Shiv Kumar Saini, and my today's topic is a benzoic acid derivative protects the brain by preserving the mitochondrial dynamics in zebrafish model of cerebral ischemia reperfusion injury, which is carried out under the supervision of Dr. Davanpreet Singh. So basically, in what is a stroke? Stroke is a reduction in the oxygenated blood supply, in which the high prevalence is ischemic stroke. It is due to the adapting the modern lifestyle. Although reperfusion therapy is given to protect the brain from further damages, still after the reperfusion therapy, many patients loses their lives due to the excessive oxidative stress, inflammation, and mitochondrial dysfunction, including some famous personalities also lost their lives because of reperfusion therapy in recent years. These losses show that only the reperfusion therapy is not itself sufficient to protect the brain. For that, we need some molecule or therapy which is given during the reperfusion. For that, we need to require to understand the pathophysiology behind the reperfusion injury. So initially, what happened in ischemic condition, hypoxanthine was formed, and after the reperfusion, sudden burst of oxygen hypoxanthine reacts with oxygen, and in the presence of xanthine oxidase, it causes then superoxide, which is responsible for excessive oxidative stress, which is mainly due to the down-regulation of NRF2 and up-regulation of DRP1, which is a fission protein, and translocate the cytochrome C into the cytosol, which causes apoptotic cell death, which is majorly causes neuronal death. According to our pathophysiology, we design our objective, which shows first objective is efficacy testing of benzoic acid derivative, that is bezafibrate, in zebrafish larva model. Initially, we check the toxicity endpoints, in which observed that some endpoint like on the basis of OECD guideline, that is 236, and after that we calculate the benchmark dose, that is 50 micromolar concentration. After confirming that, we performed our experiment in which we used then 6 dpf zebrafish larva and exposed in the deoxygenated water for 10 minutes for the hypoxic condition. And for reperfusion, we exposed to in normal water for 4 hours for reperfusion. But in the treatment group, we treated the molecule in the reperfusion therapy within normal water. And we check the locomotion behavior, in which we observe that the disease group shows the locomotion impairment as compared to NAPE. But after the treatment, that is 10 micromolar and 20 micromolar concentration, reverse these parameters. And for further, we selected the 10 micromolar concentration for further experiment. Second objective is molecular mechanism of benzoic acid derivative in our model. Initially, we check the oxidative stress, in which NRF2 gets upregulated in the treatment group, which is a transcription factor which upregulates the antioxidant enzyme. Further, we also check the whole mount DCFD staining, which shows that the high fluorescence intensity was observed in the disease group, but after a treatment, it gets reversed, that is decrease the fluorescence intensity, which shows that it shows an antioxidant property. Further, we check the inflammatory markers, which shows that then pro-inflammatory cytokine, that is NF-kappa B and TNF alpha, gets down-regulated after the treatment. And blood-brain barrier permeability also protected by increasing the expression of the claudin 5, which is a tight junction protein, which maintains the integrity of the blood-brain barrier, which shows that our molecule also shows an anti-inflammatory property and protects the blood-brain barrier. Further, we check the protein expression, in which we isolate the mitochondrial protein and check the expression of an DRP1 and VDAC1, which is an DRP1 upregulated in the disease group, which increases the immature mitochondria, which is responsible for the oxidative stress, which shows that downregulation of an NRF2. After the treatment, this experiment gets altered, and it also shows that the VDAC1 also gets upregulated, which is maintain the mitochondrial membrane potential. Further, we check the JC staining for the MMP and observe that after the treatment, JC gets aggregated into the treatment group, which is indicated by the high red fluorescence intensity as compared to the disease group. And further, we check the tunnel assay for the apoptotic marker and observe that the apoptotic cell death was observed high in the disease group in the form of fluorescence intensity, which is get decreased in the treatment group. It is because of the R molecule inhibit the cytochrome C translocate into the cytosol. Further, we check the new end protein marker, which is new end protein expression in a whole mount in the form of green fluorescent intensity, which is observed that the fluorescent intensity was decreased in the reperfused larva, which gets increases after the treatment. So finally, my conclusion of the study that cerebral iron injury causes excessive ROS, overproduction of ROS, which causes a mitochondrial dysfunction and results into the neuronal death. But after the treatment, bezafibrate combats these all events and shows an protective effect. Then after the promising result, this study also some limitation that bezafibrate needs to require the higher molecule, longer studies will carry out to understand the more molecular mechanism behind our molecule, and higher animal model is required. And third limitation is that immersion of an R molecule doesn't mimic the human pharmacokinetic. So I like to acknowledge everyone and thank you. Yes, sir. So standard dose is DRP1 inhibitor, that is MDI-VI1. I have been using in. No, sir. Yes, sir. It is used clinically, that is MDI-VI1, which is act as an DRP1 inhibitor.
It is clinically used for ischemia?
Yes, sir.
Okay. So you use as a standard?
Yes, sir.
Okay. So you carried out work on the larvae?
Yes, sir. Larva, zebrafish larva.
Okay. So which kind of injury you are focusing upon?
Sir,
Because there is a once there is a cerebral injury, acute, if there is a cerebral ischemia, initially there is an injury because of inflammation and all that.
Yes, sir.
Because of ischemia, then it is followed by excitotoxicity.
Excitotoxicity, yes, sir.
Can you put some light on the excitotoxic mechanism of ischemia reperfusion injury?
Yes, sir. After the reperfusion injury, there is an excessive excitotoxicity which increases glutamate. Actually, this work is ongoing process. I have to carry it out to check the excitotoxicity marker, but according to the reporting papers, it shows that the glutamate gets increases, which is responsible for the excessive excitotoxicity. Yes, sir. Sir, yes, sir. Yes, sir. Yes, sir. Sir, because neurobehavior parameters is in larva, it shows the on the initially we check on the basis on the behavior because it damages the cortex injury during the reperfusion injury, damages which is responsible for the loco. So for mammalian, it is already established model that is Macau, and for that behavior we can go for then like open field test and water maze test, which can mimic reperfusion injury. Yes, sir. In rodents, we use the Bacau and Macau models, which are most commonly used. Um, in my knowledge, that is Bacau and Macau only, and one is ICV also was today also doing that by using the Rose Bengal. It's a Macau model.
Our next presenter is Dr. Sumit Singh from Central University of Punjab, Bhatinda.
Good morning, everyone. Good morning, everyone. I am Sumit Kumar Singh from Central University of Punjab. My presentation title is Combination Therapy Targeting Metabolic Inflammatory and Epigenetic Pathways in Metabolic Dysfunction Associated Steatohepatitis, a Network Pharmacology to Preclinical Validation. So, coming to the background, so steatohepatitis terms define that excessive fat accumulation leading to the inflammation of liver, coming to the hepatitis, and this leads to the MASH, which is also known as metabolic dysfunction associated steatohepatitis, and it is a progressive form of simple fatty liver disease driven by metabolic overload, chronic inflammation, fibrosis, and the epigenetic reprogramming. Over 4 decades have been done in the extensive research has been done to find the drug, and recently 2 drugs, resmetirom and the semaglutide, have been given FDA approval. And although these drugs have come with some toxicity of the liver and gallbladder related side effects. So the present study proposes the multi-pathway therapeutic approach to employing a rational combination of 3 compounds in different molar ratios. Compound 1 is a monosaccharide, compound 2 is modified amino acid, and compound 3 is the epigenetic regulator, more precisely HDAC inhibitor. So our study is designed to simultaneously target the metabolic dysfunction, inflammatory cascade, and the epigenetic alteration simultaneously, and this will address the multi-hit pathogenesis of the MASH. So first we have done the network pharmacology to identify the targets, and the first figure 1A represents the common targets of the MASH and our 3 compounds, and the over 300 targets have been identified, and this is the figure 1B is the PPI network of those common targets. Later this string network was analyzed with the cytohubba, and we found 47 hub genes. So the CAG enrichment pathway was done, and to validate these hub genes, we have done the validation through the geodata sets. That 4 geodata sets are taken, and we have found that 6,172 genes are present in all these geodata sets, for all these 4 geodata sets. And later we have overlapped our 47 hub genes with these 6,172 genes, and we found that 18 of them is present in all the geo datasets as well as in my hub genes also. This is the heat map expression of those hub genes. So coming to the in vivo section, we have taken C57BL6 mice, and the total study was for 18 weeks, in which 12 weeks was for model development using high fat and high fructose diet, and later 6 weeks was for the treatment with the formulation of A, B, C, and D. The formulation A, B, C, and D consist of those three compounds in different molar ratios. After the sacrifice, this is the representative image of the liver of mice, and we can see clearly in the MASH group there is excessive fat accumulation in the liver, leading to the whitish coloration of the liver. So we have done the liver function test and the glucose profile test. In the liver function, we have checked the SGPT, SGOT, and the ALP level. And in the glucose, we have in the glucose profile we have checked the fasting glucose, insulin level, and the HOMA-IR. And we have found that these were significantly increased in the MASH group. And after the treatment, it was significantly reduced in all the formulations. Later, we have checked the lipid profile, in which triglyceride, cholesterol, LDL cholesterol, and the hepatic TG was was checked, and in the MASH group, these were significantly increased, although the HDL cholesterol was significantly reduced in the MASH group, and after the treatment, it was reversed. Later, we have checked the relative mRNA expression to check the what molecular changes have been done, and we have checked the SRBP1C, CHRBP, HMG-CR FASN, and the ACC1, which are the lipogenic genes involved in the de novo lipogenesis. And in the MASH group, all these have been relatively significantly high, and after the treatment, it was significantly reduced. We have also checked the epigenetic changes, and we have identified that the HDAC 7, 8, and the 11 is relatively high in the MASH group as compared to the control, and after the treatment it was significantly reduced. We have also done the histopathological analysis, and in the MASH group it is the micro and the microcytosis has been majorly in the MASH group, and after the treatment it was reduced. Notably, we can see that this formulation C has more effect as compared to the formulation A, B, and D. Conclusion, our study support the efficacy of rationally designed multi-targeted combination therapy that targets metabolic, inflammatory, and the epigenetic pathways of the MASH. Thank you.
Okay, again, quite a interesting body of work. Thank you for that. You said you have used a high-fat, high-fructose diet for 6 weeks for inducing MASH.
Sir, for the 12 weeks.
12 weeks?
Yes.
Okay, is that a standard model for?
Actually, in our lab, it is standard. We have already standardized that model in our lab. Like 12 weeks is sufficient to develop the MASH model. And for that, we have already checked the parameters liver function test at the 10-week, at different time intervals.
But does it give in 100% of the exposed animals MASH?
Almost 100% animals. Almost 90 to 100% animals have high biochemical SGPT, SGOT liver function test.
Okay, and what was your rationale behind including a histone deacetylase inhibitor as one of these anti-MASH agents?
Thank you, sir, for this question. Actually, what happens that in the MASH pathophysiology, the epigenetic changes has been underlooked. So we have included the HDAC because HDAC 7, 8, and the 11 are responsible in the fibrogenesis and the lipogenesis of the genes in the liver.
So that is still not yet established. No? Those are hypotheses which are still being explored.
Sir,
these epigenetic changes which you are referring to, their contribution to the development of MASH is still under exploration.
Sir, actually there are several publications in which 7, 8 is already being targeted to mitigate the MASH.
Okay.
And HDAC6 is also targeted, sir.
Thank you. Did you monitor body weight of the animals as well?
Yes, sir.
You didn't give the data.
Actually, some data has been not shown due to lack of space.
So what
We
was
have
the
shown
starting-
the liver weight to the
You
T-bone.
incorporated, you took mice of very specific strain. What is this mice special about?
Actually, sir, this specific mice is used in the metabolic disorders because there is it is more susceptible to the obesity and the metabolic disorders. So that's why we have chosen the C57BL6.
So where did you cure the animals?
Sir, from Naipur Mohali.
It is available in the Naipur?
Yes, sir.
So you employed 6 to 8 week old mice.
Yes, sir.
So what is the average weight of these mice once you incorporate in the study?
Sir, it was like 18 to 20 grams when we have procured. Actually, what happens, sir, our
18 to 20.
Yes, sir.
So once you started administering the drug, what was the last weight?
Sir, the maximum
In
weight
HFD, fructose high food.
the range was 35 to 42 gram.
After 12 weeks of HFD.
After 18 weeks, sir.
After 8.
For at yes, sir.
So you administered the HFD up to 12th week
Yes, sir.
and you continued up to 18th week.
Yes, sir.
But अभी आपने बताया 12 week का भी सबको तो 12
Sir,
week
for
बता
the
रहे।
model confirmation, first we have developed the model,
Okay.
and later we have give the treatment.
Okay.
But the high fat diet and high fructose was continuously to the 18 week till 18 week.
And what was the weight of the treated animal?
Sir, it was reduced to 30 to 35. Actually,
It was significant reduction or it was
no,
not
sir,
significant?
not significant reduction.
And you didn't mention here.
No,
Anyways.
sir, actually, in all the formulation
You give reference of 2 very important drugs. One of which is very a blockbuster molecule these days, Ozempic.
Yes,
What is
sir.
that Ozempic?
Sir, semaglutide from the Novo
What,
Nordisk.
why I said it's a blockbuster molecule these days?
Sir, actually, it is already used to reduce the weight, obesity.
So you very specifically mentioned, but you didn't present the data of your weight.
Sir, actually, when the protocol was designed, it was
Okay.
not FDA approved for the
Okay.
mesh.
Okay.
So we, to address this issue, we already are doing obesity model in our lab, and we are using this same formulation.
But it's a very important parameter, the study that you are
Yes,
carrying on, and you did not present. Anyways,
Okay, what is the full form of MASH?
Sir, metabolic dysfunction associated steatohepatitis.
And what is NASH?
NASH is the former name of this disease, and
What
it
is
was
the full form?
non-alcoholic steatohepatitis.
And why it has changed from NASH to MASH?
To tell that non-alcoholic is not right, because ma'am, they to categorize that disease is not the right way to set the non-alcoholic, because people there is met ALD also, like metabolic dysfunction is also present, and person is also consuming alcohol. So there is need to change the terminology also, and I think that NASH term was not appropriate, non-alcoholic term was not appropriate.
Okay, that was not, but could you explain why it was not appropriate?
Ma'am, there was only two terms previously, NASH and NFALD, non-alcoholic fatty liver disease, and later the progressive form was called non-alcoholic steatohepatitis. But there is also condition that a person consumes alcohol and person is also on high-fat diet. So there was need to change the
Only
overall
high-fat
name.
diet?
Yes,
Only high-fat diet was the reason?
Western diet, more precisely Western diet.
Okay. Thank you.
Thank you.
Our next presenter is Dr. Rohan from BITS Pilani.
Hello, good morning. Okay. Hello, good morning to all. My name is Rohan Badange. Here I am presenting the research work entitled as Ambrisentan inhibit the AKT-GSK signaling axis to mitigate chronic kidney disease. So talking about the introduction, so chronic kidney disease is manifest as a progressive impairment of the renal function, and that leads to severe health complication, and with prevalence is about 14%. That account for about 850 million people are suffering from chronic kidney disease globally. So there are a number of medication or the therapeutic option which are available to combat the chronic kidney disease, which include AC inhibitor, angiotensin receptor, then angiotensin receptor 2 blocker, then mineralocorticoid receptor antagonist, then some anti-inflammatory and anti-fibrotic agent. Although these therapies are available, but they're often targeting the symptomatic management, and they are come up with a serious side effect. Hence, that restrict the potential use in the long-term treatment. Hence, drug proposing this one of the strategy where the cost-effective and practical approach that can be employed for the treatment of chronic kidney disease, since the pharmacokinetic, pharmacodynamic, safety and efficacy data of the drug which is already available in the market. So ambrisentan is one of the selective endothelin receptor antagonist, which is already there in the market to treat the pulmonary arterial hypertension, and that has been shown to have the renoprotective potential in the CKD. So based on our hypothesis and conceptual analysis, we propose that the ambrisentan may mitigate unilateral ureter obstruction induced renal fibrosis in spec dolly rats, and recombinant human TGF-beta, that is RAT-TGF-beta, induce fibrosis in NRK52 cell line, that is rat normal kidney cell lines, by modulating the AKT-GSK signaling axis. So this is our experimental design, which is divided into two parts. The first is the in vivo study, and the second is the in vitro study. In vivo study, we have employed the male spec dolly lot with n is equal to 6 animals in per group, and we have employed the 3 group. That is, one is the sham control group. That is, the second is the unilateral ureter obstruction group. That is the disease control group, and the last is the treatment group, which include UO plus ambrisentan, that is AMB, with a dose of 0.5 mg per kg per oral for per day. That is given for 21 days. Post 21 days, we have sacrificed the animal, and the plasma, urine, and kidney sample were collected, and they are employed for the biochemical, histological, immunohistochemistry, and the qRT-PCR analysis. Further, we also employed the in vitro study, where we have employed the rat kidney tubular epithelial cell line, that is NRK52E cell line, and which were divided into 3 groups. That is normal control group, the normal control with higher TGF-beta, which is again to mimic the fibrotic condition, and the last is AMB group, that is ambrisentan group, with a 10 micromole dose for 48 hours. Post 48 hours treatment, we have collected the sample, and which were further analyzed for the cell viability, morphology, FACS analysis, as well as immunocytochemical assay. Talking about the result section, now we have checked the effect of the basic parameter, that is the kidney function analysis, as well as the histopathological analysis. So here we can say the obstructed kidney that must have that increase the kidney weight and kidney weight to body weight ratio, and we have also performed some basic histopathological analysis, that is plasma creatinine, urinary creatinine, BUN, and EGFR level. So as we can see, the plasma creatinine level and the urinary and the BUN level, there is a significant increase in the upregulation of this creatinine and BUN that has been reported, that has been noted in case of disease group compared to the sham group, and that's level that has been significantly restored upon this ambrisentan treatment. Similar case that has been observed in case of urinary creatinine as well as EGFR level. So renal fibrosis often characterized by initial stage, it is characterized with the histopathological alteration. So as we can see the HNE staining, which is, we can see the tubular dilatation or the flattened tubular epithelial cell that has been reported in case of UU, that is disease control group, which is shown by the yellow arrow. When compared with the sham group, there's a flatten and the dilation of this epithelial cell that has been reported, and there is significant improvement of this or the restoration of this epithelial cell that has been reported in case of AMB treated animal. Similarly, the past staining, that is picrosidase periodic acid staining, that is used to evaluate the glomerular sclerosis score or the dilation of the Bowman capsule that has been that can be shown by this past staining. So as we can see, the UO group, there is a marked dilation of the glomerular or the Bowman capsule that has been shown by the yellow arrow, which has been elevated, the dilation that has been reported when compared with the sham group, and that this dilation of this glomeruli which has been restored upon the AMB treatment. Then we have used the AMB, we have used the NRK52 cell line to evaluate the efficacy of this AMB, that is ambrisentan, in case of NRK52 cell line. So here we have employed, we have performed the basic cell viability and the cell morphology study. So based on this study, we can see that the 100 micromole dose that has been shown safe and effective concentration of this dose that has been reported. And we have went with this 100 micromole dose concentration of the emrysandin in a subsequent experiment. So here we can see that the AKT GSK, this is one of the signaling axis, and that is often reported to produce the renal fibrosis in NRK52 cell line as well as in renal fibrosis model. So here we can see the AKT, that is the phospho-AKT and GSK level that has been elevated in case of IRTJ beta induced NRK52 cell line, and that has been significantly restored upon AMB treatment. Similarly, we have performed the apoptosis, that is by using FACS analysis, and here we can see the IRTJ, the number of apoptosis cell in IRTJ1 TJ beta 1 group that has been increased compared with the normal control group, and that has been significantly restored upon the AMB treatment. Similarly, we have performed the QRT-PCR analysis. Here the pro-apoptotic marker, that is BAX, and the anti-apoptotic marker, that is BCL2, that has been checked, and we can see the M1 expression level of this pro-apoptotic and apoptotic marker that has been significantly restored upon the AMB treatment. Then we also check the EMT level, where we can see the epithelial cell is converted into the mesenchymal characteristics, where the fibrosis process begins. And here the alpha SMA vimentin level that has been significantly upregulated in case of disease control group, and that has been significantly restored upon the AMB treatment. Similarly, the E-cadherin level, where the epithelial cell loses its epithelial characteristic, and then they are converted into mesenchymal characteristics. So here again we have seen the again fibrotic marker, that is fibronectin, and the empty staining, that deposition, the collagen deposition that has been reported, and our results suggested that the ambulation treatment significantly restored this level. From the conclusion, we have seen that the therapeutic potential of the emrysandin in vitro in vivo model, and here we have checked it by using the kidney function test, then the histopathological analysis, as well as some fibrotic and apoptotic marker that has been reported, and by modulating this AKT-GSK signaling. However, further preclinical and clinical investigation is necessary to investigate the potential of this emrysandin in treatment of chronic kidney disease. Thank you.
So thank you, Rohan. You employed male rats only. So this chronic kidney disease is confined to male?
No, actually, concerning about the experiments, so as we know that when we are performing surgery in the male rat, that is easy to locate the ureter. So as we can see this diagram, so here we can see the diagram, that is the ureter, the left ureter of the animal that has been ligated, and it is easy to locate in case of male animal, since the reproductive system can be easily.
This is the only reason that is easy to locate the ureter?
As far, I mean, we can see.
But otherwise, it is wise to use equal number of male and female both.
Actually, the study reported the male mice, so based on literature survey, and based on the experimental feasibility, we have employed the male mice in the current.
Okay, so you use unilateral ureteral obstruction for inducing your disease. What are the other models of chronic kidney disorders?
There is adenine-induced CKD model. Then there is a this is basically adenine, which is a chemical molecule.
So I'm again coming to my previous question. Yeah. If there are other models available, why then need you opt out for this model only?
We also have the 5-6 infectomy model, which is surgical model. We have employed this UO model since we are going to check the renal fibrosis. The renal fibrosis, the pathological condition which is found in the CKD, and we have to target this condition, which is a pathological driven, which is caused by the TGF beta. Hence, we are particularly focusing on the fibrosis. Hence, the UO model is well-established model in the literature. We have employed this model. It is also established in C57 mice. We have employed this SD rat in this case.
So you used endothelin inhibitor, emrysandin.
Yeah.
So what is the role of endothelins in chronic kidney disorders?
This emrysandin is ET1 receptor, specifically endothelin receptor type 1 receptor antagonist. So endothelin type 1 responsible for vasoconstriction. Then further, because of the vasoconstriction, it may be responsible for the hyperfiltration, podocyte damage, then leukocyte migration, inflammation, and that further progress the renal fibrosis in the CKD.
Is there any role of endothelial dysfunction in chronic kidney disorder?
Sir, I'm not getting.
Endothelial dysfunction. Is any endothelial dysfunction is related to chronic kidney disorders?
Yeah, actually it is related. So that's why, I'm not getting, but I have explained the previous question. I'm not clearly understanding the question actually.
Okay. So you carried out some cell viability assays for,
yes, yeah.
आपने कौन सा किया है?
So we have
XTT assay.
Yeah, XTT assay.
इसके बारे में थोड़ा सा बताइए.
Again, we have employed the cell. We have again grown the cell in the 96 well plate, and again.
I'm asking about the principle of this assay.
Okay. So the formation crystal which is formed at the end, that is after treatment period, we are adding the XTT. The one is substrate and the reagent, and here we are added, and after 4 hours the formation crystal that has been formed. And this formation crystal that has been solubilized, that's why we are not using DMSO. In case of MDT, we are using DMSO since the formation crystal which is formed, which is not solubilized. So the number of cell, what we can say, the percentage, we have calculated percentage. So the number of viable cell which is shown by the formation crystal formation that has been reported and that has been noted and then again recorded by using the spectrophotometric analysis.
Okay, Rohan, first of all, congratulations. We are getting very high quality work presented in this round. So my question on the lines of what sir has been asking, if you look at the burden of human chronic kidney disease, only a small proportion are due to primary renal disease or renal vaso-occlusion. Majority are secondary to, let's say, diabetes or hypertension. So how far is this model of unilateral ureteral obstruction relevant to that kind of chronic kidney disease?
Okay, so CKD caused by other complications you're talking.
CKD is mostly the result of some other primary disease, majorly diabetes and hypertension.
Yes.
Only a small proportion is due to primary renal disease or renal vaso-occlusive disease. But your model is mimicking vaso-occlusion, ureteral obstruction. Ureteral obstruction. So how far is this model relevant to the majority of human chronic kidney disease?
So talking about the diabetes, so diabetes is a diabetic kidney disease. So CKD is also again formed by, CKD is a form of, DKD is a form of CKD. Means DKD is coming under the.
So if you look at the pathophysiology of DKD, so fibrosis is there, but more important it is tubular necrosis and glomerular injury.
Okay.
So does this model mimic those changes? I don't think it does.
It is mimicking actually in DKD, it is caused by the diabetic kidney disease caused by estrogen-induced diabetes, hyperglycemia. In this case, caused by the obstructive nephropathy model.
You're mainly focusing on the fibrosis aspect.
Yeah, that's why we utilize this UO model, that is unilateral ureteral obstruction model in rats.
But there's a discord, no? How does it replicate the?
This is again diabetic nephropathy, DKD and CKD. This is one answer. Second answer could be the AKI, that is the acute kidney injury that may leads to AKI to CKD transition, that further progresses CKD.
That of course for a small proportion of CKD.
Yeah, actually.
Okay, madam.
Our next presenter is Dr. Kanchan Kalra from Government Medical College and Hospital, Chandigarh.
Good morning all. I'll be presenting my study on which is entitled Exploring Biliary Tract Adverse Events Linked to GLP-1 Agonists and Tirzepatide, a real-world disproportionality analysis. A real-world disproportionality analysis from the US FDA Adverse Event Reporting System, FAERS. Starting with a little introduction, GLP-1 receptor agonist and tirzepatide, which is a GLP-1 as well as GIP analog. These are used for type 2 diabetes mellitus, obesity, and for cardiovascular protection also. And the key agents include semaglutide, dulaglutide, lixisenatide, tirzepatide, and the emerging evidence has indicated that there is an increased risk of biliary adverse events associated with their use. And the proposed mechanism includes two pathways. Firstly, it can be hormonal, which says that decreased levels of cholecystokinin can lead to impaired motility, which can further lead to stasis and further leading to biliary cholelithiasis. And the second theory says, the metabolic one, that there's a drastic weight loss associated with these. So that weight loss can lead to increased cholesterol load in the bile, which can lead to a supersaturation of the bile, ultimately leading to cholelithiasis. And various clinical trials have also supported this association. The SCALE trial, in which it was seen that liraglutide was compared with placebo, and it has shown cholelithiasis in some patients. And similarly, semaglutide obesity trial, it also showed cholelithiasis in 2.6% of the patients. In other trials at PASS 2, both tirzepatide and semaglutide groups, they reported cholelithiasis. And in another trials like SURMOUNT-1, SURMOUNT-CN, they also noted higher rates for tirzepatide. Further, the leader trial, it included over 9,000 patients, which found liraglutide increased the acute gallbladder and biliary disorders with a hazard ratio of 1.60, which is significantly higher. Although these clinical trials suggest a potential safety signals, a real-world data remains scarce. Therefore, we conducted a disproportionality analysis using the FAERS data to assess the association between GLP-1 receptor agonist and the biliary disorders. The methodology included, it was a retrospective pharmacovigilance study design using the FAERS data. We included quarter 4, 2003 to quarter 2, 2025 data. The tool used was OpenVisual 2.1 software for disproportionality analysis. It is used to detect safety signals, to compare the drug profiles, and for adjusting the confounders. The drugs included were liraglutide, semaglutide, exenatide, dulaglutide, lexenatide, and tirzepatide. Biliary adverse event terms, cholelithiasis, acute cholecystitis, and cholecystitis, and biliary colic. They were identified to MEDRA lowest level terms. It included only primary suspect cases. We excluded any secondary or concomitant cases. And the duplicates were removed in Excel using advanced filter to retain the unique case IDs. And the software, I've already told, OpenVisual 2.1 was used, and we used MS Excel for the deduplication of the data. And ethical approval was not required, as FAERS uses publicly available anonymized data. Now, the association between GLP-1 agonist and tirzepatide and association with biliary tract events were evaluated using standard disproportionality matrix, PRR, which is proportional reporting ratio, and the ROR, which is reporting odds ratio, which is commonly applied for signal detection and pharmacovigilance. Both these metrics were derived from 2x2 contingency table, like this table was made, and which A value says the report of biliary adverse events with a drug of interest, B says report of other adverse events with a drug of interest, and C says the report of biliary adverse events with all other drugs, and D is report of other adverse events with all other drugs. And these signals were assessed for significance using Evans criteria, which says that PRR should be more than equal to 2, and chi-square value should be more than equal to 4, and N is the number of case reports, which it should be more than 3 to say it a significant one. And these were our results. The disproportionality analysis of biliary tract adverse events. In the drugs, we can see semaglutide, tirzepatide, exenatide, and liraglutide. It was the association evaluation per Evans criteria was significant, whereas it was non-significant in the case of lexenatide and dulaglutide. The limitations of our study can be a reporting bias and the underreporting, because FAERS is a kind of spontaneous reporting data. And confounding factors can be there. There can be disease severity can matter, and any concomitant medications can also be there. And thirdly, it can be the signal which we are talking about. It is just an hypothesis generating. We cannot confirm it. The causality is not established. We are just detecting the signals. And fourth can be generalizability concerns, because FAERS mostly contains the US data. I'll be concluding my presentation. The study highlights a potential association with GLP-1 receptor agonist, particularly liraglutide, semaglutide, exenatide, and tirzepatide, and biliary tract adverse events in the real-world data. The clinicians should weigh the risk-benefit profile while prescribing these patients, especially for the individuals who are already having any gallbladder-related problems. Further mechanistic and prospective studies are needed to validate this signal and inform risk mitigation strategies. Thank you. Yes, ma'am. Ma'am, sema and tirzepatide. Ma'am, semaglutide, it basically acts on GLP-1 only. And the tirzepatide, it acts on GLP-1 as well as GIP-1. So it has a dual mechanism. There's one more drug has come, retatrutide. It is a triple agonist, GLP-1, GIP-1, and one more is there. Unable to memorize. And because it is not an established side effect, मतलब established नहीं है, जैसे pancreatitis है, injection site reactions होते हैं GLP-1 के, मतलब gastrointestinal adverse effects, they are already in the जो FDA के accordingly जब drug approve होता है उसके जो side effects profile देते हैं वो उसमें होता है. It is a rare, हम इसका सिर्फ जैसे post marketing surveillance, we are seeing in how many cases it is, मतलब it is significant or not. कि कितने केसेस में अगर आ रहा है इट इज मैम मैम इन द रेयर वन वी आर जस्ट सीइंग द सिग्निफिकेंस ऑफ दैट सिग्निफिकेंस. Ma'am, it is higher because many trials have been conducted nowadays. These drugs, many studies are going on with associated with these drugs.
So, do we have access to the FAIRS data here?
Yes. Yes, sir.
Okay. The hypothesis with which you started your study, that probable mechanistic reasons why these drugs can be associated with biliary tract disorders,
Yes, sir.
and you're finding associations. They're not necessarily causal associations, they're possibly signals only. But for two drugs, dulaglutide and lixisenide, you have not got any association. Can you explain that?
Yes, sir. There can be varying receptor affinity in their case, and the potency can be different for those drugs. And the number of cases was also less because they were given in less number of patients.
Okay.
The N value was less for them. Like in lixisenatide, only 6, the frequency of biliary tract adverse events was only 6. And the number of other adverse events was 2476. So this was less.
Okay, so it is less, but that's because the overall use also is less, means
Yes.
number of documented cases is less. So could it be that this is not a class effect of GLP-1 receptor agonist?
Sir, that thing I'm saying that we are not confirmatory about it. We are just saying that these signals are significant. And we have a probable mechanistic theory associated with these also.
Okay.
Okay, thank you.
Our next presenter is Dr. Satnam Singh from AIIMS, New Delhi. We are running short of time. Please try to finish as early as possible.
Good morning to all. My topic of presentation is beyond iron intake, multifactorial role of stress, aluminium, vitamin D, and essential elements in iron deficiency anemia. So, I would like to disclose it is a ICMR funded trial, and as an introduction, iron deficiency anemia (IDA) is a global burden which affects over 1 billion people by its severe symptoms such as extreme fatigue and weakness, shortness of breath, chest pain and tachycardia, pale skin, hair fall and many more, which affects the quality of life. WHO recognized it as a predominant form of anemia. Emerging evidence suggests that there is the stress, aluminium exposure, vitamin D status and essential elements may influence erythropoiesis and iron metabolism. However, the relationship between these factors with IDA remains still underexplored, and the evidence supporting the benefit role of vitamin D and essential element supplementation in IDA is still limited. So this study was designed to evaluate the stress levels, exposure of aluminium, vitamin D status, and essential element profiles in IDA patients at baseline. Additionally, the study compares the therapeutic effect of vitamin D and essential element supplementation alongside standard IDA treatment within the same cohort. To best of our knowledge, this is the first trial in India to assess multiple baseline factors and their impact on treatment response in IDA. So, comes to methodology, this is a quasi-experimental and controlled treatment trial which is being conducted in the Department of Hematology and Medicine at AIIMS, New Delhi, spanning the period from August 2022 to July 2025. The minimum sample size was 44 per group, which were required to determine the median effect in primary marker hemoglobin with 80% power of the study. This study had 3 phases: patient recruitment, baseline evaluation, treatment phase 90 days, and follow-up after 90 days final evaluation. The intervention which was given in the study first is standard IDA treatment, which involves the combination of iron, vitamin B, and folate therapy. Next intervention is vitamin D supplementation, 60,000 international units for 8 weeks. Essential elements, magnesium, zinc, copper combined in all the essential elements combined in a single tablet were given. So lastly, statistical analysis in which paired t-test, unpaired t-test, Mann-Whitney, ANOVA, Kruskal, and ANCOVA, Pearson, Spearman correlation, multivariate, univariate regression, and ROC curve analysis was performed. So coming to the enrollment of the patients, 1873 patients were assessed for the eligibility criteria, in which 390 patients were included in this study based on the inclusion criteria mentioned, and 1483 were excluded from the study. After baseline evaluation, all the patients were allocated for the specific treatments, in which group 1 received standard IDA treatment, group 2 received standard IDA treatment plus vitamin D supplementation, group 3rd received standard IDA treatment plus essential element supplementation, and group 4 received standard IDA treatment plus vitamin D plus essential element supplementation. So after 90 days follow-up, all the patients were reassessed, and 1 patient from group 1, 3 patients from group 2, and 1 patient from group 4 were excluded from the study due to loss to follow-up and discontinued intervention. And lastly, final evaluation for statistical analysis evaluated 385 patients. Further, I would like to mention that stress was determined in the patients at baseline with perceived stress score comprising of 10 questions, and blood parameters such as hemoglobin, hematocrit, RBC count, and iron markers, along with all the essential elements mentioned, were measured both at baseline and final. So coming to the results, hemoglobin, there was significant presence of iron deficiency anemia in patients marked by low hemoglobin and ferritin levels. There was coexisting deficiency of vitamin D. Also, copper, iron, and magnesium level was also less in significant populations. Next is there is a presence of microcytic hypochromic anemia determined by low MCV, hematocrit, and increased RDWCV. And there is a decrease in hepcidin also. And lastly, the trace element aluminium was significantly high in all the study population. Next is the prevalence and impact of stress in IDA patients. So coming to the stress part, 75.1%, 290 subjects had experienced high stress in the study cohort. Stress was negatively correlated with hemoglobin. At baseline, patient without stress had significantly higher hemoglobin compared to the non-stress patients. Further negative association was also observed with other parameters. Next is impact of aluminum exposure in IDA patients. So aluminum was significantly high in patients, paper population. Almost 90% subjects utilized aluminum-based cookwares and had significantly higher levels of aluminum. Aluminum negatively correlated with hemoglobin and vitamin B12. Ferritin was significantly high in patients with low aluminum levels, which was defined by ROC curves. Prevalence of vitamin D status in iron deficiency anemia. 71% population had significant coexisting vitamin D deficiency. Vitamin D showed positive correlation with hemoglobin and ferritin and other parameters such as B12 and folate. At baseline, subject with high vitamin D had significantly higher levels of hemoglobin and ferritin levels compared to the patient who has low vitamin D, or you can say vitamin D deficiency. Last is prevalence of essential elements status in iron deficiency anemia. So copper, magnesium, iron was significantly below. Multivariate regression shows that hemoglobin has significant moderate association. MCHC hematocrit has slightly moderate, and further copper and iron was positively associated with hemoglobin and magnesium with hematocrit. So last is effect of therapy and supplementation on IDA. So after 90 days, within all 4 groups, hemoglobin and ferritin was significantly improved from baseline. After adjusting for the baseline, and COVA finds no significant difference in among all 4 groups. However, the maximum subjective improvement of patient overall well-being was observed in group 4 in terms of symptoms and their biomarkers. So in discussion in overall IDA patient, there was a varied clinical syndrome was observed, which was significantly improved after 90 days. There was significant coexisting deficiency, vitamin D, copper, iron, and magnesium was decreased. High stress increase was markedly correlates with low hemoglobin and ferritin. High aluminum was significantly associated with low hemoglobin and iron, which was decreased after 90 days of iron therapy. The low vitamin D status was significantly linked to disease severity, and low essential elements were there in overall cohort. And after 90 days therapy, it was very safe and well tolerated with no adverse reported side effects. A few patients experienced mild indigestion, which was effectively managed without requiring any change in therapy or additional medication. So in conclusion, to best of our knowledge, this is the first trial in India to demonstrate breakthrough findings through a combined regimen therapy and supplementation approach gives the highest improvement in hemoglobin, ferritin, and other hematological markers. Effective recovery and therapeutic response in IDA patients requires more than iron supplementation alone. Optimizing vitamin D therapy, correcting essential element deficiencies, and integrating stress management therapies can be a new guiding strategies for the comprehensive management of iron deficiency anemia. However, these findings should not be generalized to all IDA patients, and nutritional supplements should be prescribed without prior assessments. With this, I would like to end my presentation. Thank you.
Okay, Dr. Satnam, thank you for presenting your work.
Yes.
Several questions arise. The ones which immediately come to my mind is, number one, why you are calling your study a quasi-experimental study. Number two, your calculated sample size is 44 per group, but still in some of the groups have included more than 160, 130 patients. Why is that? That is not how clinical trials are done. Third, your multimineral formulation. How is a formulation like that marketed, or did you get it manufactured de novo? So these are 3 questions which immediately come to mind.
So I forget your first question, sorry.
Why are you calling it a quasi-experimental study?
Quasi-experimental study, because sir, it is a 2 time point study. What we have observed, we have first recruited the same cohort. And
did you randomize or not?
Yes, sir. We have not randomized.
So that introduces a lot of bias, na?
Yes, sir. But the study design does not allow us to randomize the patient, because we have first evaluated at them baseline. We have then after the baseline evaluation, because all the patients are IDA, and we do not have control group, and among IDA we have considered a single control group in which only iron basic IDA standard therapy was given, and rest other groups were compared with the same group.
When you are doing a therapeutic trial,
yes, sir,
you have to have strong reasons why you don't randomize, but this does not justify those strong reasons. Number two, if you are planning to include as many patients as you can get,
yes, sir,
then what's the point of doing a sample size calculation?
Okay, sir. Sample size calculation was only determined for the baseline to final evaluation of hemoglobin. That is medium effect. Sir, for small effect, we had a sample size of 120 patients in each group, but we could not able to get it in what we have proposed in timeline. So there was only 60 and 60 in group 3rd patients, sir.
But then you shouldn't be showing a sample size calculation of 40.
So for significant difference to find at 90 days within groups, between groups at 90 days, it requires 120 samples per group. That is why our samples, results are not significant. However, so if you can see, there is in 4th group, if we comparing group 1 versus 4, the p-value is 0.07. It is not significant, but still there was a significant, there was improvement in last group.
In several of your associations, you have shown that we have found significant associations or positive associations,
yes, sir,
whereas the correlation coefficient values are hardly exceeding 0.4.
Yes,
in fact, some of them are 0.3 onwards. So these suggest very poor correlations.
Yes, multivariate we have performed multivariate regressions, and they were...
No, you have performed Spearman's and
yes,
Pearson's correlation.
Spearman and Pearson were was only performed with aluminium and stress levels.
But those values, you see, you're getting R values of 0.38, 0.32. These doesn't suggest good correlations at all. You can look at your scatter plots.
So this is not a strong correlation, but beyond the value of R.
But you have showed positive correlation.
Yes, sir, positive correlation was there. But with the value of...
I would say rather there's no correlation. If we look at the scatter plots, there's no correlation.
But sir, we have observed significant correlation. Stats value was there, but...
Even your P is non-significant. See, the first P is
point. This is not a strong correlation, and this is not a strong predictor.
This is neither strong nor good nor fair. It's approaching poor correlation. You have to have at least 0.5 to say it's a good correlation. Anyway, okay.
Yes, sir. Yes, sir. So stress they have already from their lifestyle. Actually, perceived stress score measures their lifestyle stress. It's a set of 10 questions, which comprising of total 40 scores. And above 16, we can say there is a moderate stress, and above 24 there is a high stress. So based on that calculation, we have considered two categories, with stress and without stress. So the stress was already, which they are facing from their lifestyle, were measured. It's a no, sir. Stress was only measured at baseline. We did not manage through any therapies in this trial. Yes, sir. Ma'am. Yes, ma'am. PSS 10 and PSS 14s are internationally accepted perceived stress scores to measure perceived stress in the patients. PSS 10, ma'am. Yes, ma'am. Thank you.
Thank you, Dr. Snau. That concludes the GHRE Prize session for neuropharmacological research. A big round of applause for all presenters. We are honored to have our chair, Dr. Nirmal Singh, Dr. Avijit Hajre, and Dr. Sangeeta. We now take this moment to express our sincere gratitude to our chair. Please come on the stage and have a seat. Sir, please come. Now I request Dr. Manish Jain to kindly felicitate them with a token of appreciation. For Dr. Nirmal Singh. To Dr. Avijit Hajre. Please, sir, please have a seat. To Dr. Sangeeta Yadav. Sir, please have a seat. Now I request to all presenters, please come nearby stage. We will have together a picture with you. Please come.
So, first of all, let me congratulate all the participants. Indeed, the quality of the research work that they presented for Acharya Oration Award, just wonderful. They are carrying out excellent work. It hardly matters who won the prize. There will be a slight difference. So all of you are excellent researchers. Keep going good work. And I wish all you for your best for your bright future.
So all the participants, presenters, please come for a group photo. Come on stage.
Thank you everyone for joining this vibrant session. Now we are going to start our next session, Audiogularity Prize session. Please be seated. Hello. Thank you, ma'am. Thank you. Okay, sir. Thank you, sir. Hello. Thank you, ma'am.
Okay, last minute.
Sir, carry bag.
Achha, your original judges have come?
3 judges are there.
Then it's okay. I've already judged 2 stations.
I have to be in another session.
Okay, sir.
So, तीनों आ गए ना? बस.
Thank you, sir. Thank you so much. Now we are going to begin our next session, Audi Gulati Prize session. Good morning, respected judges, esteemed delegates, distinguished faculty members and fellow researchers. A very warm welcome to the Audi Gulati Prize session for oral presentation in neuropharmacology basic science. Here at IPSCON 2025 being celebrated under the theme Empowering Young Minds to Transform Pharmacology for Viksit Bharat. This prestigious session brings together pioneering research that explore the fundamental mechanism of the brain and nervous system, from synaptic signaling and receptor dynamics to novel molecular targets of neuroprotective strategy. Today's work captures the depth and innovation of modern neuropharmacology. This session honors the memory of Professor Omdat Gulati, a pioneer in autonomic pharmacology whose contribution laid the foundation for cardiovascular, autonomic and autocoid pharmacology in India. The Audi Gulati Prize recognizes the scientific excellence, creativity and impactful contribution in these disciplines. We are now delighted to welcome the presenters whose remarkable research reflect this spirit of innovations. A big round of applause for them. We are privileged to have with us our esteemed chairs, Dr. Puneet Bansal. It is a pleasure to introduce Dr. Puneet Kumar Bansal, PhD, a leading neuropharmacologist from Punjab and senior faculty member at the Central University of Punjab, Bhatinda. A former head of the Department of Pharmacology, he is renowned for his work in movement disorder, neurodegeneration, behavioral pharmacology, and natural product-based neuromodulators. A graduate of Punjab University, Chandigarh, Dr. Bansal has over 200 publications with high citation impact, contributing significantly to Parkinson's disease, Huntington disease, anxiety, depression, and cognitive dysfunction. His expertise include animal behavior model, neurochemical assay, oxidative stress biology, and molecular pharmacology. He has guided several PhD and M.Pharm scholars with funding from national agency. Welcome, sir. Now we are honored to introduce our next chair, S.K. Bandyopadhyay, one of the West Bengal most respected medical education leaders and a distinguished pharmacologist, former Director of Medical Education, EO Secretary, Government of West Bengal. He currently serves as Chief Advisor at KPC Medical College. He has led premier institution as a principal and served as a professor and head across multiple medical college. A prolific researcher with 100+ publications, he has guided major projects on oxidative stress, immunomodulation, herbal formulation, microalbuminuria, and critical care inflammation. A recipient of prestigious fellowship and an invited speaker at national and international platform. He remains a guiding force in medical academia. Please welcome, sir. Now, it is my honor to introduce our next chair, Professor Rajesh Goel, Professor in the Department of Pharmaceutical Science and Drug Research, Punjabi University, Patiala, with more than 2 decades of contribution in neuropsychopharmacology, phytopharmacology, and pharmacogenomics and drug discovery. Professor Goel has authored over 90 publications and led numerous nationally funded projects, a fellow of the Indian Pharmacological Society, and a member of several prestigious scientific bodies. Professor Goel is a widely respected academic leader. We warmly welcome you, sir. Now I introduce our next chair, Dr. Ashok Dhatusalia, is currently working as Associate Professor in the Department of Pharmacology and Toxicology at Naipur, Raebareli. He received his PhD from Naipur SAS Nagar and his postdoctoral training at National Brain Research Center, Gurgaon, University of Milan, Italy, and Department of Neuroscience and Pharmacology, University of Iowa, USA. His research interests include age-related neurodegenerative disorders, stress disorder, and neurobiology of metabolism. He significantly contributed to the work investigating neuroinflammation, diabetes-associated cognitive deficit, and brain injury. Welcome, sir. Now we are going to begin our presentation. Each presenter will have 6 minutes for their presentation and 2 minutes for the discussion. Let us maintain enthusiasm and extend our appreciation to every researcher for their scientific contribution. Now I would like to call our first presenter, Soumya Upadhyay from Naipur, Mohali.
A very good morning to one and all present here. My name is Soumya Upadhyay. I'm from Department of Pharmacology and Toxicology from Naipur, Mohali. And the topic of my presentation is exploring the therapeutic potential of transient receptor potential mucolepin 1 agonist, MLSA1, in cardiac hypertrophy, serious functions and molecular insights. Cardiovascular diseases are the leading cause of death globally, taking an estimated 17.9 million deaths each year. It is very astonishing to say that 27% of the death in India usually happens due to cardiovascular diseases. Out of different cardiovascular diseases, the one disease that we have done research is on cardiac hypertrophy. Cardiac hypertrophy, it is a compensatory mechanism which ends up in increased cardiomyocyte size in response to various physiological and pathological stimuli. There are various limitations of the available therapies for cardiac hypertrophy, like various side effects include hypotension, hyperkalemia, thrombocytopenia, as well as high surgical complications are there. So there is an urgent need for the discovery of new molecular targets that might be used as a drug development for cardiac hypertrophy treatment. So the novelty in our research is that we have taken the target as TRPML1, which is a transient receptor potential mucolepin 1 channel, which has not yet been searched for proven for the therapeutic effect against cardiac hypertrophy. As the pressure overload increases, then the expression of TRPML1 downregulates, which ultimately leads to mitochondrial dysfunction as well as decreased autophagy, thus leading to excessive thickening of the left ventricular wall, thus causing cardiac hypertrophy. So the aim of our research is to evaluate the therapeutic potential of TRPML1 agonist, MLSA1, in cardiac hypertrophy, including objectives. First is to evaluate the expression of TRPML1 channels and in the heart tissue, and second will be to elucidate the molecular mechanism behind it. Talking about materials and methods, male Vista rats was taken of 200 to 50 gram and were divided into 7 groups. That is mentioned on the slide itself. After acclimatization for 7 days, isoproterenol was given for the disease induction at 5 mg per kg via subcutaneous route, along with this co-treatment of MLSA1 at 200 and 400 microgram per kg via intraperitoneal route was injected. Further, on day 15, ECG, arterial and ventricular blood pressures were recorded. Then further, animal was euthanized and blood and heart tissue was collected. Further, biochemical and histological assessment was processed. Talking about the result effect of MLSA1 on ECG parameters, the left side of the slide shows the representative ECG waveforms of different experimental groups. As you can see, as compared to the control group, in the disease group, there was increased QT interval, ST segment, as well as R amplitude, and reduced RR interval in the disease group as compared to the control group. And when MLSA1 treatment was given at low dose as well as at high dose, there was significant decrease in all these parameters noticed after MLSA1 treatment. on the right side, it has a graphical representation of ECG parameters. Now coming to the next, which is effect of MLSA1 on hemodynamic blood pressure. Figure A to C represents arterial pressure, systolic, diastolic, and mean arterial pressure. And figure D to H shows the left ventricular changes, like left ventricular systolic pressure plus dP by dt minus dP by dt. By MLSA1 treatment, it has been seen that there was a significant increase in LVSP plus dP by dt and minus dP by dt, whereas LVEDP significantly reduced with the low dose and high dose of MLSA1. Further coming to the effect of MLSA1 on hypertrophic indices, this is the figure A represents a representative image of the heart, and it has been seen that in the hypertrophy group there was significant increase in the heart size as compared to the control group, and when MLSA1 at low dose and high dose was given, there was significant improvement in the size of the heart. Other hypertrophic indices like left ventricular thickness as well as left ventricular weight, there was significant decrease in all these parameters by giving MLSA1 dose at higher dose. Then further, effect of MLSA1 on biochemical parameters like cardiac injury markers, hypertrophic markers, as well as inflammatory parameters like CKMB, CKNAC, LDH, ANP, BNP, IL-1 beta, as well as TNF alpha was measured, and it was found that in diseased conditions, a significant increase in all these injury parameters as well as inflammatory parameters was seen, and when MLSA1 was given at low and high dose, significant improvement has been noticed. MLSA1 treatment also affected the oxidative stress as it increased the antioxidant level, that is GSH, and reduced the MDA level. Talking about the mitochondrial complex activity, all the complexes were significant, complex activities were significantly increased with the treatment, and thus showing that this particular treatment increases the ATP production and resolving the systolic and diastolic function in the heart. Coming to the morphological parameters, they were assessed with the help of hematoxylin and eosin staining, where it can be seen as compared to the control group in the hypertrophy group. There was increase in the cell size in the hypertrophied animal, as well as there was a significant distortion in the myocardial fibrils. And when MLSA1 at high dose was given, there was significant improvement that can be noticed. Further, picrocereus red staining was done for the interstitial space as well as perivascular space, and it was found that in the hypertrophied group, there was increased collagen deposition, and when high dose of MLSA1 was given, there was significant reduction in the collagen deposition, as can be seen with the representative graphs also. Further, protein expression as well as gene expression studies have been performed, and it has been found that TRPML1 expression with the treatment of MLSA1 increased significantly at both gene and protein level. Further, autophagic markers like LC32 and Beclin1, as well as mitochondrial dysfunction markers like PINK1 and PGC1 alpha, all these markers significantly increased with the MLSA1 treatment at low as well as high doses. So hence, it can be concluded that MLSA1, treatment with MLSA1 increased TRPML1 expression in the heart, and it decreased the oxidative stress and inflammation, decreased cardiac injury and hypertrophy markers, attenuated mitochondrial dysfunction, improved autophagy, and reduced collagen deposition, hence alleviated cardiac hypertrophy. Thank you.
So, I would like to know how many types of the TRP channels presents on the heart tissue.
Sir, 6 types of TRP channels are present: TRP A, C, M, V, and ML.
Which one is the most abundant?
Most abundant is C, TRP C channels, canonical.
So how many animals you were considering that it should be there in interpreting your data? That is point, because ultimately the statistical significance becomes very important in ultimate prediction. So what is your idea about that?
Sir, in each group I took 8 to 10 animals, and
no, you should fixed 8 or 10, because 8 to 10 doesn't mean anything.
Yes, sir. Actually, there is
you
some
had
mortality.
A fixed number. You have worked on it.
Yes, sir. Yes, sir. Sir, I have mentioned
either
that
you
what.
Tell 8 or 10. Right?
Yes, so whatever assays I have done, I have mentioned down that whatever n number has been taken. For biochemical assays, usually 4 or 5 has been taken as n, and for like Western blot and RT-PCR
And
especially.
what statistical method you applied?
Yes, sir.
What statistical method you applied
So Tukey's,
to interpret? Because otherwise, if you can't express in statistically significant, your result will not be acceptable. That is the point.
yes, sir.
And
Soumya, you have worked on cardiac hypertrophy.
Yes, sir.
Just one question. Is it reversible?
Sir, if it is a physiological hypertrophy, it can be reversed. But if it is a pathological, it is not fully reversible, but we can decrease the progression of this particular disease.
That means your intervention has to be there before it happens.
Yes, sir.
How you will see that? How, when to start the treatment?
Actually, we can go for pre-treatment condition. Like before induction of the disease, we can start the therapy. And then later on, like along with pre-treatment, we can do co-treatment also to see.
Along with PT is what?
Like after, along with pre-treatment, and when the induction of disease happen.
Either you will stop the induction or will treat the disease.
Yes, sir.
Either you will stop the induction of disease or you will treat the disease once it is already there. No?
Once it is already there.
Then you will treat it. Once you are, but your approach is
Yes.
to prevent it.
Yes, sir. Yes, sir.
Okay. So when you will start preventing it, I'm asking that.
If we have to check for prophylactic treatment, we should go for pre-treatment condition. And if we have to, like, if the disease is...
Okay.
Yeah, okay, sir.
Soumya, how you perform mitochondrial estimation? Complex 1, 2, and 3.
So by ELISA kit methods.
Which part of tissue? How you process the tissue for this estimations?
Sir, with the help of... sir, the kit was available, and...
it's not about the kit. How you
Yes,
process
sir.
the tissue?
Sir,
The
and,
homogenate or the pallet?
Yes, sir. Yes, sir.
It's after processing the tissue, you use the pallet or the supernatant?
Supernatant, sir.
Supernatant.
Yes, sir.
How you?
So pellet, sorry, sir, pellet. I used pellet, yes, sir. Okay, thank you so much, sir.
Our next presenter is Janvi Soni from Parul Institute of Pharmacy, Parul University, Vadodara. Hello. Hello.
Hello. Good morning, everyone. The topic I'm presenting is evaluating the pathogenic role of synthesized human neuromelanin analogs through intranigral injection in mice and its implications in Parkinson's disease. So first of all, what is neuromelanin? Neuromelanin is a black-brown pigment which is present in the dopaminergic neurons of substantia nigra. Now this pigment increases in concentration and density as the person ages, and it almost covers the whole cell body of the neuron. Neuromelanin is produced when the excess dopamine is present in the cytosol and not in the vesicles. So the excess dopamine will lead to autooxidation, it will form protein adducts, it will trap iron, it will fuse with lysosomes, and ultimately it will form a neuromelanin autolysosome. Now this neuromelanin, when it is inside the neurons, it is having a protective role. For example, it will trap the reactive metals, trap the ROS, etc. But during Parkinson's disease, when the dopaminergic neurons are dead or are dying, they will gradually release the neuromelanin outside, and this neuromelanin, when it comes outside, it is increasing neuroinflammation and neurodegeneration. Now, how it increases neuroinflammation or neurodegeneration is not clear. But it is clear that there is a preferential loss of neuromelanin containing neurons in Parkinson's disease. So thus, coming to my hypothesis was, after the intranigral neuromelanin injection, this neuromelanin will be phagocytosed by the microglia and it will lead to microglial activation. This microglia, which is activated, will release the inflammatory mediators. These inflammatory mediators will lead to inflammatory processes in the neuron, and ultimately it will lead to neurodegeneration. So for this, my timeline looked as such. I had 4 different time points and different groups. First group was sham, then I had 3 groups of different concentration of neuromelanin analog 1 and 3 groups for neuromelanin analog 2. Coming to the results, so I performed extensive behavioral studies, starting with the round beam walk test. So in round beam walk, it is like you have to place the animal on the round beam and number of slips are calculated. So we can see the animal is slipping on the rod. This suggests the motor impairment. So then I performed grip strength test, apomorphin-induced rotation test, rota-rod test, etc. on all the time points. On every time point, I could see that there is a trend going on and motor impairment is increasing when at the 28th day. But the results were not significant, but I could see the trend. Coming to the histochemical studies, the histochemical studies were performed at all the time points again. So at 7th day, I could see that microglial activation is very high in the neuromelanin analog 1 and 2 groups as compared to sham. Here in the first image, we could see that's a EBA1 marker, and the same thing I have done with the astroglial activation, which is the DAP staining. At 14th day also, I could see that as compared to sham, neuromelanin analog 1 and 2 showed a higher microglial as well as astroglial activation. So here you can see in sham group, there is no activation, whereas analog 1 and 2 group, at the injection site, clearly we can see the DAP stained astroglia are present. So then coming to the 21st day histochemical studies, again I performed astroglial activation for both the analogs, and also it was a co-staining using a TH marker and a GFAP marker. So as compared to sham group, we can see that in the neuromelanin groups of different concentration, there is high astroglial activation. Same thing was observed for neuromelanin analog 2. Additionally, are performed iron staining, that is the Pearl staining for visualizing iron, as neuromelanin traps iron, and when the neuromelanin is phagocytosed by microglia, there should be release of iron. So same thing, I have performed the Pearl staining for iron, and moreover, I performed Mason-Fontana staining for neuromelanin, just to check whether neuromelanin is going in the proper exact that coordinates in the substantia nigra itself or not. After that, to check that is there a neurodegeneration which is actually happening or not, I performed the TH staining, that is tyrosine hydroxylase enzyme, which is specifically present in the substantia nigra only. So in sham group, this is also a DAP staining. So we can see this is a substantia nigra region, and here what I did is I took images and counted the cell bodies of the neurons present, exactly how many neurons are alive. So after that, I got these results. In the sham, we can see that density of neurons is very high, whereas in neuromelanin analog 1 and 2, we can clearly see the degeneration has happened. And these last results are very interesting to me. On the 28th day, I compared the GFAP and TH staining in the single animals. So in sham animal, we can see there is no GFAP activation and TH neurons are present. Whereas in analog 1 and 2, we can see that at the site of injection, there is GFAP activation, and at the same site, there is neurodegeneration. Same for analog 2, there is the GFAP activation, and the same site, there is a loss of TH positive neurons. So finally, I concluded that neuromelanin analog 1 induced significant astroglial activation, increased ferric iron, and induced TH positive neuron loss, and same for analog 2. But however, there was no dose-dependent effect which I could see. This ultimately, after this, the goal was to compare these results with using the human neuromelanin. Thank you.
So actually, what is your final conclusion?
So, so I wanted to check whether this synthesized neuromelanin analogs could induce neuroinflammation and neurodegeneration. So my conclusion is that they were able to induce neurodegeneration in the mice.
And what is the interrelation between this iron and the neurotransmitter?
Yes, sir. So, sir, when neuromelanin is present inside the neurons, they trap reactive metals. Now, when this neuromelanin, after neurodegeneration in Parkinson's disease, comes out of the neurons, it will release all the reactive metals and it will become, it will play a negative role in the pathology. So that's why I wanted to check whether it is releasing iron and this iron is leading, because my ultimate goal was to check ferroptosis in the neurons.
So I have two queries. One is like you train the animal before putting them on this neuromelanin treatment. So what is the relevance for that?
Sir, can you repeat? I could not.
So in your study design, you train the animal for the rotarod and the beam walk before starting your treatment.
Yes, sir.
So did you see any difference while like injecting a stereotoxic insult to them?
Yes, sir, I could see motor impairment.
So the motor impairment is due to the surgery or due to your
No, sir.
treatment?
Sir, I performed that for sham group also and neuromelanin surgery group also. So I could say different. Sham animals were behaving normally, whereas neuromelanin animals were, in sham group also I injected PBS.
Okay.
So,
And another things like where this dopamine is being synthesized, either it in the cell body or it is like elongated axons and dendrites?
sir, it is in the cell body.
So then white TH staining is like
Sir, tyrosine hydroxylase, sir, you are talking about this image? Sir, in the axons also some tyrosine hydroxylase will be present, that's why.
You perform so many behavior experiments.
Yes.
What is the timeline to perform all this? On the same day or same time? There is some difference.
No, sir, like same day, yes. So if the 7th day time point, then I performed the behavior experiments on the 7th day itself.
Continuously?
Yes, sir.
So when you perform the behavior experiments on the same day with the timelines, is there any effect like you perform the grip strength or the rotarod, then you go for the narrow beam walk?
Sir, so actually, for both the groups, there were different sets of experiment. For any group, let's say group 1, there were only 2 experiments, grip strength test and rotarod, or round beam walk test and grip strength test. So grip strength test was common, and other than that, there was additional 1 behavioral experiment, so 2. So after giving some gap, I performed the second one. No, sir, I performed this myself in Naipa, Ahmedabad. So we had these facilities.
Okay, Naipa, Ahmedabad, but your address is Parul University.
Yes, sir. Currently, I am working there.
So this work you did in your PhD or?
No, sir, my master's.
Okay.
Yes, sir. My thesis project. Thank you, sir.
Our next presenter is Dr. Dipanshu Goel from Central University of Punjab, Bhatinda.
Hello. Okay, so good morning to all the respected dignitaries and the delegates present here. Ladies and gentlemen, my name is Dipanshu Goel and this is the topic of presentation. Okay. Okay, so this is the topic of my presentation, which is bridging computational and molecular approaches to unlock Epleron's mechanism in experimental models of epilepsy. So as you all would know that epilepsy is one of the most common neurological disorders that affects close to about 50 to 70 million people across the globe. And as of now, more than 20 to 30 FDA-approved drugs are available in the market, but still a third of the cases of epilepsy are refractory. Hence, we need to look into more options. I selected eplerenone to check the anti-epileptic potential of it because of its already established mechanism in Alzheimer's and various psychiatric disorders. So again, the aim is to investigate the anti-epileptic potential of eplerenone in pentylene tetrazole-induced kindling in mice with various objectives to perform in silico, in vitro, as well as in vivo analysis. So the rationale says that pentylene tetrazole kindling was performed with a dose of 35 mg per kg, and it will decrease the seizure threshold to induce activation of the GSK3 beta. And MR antagonist, which is eplerenone, it will inhibit the GSK3 beta activation to prevent the FIR-mediated NRF2 degradation to decrease the oxidative burden in the epileptic disease. So initially beginning with the in silico outcomes, I performed various modalities for in silico outcomes. So first coming to the network pharmacology, 87 common genes were identified that were targeted by eplerenone at various targets, as well as that were common with the genes that are related to epilepsy. And as you can see that the pathway analysis shows that epilepsy was one of the most commonly enriched term. Other than that, biological processes, cellular component, and molecular function also speak of the kinase-related activity and various other pathways. To validate the results of gene ontology studies, I performed the M-code analysis, which tells us about the cellular response to hormone stimulus, regulation of apoptotic signaling, and toll-like receptors, which is related to inflammation. That is, we can say that inflammatory pathways are also getting regulated. Then coming to the cytohab analysis via the Cytoscape app, I got the top 10 genes which were taken for the molecular docking analysis, and it was found that caspase-3, GSK3 beta, and TNF-alpha were having the most negative docking score, which prompted us to take them to the molecular dynamic analysis, which also relieved the similar results, although there was a bit of ambiguity with the caspase-3 molecular dynamics, but still they were in the considerable range. Then the in vitro outcomes. I performed the cell viability analysis for the IC50 determination of pentylene tetrazole, which was found to be 30 millimolar concentration. Then for the diazepam, I took the 10 micromolar dose, and for eplerenone, 2.5, 5, and 10 micromolar dose concentration. Then again, they were taken for the MTT assay for the neuroprotective potential via the cell viability analysis, and it was found that eplerenone was able to significantly increase the cell viability versus the pentylene tetrazole, which was decreasing the cell viability. Similar was the case with the DCFD analysis, which tells us about the oxidative burden that is present in the cells. So oxidative burden via the fluorescence intensity was also getting decreased by the treatment of eplerenone, just like diazepam. Then here's the trypan blue assay, which tells you about the imbibition of the trypan blue within the cytoplasm. And also, as you can see, that there's a bit of clumping in the pentylene tetrazole. In the pentylene tetrazole group, there's a clumping of the cells, which tells that the degeneration of the cells as well as the necrosis. But this was not the case with the 5 micromolar, 10 micromolar dose of eplerenone, as well as with the diazepam. So this prompted us again to take it to the ethidium bromide and acridine orange co-staining assay, and the fluorescence intensity, as you can see, for the merged assay, I would like to say that there are various colors that are mentioned in front of you. So the live cells are shown with the green color, yellow for early apoptosis, orange for late apoptosis, and red for the dead cells. So you can see that in the 10 micromolar concentration, 5 micromolar concentration of eplerenone, as well as with the diazepam, the very less number of cells are showing the apoptosis as well as dead cells. Finally, for the in vivo outcomes, I performed the pentylene tetrazole-induced kindling in the various animals. So Swiss albino mice were taken, and 14 injections were given on the alternate days to the animals, along with the pre-treatment with either the diazepam or the eplerenone doses. After that, the challenge test was performed on the day 33rd, followed by brain isolation. Initially, for the development of the kindling, as you can see that pentylene tetrazole was able to successfully induce the kindling by relieving to the stage 5 of the seizures, as is shown by the black dots. But it was significantly decreased in the progression of the kindling by the treatment of eplerenone and diazepam. Similar is the case with the challenge test. As you can see that the latency time to reach the level of various scale, seizure scoring in the modified decision scale, and it was interesting to note that the stage 4, which is falling clonal with rearing and falling, and GTCS had no significant difference between the higher dose of eplerenone and the diazepam group. Then coming to the neurochemical analysis, GABA and GAD1 were significantly decreased upon treatment with the pentylene tetrazole, which were replenished again by the higher dose of eplerenone as well as diazepam, which tells us about the potential non-excitatory effects of eplerenone. And similar was the case with the nitrosative stress as well as the oxidative stress. So nitrite levels tells us about the nitrosative stress, and MDA tells about the oxidative stress burden, which was getting relieved upon the treatment with eplerenone. Then finally, the inflammatory parameters of TNF-alpha, GSK3 beta, and interleukin-1 beta also had the similar trend, along with the apoptotic marker caspase-3, and the HMOX1, which is for HO1, and NRF2 also were getting significantly decrease in the pentylene tetrazole, but were relieved by eplerenone. And the last was the immunohistochemistry of GFAP, which are the brown blemishes, as you can see in the pentylene tetrazole group, which were not the case with the treatment with eplerenone and diazepam. So to conclude my study, I would like to say that GSK3 beta would be the culminating point, which is decreasing the pathological triad of epilepsy via the oxidative stress, apoptosis, and inflammation, which is disrupting the GABA and GAD1 levels, which is getting relieved by eplerenone. So thank you so much for letting me present my work in this IPSCON conference. Thank you so much.
Dipanshu, you did this study with in silico, in vitro, and in vivo approaches.
Yes.
What is the significance of your in silico approach for this study?
Sir, actually, in silico study was conducted to check the particular target that eplerenone might be working, only because we are coming into the world where it is not just one drug and one target action, we have come into multi-modalities of it.
Targets you have selected, they are already explored for this purpose.
Sir, diazepam is taken as a standard, but not eplerenone.
No. Targets, the genes which you have selected.
Yes, sir.
They are already explored for epileptic management, experimentally at least.
Yes, sir.
So what is the new thing we get from in silico outcome?
Sir, actually, when I tried to look for the targets of eplerenone, initially I wanted to check the MR and GR disbalance. That is why I selected this drug. But whenever, upon conducting the in silico analysis, I found that there was no significant difference. But then I took the targets of GSK3 beta and other TNF-alpha and caspase-3, which were highly enriched in the in silico study. So that is why I took them forward. Otherwise, initially my work was supposed to check the MR-GR disbalance in it, but it was non-significant as per the in silico study, so I did not conduct that.
Okay.
Yes, sir. Should I show them again, sir? Yeah. So yellow is for the early apoptotic marker, orange is for late apoptosis, and red is for the dead cells. Yeah. Yeah. Yes. So actually it is established. I have some literature study for it, and I found that it is for early and late apoptosis. It is established, sir. It is called localization, and it can be an indirect marker for the early and late apoptosis.
And in fact, in your control and pentylene tetrazole, what was the significant difference?
So actually, since I conducted, I did not conduct
what control you used for this?
So control was 0.1% DMSO.
And what is the significant difference?
I did not calculate the significant
ultimately,
difference.
What is your aim? What is your aim is to control the...
I beg your pardon,
What is
sir.
your ultimate aim?
For this experiment?
Yeah.
To check the prevention of apoptosis by the treatment of eplerenone.
So like in this same image, if you see the third panel, where there are some red fluorescence is there, that cells are not available in the first panel green.
Yes, sir.
Moreover, like what are the cell size, we cannot like predict from, because the scale bar is not absent. So what magnification did you use in that?
So this is 20x.
Okay, but it has to be some mark.
Okay.
Fluorescent intensity has been increased. Go to oxidative stress where you have one slide before.
Yes, for the DCFD analysis.
Yes, go back next. So yes, so like here you have seen the PTZ, the cells are really very less because due to the viability.
So I did not check the fluorescence here. I am just checking for the clumping and the imbibition of tryptophan blue into the cytoplasm. This is not for, this is simple microscope, so this is not fluorescence.
In the first graph before that.
DCFD, sir? Sir, actually, I'm using the control, the fluorescence that I found in the control. I'm using it as 100%, and increase in the concentration tells about the increase in the oxidative stress parameters. Thank you so much, sir.
Our next presenter is Achal Arora from Punjabi University, Patiala.
Good morning, one and all present here. I, Anchal, from Department of Pharmaceutical Sciences and Drug Research, is here to present my first objective of research work, that is a system biology approach to identify the hub genes and therapeutic targets in the Parkinson's disease and L-DOPA-induced dyskinesia. So as we all know that Parkinson's disease is a neurodegenerative hypokinetic movement disorder that primarily affects the dopaminergic neuronal loss in the substantia nigra with a major dopamine neurotransmitter loss. So here, as we all have seen that from 1960, we are only using levodopa for the treatment of the Parkinson's disease. But the major side effect that will appear after 3 to 4 year use of the levodopa is levodopa-induced dyskinesia. That is the major motor symptoms that will appear after 3 to 4 years of use. So for this, there must be improved therapeutic strategies that requires the understanding for the underlying molecular mechanisms for both the diseases, that is for PD as well as for LID. So this is the mechanism, this is the dopaminergic as well as non-dopaminergic mechanisms that will lead to the LID. So there will be nigrostriatal degeneration, that is dopamine loss. There is a pulsatile L-DOPA release, aberrant signaling will be there, striatal plasticity will be there, other non-dopaminergic pathways like glutamatergic, serotonin and other pathways are involved, and major target is neuroinflammation. So there must be a gap in this knowledge, as both the diseases, both are interconnected, both are complex diseases, so there must be a molecular targets that we have to chosen. And traditional approaches like one gene, one target approach will now be reduced, and these approaches will make major focus on the key interaction and major shared molecular mechanisms that involve the both diseases. So here the computational approach, that is system biology approach, and the network pharmacology approach, it will integrate the multi-source gene data that are using to identify the hub genes that are implicated in the diseases to target the therapeutic approach. So the first objective, that is we have to identify the pivotal genes, molecular pathways and potential drug targets that are implicating. For this we have constructed the PPI network and we have go for the pathway enrichment analysis. The data sources which we have used is string database and David Co. For the purpose, here we mainly identify the hub genes and we have revealed the biological processes. Tools we have used are Cytoscape, KEGG and Go. The outcomes are, we have identified the major top 10 hub genes that are implicated in PD as well as in LID, and then the mechanistic pathways for both the diseases. So this is the methodology, various databases we have referred like OMIM, therapeutic target, DisGeNet ensembles, from where we have collected the genes, then the data mining was done, then we have go for the Go and KEGG enrichment analysis. Afterwards, PPI network was constructed, then module extraction was done, then hub gene identification, and last one is the drug gene interaction analysis was done. So this is the, now come to the results. 522 genes, including for PD and LID, was identified, out of which 496 was shared for the enrichment analysis. Afterwards, here 486 was considered for the nodes and 640 edges were found, and then 9 major clusters were found. So these are the top 10 hub genes with their pathways that have been selected for the PD as well as for LID. Now come to the therapeutic implications. These genes have a relationship with these kind of drugs, and drug gene interaction database analysis has revealed 722 total drugs interaction across these 10 hub genes, with sharing these three have the highest interaction counts. 10 FDA approved drugs with highest interaction scores were prioritized. And so for these repurposing candidates, we can say offer the accelerated clinical translational potential with established safety profile that will provide therapeutic opportunities for PD management and LID prevention. Coming to the discussion and conclusion, as it is the integrative bioinformatic approach that will uncover the complex pathways that are involved in Parkinson's disease as well as levodopa-induced dyskinesia. So discovery of core genes and their pathways helps in the targeted therapies. So these results support the rational drug repurposing and new strategies for symptom management. So here the top 10 genes were identified for further research. These are my references, and thank you.
Thank you, Anchal. Anchal, the genes you reported,
yes, sir,
for this through this software,
yes, sir,
it's already reported in a literature that these are involved in PD pathogens or not?
Sir, most of the genes are reported, but I have used some different softwares or different databases. There are too many databases. Every database will give you a different gene. So we have chosen, for this work, we have chosen majorly 4 databases. That is GeneCard, Ensemble, DisGeneNet, and Therapeutic Target Database.
What animal model you choose to precipitate Parkinsonism?
Sir, animal model? Yes, sir, we will go for...
How?
Yes, sir, we will go for the rotenone as well as with the use of 6-hydroxydopamine. That is our upcoming work.
So you have mentioned 10 gene you have identified.
Yes, sir.
So which one is the most prominent according to you?
Sir, here neuroinflammatory genes like interleukin-1 beta, tumor necrosis factor 1 beta, as well as TP53 are the 3 who shows more interaction and are involved in neuroinflammatory pathways.
Okay.
Thank you. Thank you, sir.
Okay.
Sorry.
Our next presenter is Manvi Dureja from Central University of Punjab, Bhatinda.
Hello. May I start, sir? Good morning, myself Manvi. I am here to present my presentation on topic network pharmacological and experimental approach to explore the mechanism of dapagliflozin and ticagrelor in the management of epilepsy. This will be the flow of my presentation, starting with the introduction. Epilepsy, as we all know, epilepsy is a disorder of neurotransmitters where the imbalance between the inhibitory and excitatory neurotransmission leads to the epileptogenic events. And epilepsy is also associated with several secondary comorbid conditions such as anxiety, depression, and cognitive impairments. WHO data suggests that epilepsy is the most prevalent neurological disorder, as 65 million people worldwide suffer from epilepsy, and WHO also suggested that 1 in every 26 people develop epilepsy once in their lifetime. There are now FDA has approved more than 30 anti-seizure medications, but still the count of epilepsy patient rises day by day. And recent research have suggested that the imbalance between the hematological profile and the glycemic index are major risk factor associated with the occurrence of epileptogenic events. So to elucidate the hematological profile and glycemic index as a risk factor of epilepsy, the aim of the study was to explore the effects of dapagliflozin and ticagrelor in the management of epilepsy. Dapagliflozin is an SGLT2 inhibitor approved for the diabetes management, and ticagrelor is a P2Y12 receptor FDA approved for the management of ischemic stroke and act as an antiplatelet agent. To achieve the objective was to elucidate the possible targets of dapagliflozin and ticagrelor in managing epilepsy using a network pharmacological approach. Second objective was to assess the anticonvulsive effects of dapagliflozin and ticagrelor using a maximal electroshock convulsometer. Third objective was to validate the network pharmacological findings of dapagliflozin and ticagrelor using experimental approach. To achieve this objective, the methodology for objective 1 was, I have used several bioinformatics software such as Swiss Target Prediction. This software was used to predict the targets for dapagliflozin and ticagrelor. Second one is draw Venn diagram. Draw Venn diagram was used to find the common targets between the dapagliflozin, ticagrelor and epilepsy-related genes. Cytoscape was performed to evaluate with the associated with the epilepsy. String analysis was performed to elucidate the protein-protein interaction and to perform the pathway enrichment analysis. Shinew was also used to perform the pathway enrichment analysis. To achieve the objective 2, that is the experimental approach, I have used the maximal electroshock convulsometer. Pre-treatment of dapagliflozin and ticagrelor.
Which detector?
Which detector? PID.
Is beyond the acceptable limit. So coming to the rationale and hypothesis, the lead crosses the lead-induced neurotoxicity. The available treatment is the chelation therapy, but it is with the major side effects such as the bone marrow depression, kidney failure, such as IGF-1 and GLP-1 pathway that plays the major role in the synaptogenesis, learning and memory. So we hypothesize that the ezetimibe and sericin.
Due to Dr. Rajesh Goyal.
All the presenters, I'm sorry I have to leave to the another session where we have to give a talk. So for that I have to rush. Okay.
Thank you.
So, so main thing in most of the anti-cancer drugs, we are using the doxorubicin, which is this a well-proven fact. And in most of the papers, if you see.
Yes, sir.
So, what method you applied to induce diabetes?
Yes, sir. For the induction of diabetes, the single dose of streptozotocin at a dose of 55 mg per kg. Yes. So the model comprised of 56 days. First 28 days was for the induction. For the next 28 days, the treatment was given. Entering it via GLUT4, which is the transporter.
Which is the create the research gaps. For the in starting there is a mortality of the high dose of there is also solvent like causes, but there is no mortality. And number is 12. So mortality.
So which cell line did you use?
In PC12.
Okay.
Which will be coming into the market soon.
IT IPR Chair Professor, leading innovation and intellectual property outreach. We are privileged to have Dr. O.P. Katare as a chair for this session. Thank you, sir. Now I request to our chair, please introduce our speakers. Anyone, Doctor?
Into the scaffold and then you implant it. And we try to do it in any presentations. And these are my funders. You can see this logo is same as this, you know. So I think that one is South African National Research Foundation logo. I only observe it now. G20 is happening this weekend, tomorrow and day after tomorrow. Modi ji will be there, I hope so. Okay, it is in Johannesburg, our own city. And I invite you all to Cape Town next year for Behavioral Neuroscience Society Conference. It is in Cape Town in June. It's very cold, it's very wet, so please be prepared for that one. And I thank you for your attention. And I'd love to have any questions.
So excellent presentation, Dr.
Thank you so much, bro.
Quite gratifying to see the progress in nanoparticles reaching out into the bone. You know.
Yes, the direct implant, that was easy, Prof.
So amazing to see that. For not preferring on lipid delivery system. You know, mostly the phospholipid-based systems. You know,
Liposomes. Yeah.
Liposomes are maybe
Lipid.
Other
Okay.
versions of the lipid delivery the system. How do you see that? How do you differentiate in terms of their performance, efficacy? Why don't you choose the lipid delivery system?
Okay. No, the good question, Prof. So I also work with liposomes, but only for neural applications. Okay, so the lipids are very expensive. Phospholipids are expensive, and TB is a disease of burden. A disease of burden. So this means it has a big burden on a healthcare system. So if we use phospholipid-based systems, then it will become expensive. So cost is the biggest reason. So if the study is anti-cancer or if the study is neurological, I will use liposomes. But if it is infection-based systems, then we use polymeric because they are cheaper and cheaper to make.
Don't you think these phospholipid-based systems like liposomes or any vesicular systems, they are more interactive? They are more interactive.
No, 100%, Prof. I agree with that. You know, so but if that's what I said. You know, our funders only fund things that are what you say not commercializable, but can be used later on for real medical use. So that's our. So if you see all my papers in infectious disease, all are polymeric, and neurocancer are liposomal, and trauma is all scaffolds. Yeah, that's the reason. But it's 100%. You are 100% correct, Prof. It's I love lipid-based nanoparticles. They are very good efficiency, easy to target, easy to make, very stable. Yeah. Yes, 100%. Yeah. Mm-hmm. Okay, in South Africa it's already being used. Yes. Yeah. Finance. Okay. Yeah, the first one is for clinical phase. Yeah, the second one is not. Yeah, sorry. Yeah, so it's a Gates grant. Actually, Gates Foundation finds partner for us. Okay, yeah. So Bill Gates and the Gates Foundation as a partner, and so WITS has 7 hospitals, and our university, and the Bara Hospital is the world's 3rd largest hospital, 3,500 beds. So their most clinical trials are happening. Yeah. Yes, 100%. Prof. Thank you so much. Thank you for listening. Thank you. Thank you for your question. I just want to say one thing. There is a BRICS grant is open for anyone who wants to come to South Africa and visit our labs or in Cape Town. So for visiting fellows. Thank you.
Objective was that how interaction with the right people can take you from what you call nothing to something. So just want to narrate a small incidents that I, being a expert review panel member in ICMR, and reviewing the second year report of the project, and the investigator from the IIT Bombay expressed that he is a biomedical engineer but not able to find a pharmacologist to test their formulation. And I, being a panel member, I volunteer, and then he happily agreed. And this is how the ICMR sanctioned us a small grant to complete the pharmacological study or animal study of the formulation. And based on the fabrication, characterization, in vitro evaluation, and then pharmacological evaluation, animal study, we applied again together a major grant to ICMR for clinical trial of the same formulation. And this is how the last year we got 1.2 crore for a clinical study of this formulation. So this is a little bit about the how that helping or collaborating with the right people can maybe from concept to a product which can be taken to the advanced stages. So, you know, this chronic wounds is not India's problem. It's not about the Asia's problem. It's a global problem. And there are different kinds of wounds, like maybe in wars, maybe in diabetic wound, and so many accidental wounds. So problem is huge. When we talk about the diabetic wound, it becomes very difficult because of two reasons, that pro-inflammatory macrophages persist in a diabetic wound and re-epithelialization does not occur. And collagen-rich scar tissue does not form. And that's why it becomes very difficult to heal. And in fact, the situation comes then gangrene and then sepsis, and maybe the amputation is the outcome. Now, when we talk about the various marketed products for wound healing, you have so many with the brand name, but then either they have not for the deep wound, or maybe swelling risk, not, and they are animal derived or expensive, and then they are human plasma derived and costly. So there is a still need, specifically formulation of the product with a better safety, better efficacy, and bioactivity and optimal adhesivity. So what we did, we synthesized, characterized silver nanoparticles, gelma and methyl acrylated gels, gelen, then fabricated the UV cross-linked gel and gelatin hybrid hydrogels, loaded with the fibroblast growth factor and silver nanoparticles, and studied the release characteristics of these hybrid gels, and evaluated their in vitro antibacterial activity, cytotoxicity, cell adhesion, migration, and then proliferation. And then after the in vitro evaluation, we studied its animal activity in excision wound model. So This is a little bit about the what you call biofunctional mechanism of AG NP integrated Gelma hydrogel, which we already published in ACS Applied Biomaterials in 2019. And what we wanted, we want to regenerate the tissue, we want to restore the perfusion to the what you call wound, and then simultaneously provide the antimicrobial effect. And this is what our hybrid gel do. Fabrication, this is the schematic showing the synthesis of Gelma, methacrylated gelin, and fabrication of this hybrid hydrogels. As it is already published work, you can refer the paper for further details. We characterized with 1D NMR, then what you call various properties, structural and mechanical properties, porosity, and then wettability and other measurements. So if we see the wettability, mechanical properties and degradation behavior of the hydrogel, the contact angle measurement was done, rheology analysis was done, and then a degradation kinetic was done of all the what you call hydrogels and compare. And then, so when we formulate, like fabricated the hybrid gel, then we synthesized the silver nanoparticles, characterized it, and then studied the cytocompatibility. And then when we did the TEM analysis, that silver nanoparticles showed the uniform spherical morphology, size distribution was also confirmed. Then release profile also provided the control and sustained release of the silver nanoparticles. And then we carried out the cytocompatibility assessment using NIS3T3 cells, which are specifically used for cell proliferation activity. And it showed that cells on silver nanoparticle loaded hydrogels remain viable. Then minimum dead cells were observed, and DAPI staining showed the intact nuclei and confirming non-cytotoxicity of these silver nanoparticles. Then we studied this hybrid gels for cell adhesion and then motility and proliferation. Because when we incorporate this, entrap this fibroblast growth factor, whether it is what you call helping for the purpose for which we are synthesizing. Then this this graph shows the release kinetics of the FGF by ELISA, and it showed that this gel showed the reduced cell spreading, fewer focal adhesion, weaker adhesion than the Gelma. And then trypsin assay showed the lower contractility and adhesive strength on a hybrid gels. And if we talk about this FGF entrainment, this fibroblast growth factor enhanced the cell migration and proliferation on both Gelma as well as the hybrid gels. Then we carried out this AGNP and this FGF entrap gel for in vitro cell experiments under the cell experiments, and we found that hybrid hydrogels significantly decrease the cell spread area and contractility in at least in NIH3T3 fibroblasts, and cell proliferation was what you call significant because of this FGF entrapment. Then hydrogels modulated the cell adhesion as well as the cell contractility. You can see this focal adhesions and then cytoskeleton. And then de-adhesion assay was done to measure the cell adhesion and contractility on the hydrogels. And we compare this what you call hybrid gels. And we found that excessive cell adhesion, decreasing contractility, and that is what the benefit we found in hydrogels. When we talk about the live cell viability and antibacterial activity, so AGNP was entrapped because we wanted to provide the antimicrobial activity. Now we saw that there was no cytotoxicity because there was a high cell viability in AGNP loaded hydrogels, and that is indicated by percentage live cells per frame. While simultaneously AGNP demonstrated strong dose-dependent antibacterial activity against E. coli and Staphylococcus aureus, and that is what you call confirmation that it is biocompatible and it possesses the potent antimicrobial action. Because Pseudomonas than Staphylococcus are the most common bacteria found in infected wound, and that is why we selected E. coli and Staphylococcus aureus. Then cell migration and proliferation assays were done of hybrid gels, and this showed slower and shorter cell migration, then significant reduction in the migration speed, and the fibroblast growth factor enhanced the cell proliferation, while hydrogels without FGF show minimal change. That means FGF is must for cell proliferation. Then, based on this in vitro characterization and antimicrobial activity, what we did, we carried out the animal study, and so we treated the what you call excised wound model rats for 21 days and measure the various parameters like wound contraction, wet granulation, epithelialization, collagen content, then fibroblast growth factor and PDGF in wound fluids, Masson trichome staining for collagen type 1 and type 3 and HNA staining. So these are the various what you call time points where various parameters were measured and the method of measurement. So this in vivo study demonstrated that hybrid gels accelerated the wound healing as we measured the wound images or captured on day 1, 7, 14 and 21, and that showed the progressive wound contraction across the treatment groups. And then we also studied that epithelialization period and wet granulation weight. So on wound contraction, epithelialization period and wet granulation weight, hydrogels showed that it is what you call having the wound healing activity, and it demonstrated the faster wound healing compared to non-treated groups. And we found this enhanced tissue regeneration with the FGF and silver nanoparticle entrap hybrid hydrogels. So to be very specific, these hybrid gels markedly lower the inflammation, higher the fibroblast proliferation and angiogenesis as seen into this HNA staining, and we saw increased collagen 1 and 3 levels in Masson trichome staining. And then both ultimately indicates there was a superior collagen deposition and better ECM remodeling, which is what you call must phenomena for faster and better wound healing. And to summarize that, what are the key features of our formulation? That it is modulable, cell adhesion for improved tissue regeneration, in situ polymerization is possible that allow application on wounds of any size or shape. So you can formulate it as a gel, you can formulate it as a film. If it is a film, you can just dip into saline and apply on a wound. It will adapt the shape and depth of the wound. And it being a film, it works as a wound contracting or adhesive kind of dressing. And so cell, then FGF enhanced the healing, promoted the wound contraction, closure and angiogenesis. These are the, and then addition of silver nanoparticle provided the stronger antimicrobial property. Now when we compared this with the existing formulation in the market, we found that Bharat Biotech already have this EGF containing gel. EGF dose is very high. It's not providing the antimicrobial effect which our formulation provide. So based on this, we filed the patent with the help of Patent Mitra initiative by ICMR, and then publish into International Journal of Biological Macromolecules. And based on this published paper and then patent, we again applied to ICMR for a larger grant, and fortunately, generously, ICMR funded us for a clinical study of this formulation. So, so this is all about from my side, and I would like to acknowledge my collaborator, Professor Shamik Sen from IIT Bombay, who is faculty in Department of Biosciences and Bioengineering, and students, PhD students, Megha Sahu and Ifat Jan, and then ICMR for generous funding to preclinical as well as the clinical, and also filing the patent. So thank you so much. So any questions? Agree. So your question is absolutely right, and what I'm claiming based on our studies is on an excision wound. I may have to carry out a separate study to carry out for a diabetic wound, which is more severe. I agree. Yes, yes. So based on the wound size and shape, you can use the fabricate the film and can be used. So same film can be used. No. Yeah. So based on your question, I don't claim that my formulation will be better for burn. I'm claiming only for a diabetic wound and the excision wound. Yeah, and the clinical study is ongoing, sir. Yes. So I'm not I'm not exaggerating the claim. I'm claiming what is done. And based on only, we got approval for a clinical study with the AIMS Department of Surgery. Say it again. Yeah, so I showed with the Mason trichome. No, we didn't find it. We carried out specifically for Mason trichome screening for collagen 1 and 3 deposition. The plus point about this formulation is that we don't have to apply this formulation twice or thrice a day. Once in 3 days is enough, because we measured the FGF in a tissue fluid and it found present even at the 3rd day. So it's a slowly release, and that is one more advantage of this. Sir.
How do you address the challenge that right now you are doing on animals? Question.
Yes, sir.
There is a different, and there is the in situ gelling. The phenomena that is taking place in the wound, excised wound.
Yes, sir.
And the real life situation in patients, right? The diabetic foot ulcer.
Yes, sir.
So how do you see the difference between the two situations?
Sir, so as I said, what is we have demonstrated and what we will find in a clinical study is aid to receive. But yes, you are absolutely right, because diabetic wound is very, yes, sir. Yes. Agree. Agree. Yes. Uh-huh. Okay. Yeah, yeah. Again, being a pharmacologist, I can, what you call, innovate, incorporate more models, more types of wounds into my study. But yes, as you rightly said, the specialization, and I'm more relied on my collaborator for biomechanical and bio. Okay, thank you so much.
The next speaker is Dr. P. Karuna Sri. She is additional professor of pharmacology at AIIMS Kalyani with over 14 years of academic and clinical research experience across premier institutes. She has led projects in drug safety, metabolic disorders, and community-based clinical research with significant contributions to the medical education and national health programs. Now I invite Dr. Karuna Sri to enlighten us with her perspectives in glycemic control of multi-system benefits, the evolving role of GLP-1 analogs and its safety prospects. Dr. P. Karuna Sri, Madam.
Thank you, sir, for the nice introduction. I'm here to talk about the glycemic control to multisystemic benefits of the GLP analogs. I would like to say before starting my presentation that I belong to the pharmacology of clinical background. So I don't have any data with related to the experiment, bringing something new or novel patency like this. But I try to harvest the things that is already being explored by the hard-working pharmacy researchers, I would like to say. So from that part, I would try to explain the things, how it can be used, how the GLP analogs can be used or explored in a new way. So this I will just cover under the background, analogs and the unique action, wide therapeutic uses, and the concerns in summary. So as we know, the globally GLP analogs, so we have the wide burden of the diabetes. So nearly the 589 million people are living with the diabetes, and especially in the low and middle income countries, the epidemic is growing. And India alone has a prevalence of 10.5%, and the management of type 2 diabetes, as we know, it will be varying and evolving continuously depending upon the patient characteristics and all. And we need to choose our drug. So the guideline says that, so suggests previously it was all the new drugs are being as add-on drugs, but the recent guidelines says that it was the national as well as the international. So they can be used as a first-line drugs, and they can be used for the multi-system benefits for apart from the diabetes. Along with it, the persons who are suffering from the obesity or with the cardiovascular risk or with the diabetic kidney disease, as well as in the heart failure and the metabolic associated steatohepatitis, which was recently approved in 2025. So, and there is a huge market for the diabetes, as we know, and the diabetic drugs. That's why the reason we are trying to the use, as well as because disease is also growing, as well as the use of drugs also growing in the same way. So among that, if we see all the drugs, the GNP analogs are the
mostly chosen drugs among all the anti-diabetic drugs and with a high market area. So because of that, I choose this one, and it's expected the market is growing very well by nearly the 62 billion market size by 35. So that is the reason many the people are doing a continuous research on this area. So coming to the GLP agonists, they usually act upon the GNP receptors. Either currently we have the drugs which are having only the GLP receptor action, or the dual agonists like the tirzepatide, which is being approved in 2024, and the triple agonists which are yet to be approved in the phase 3 trials. It has been recently finished. Either the GLP-1, GIP, or the glucagon. Triple action will be there. That is the retatrutide. And some more GLP analogs, because all the GNP analogs are most of the peptide backbone. So they are made of the peptides. So the non-peptides are also being introduced. That is the orforglipron. And depending upon the chemical structure, whether the exenatide is being used or the GLP is used, so the categories, that is exenatide group of drugs, exenatide, lixisenatide, these will come, or the GLP analogs like the dulaglutide, liraglutide, semaglutide will come. So depending upon the chemical structure. So we have the various drugs, and they have been proven right from the 2005, which is initially been approved for only the type 2 diabetes, and later been the use and the approval has been extended to the other areas also, to the obesity with cardiovascular risk as well as with the heart failure, especially the preserved ejection fraction, not the the reduced ejection fraction. If we see compare with the SGLT2, where the drugs have been approved for the reduced ejection fraction, heart failure with reduced ejection fraction, but here these drugs have been used with the preserved ejection fraction. And the various trials have proven their efficacy. And as I told you, till 2024, we have the tirzepatide is been approved. And recently, the semaglutide has been approved for MASH. This is the only drug which has been approved for the metabolic associated steatohepatitis. So based upon this, why these drugs have this wide variety of actions, and we are able to use these drugs in the different kinds of associated diseases also, because of their receptors, presence of receptors basically, where the receptors are present apart from the pancreas. There is a high number of receptors are present in the even in the CNS as well as in the other areas also. Expression is seen in the heart, lungs, kidney, as well as in the liver. So these areas which have been make them to think that whether these drugs will affect on these area parts also, or on these diseases also. So, but the pathways, if we see, ultimately the whole pathway, apart from the GLP receptor pathway, it will be improving the oxidative stress, either by decreasing the cell proliferation and apoptosis. So these pathways are interlinked actually. PI3, the phosphatidyl inositol 3 kinase, or AMPK pathway, MAPK pathway, which are improving the or decreasing the oxidative stress ultimately, and help in the improvement of the cell environment. So this you can see where the mTOR pathway, or the here you can see the insulin receptor substrate or the insulin growth factor, which are ultimately at the lower level acting through the same mTOR pathway or AMPK or the MAPK pathway, inhibiting the cellular proliferation or apoptosis. So based upon this, we thought like these are already I explained that these are the approved uses. So what we thought is like based upon the preclinical studies, because preclinical studies are the one which are doing by the pharmaceutical people, where they are trying to explore the many things. So using the search terms, we explored the PubMed as well as the other sites, and in the last 10 years, we could able to extract some 23447 articles, out of which after deleting the duplications and selecting only the clinical trials and the observational studies, so we got some 4898 articles, out of which, so I thought like, but my aim is like in what other areas can we explore. So that is the reason I chosen only the preclinical. So when we installed them in the Ryan and explored only the preclinical studies, 98 articles we got it, and among that I have again excluded the articles which are already been approved, because so many studies are there which they are comparing either the already existing drugs in the diabetes or the obesity or heart failure and all. So out of which 13 articles I got it, And among them, these are the some areas which we can potentially explore for the clinical use and try for the clinical trial especially. So one is the dementia, the neuroprotective, either in the Alzheimer's disease or as well as in the Parkinson's disease. So these areas are yet to be explored. Only few studies are there, 1 or 2 studies are there, where they are trying to explore the effect of these GLP analogs in this area. And the next is cerebral ischemic stroke. Even though it has been proved as per the guidelines that these are the drugs which can be used or prevent the risk of cerebral ischemia when the diabetic person is on these GLP analogs. And the other is, even in the non-diabetics also, they have found that in the animal studies, if you give these drugs as a before the ischemia or just as the ischemia started, they are proven to have some benefit in decreasing the amount of ischemia or the amount of infarct that is formed in the brain. So hence this can be used either the cerebral ischemic attacks as well as explored in the Parkinson's as well as the Alzheimer's disease. These are the 3 areas. And among the heart failure, as I told you, though it has been used in the heart failure in preventing the preserved ejection fraction of heart failure. But reduced ejection fraction is still not there. But some of the studies are there here where Josmam Hegedis et al. He observed that where even with the reduced ejection fraction also they can be used. But still this area is not clear. So this is one area which we can explore for the clinical studies. And the cardioprotective action is a myocardial fibrosis has been shown to be decreased in the preclinical studies. And because hypertrophy is one condition where the heart failure, the primary pathophysiology is that the heart is not able to contract or because of the fibrosis that is being developed. That is most commonly seen in the preserved ejection fraction. So this is one proven thing. And apart from that, the other areas are the muscle strength and function and the osteoporosis and pulmonary fibrosis. So these are the areas which we are exploring now. So we have submitted 2 of the studies on the muscle strength, because in this sense, few of the preclinical studies are showing that they improve the muscle strength. But how far they are helping in improving the muscle function is not clear in these studies which has been given. And few of the muscle strengths and there is also a difference in the type 1 and type 2 fibers also. Like fast contracting fibers and low contracting fibers also, there is a difference in the muscle function and strength, how they are affecting. So this area we are exploring now, we have but this is only observational study we have doing it. And the second study is on the pulmonary fibrosis. So as we know that semaglutide especially been approved for the MASH, that is metabolic associated steatohepatitis. So this is one area which we are exploring, and it has been proven in the preclinical studies that this pulmonary fibrosis is being improved, and all the anti-inflammatory markers and the oxidative stress has been decreased in the preclinical studies. So this we have submitted for the clinical trial actually. So if ICMR gets approved, then we will start this trial. And adverse effects, as we know, though the market is very high, recently we are seeing that the very serious adverse effects have been found with some of these drugs, so which is raising a serious concern. So general adverse effects and nausea, vomiting, diarrhea are the most common thing. But out of this, suddenly they may develop with the acute kidney injury, which is a severe alarming kind. And suicidal tendencies and pancreatitis or some of them the risk of cancers, as we see with the other anti-diabetics also, like pioglitazone as we see. So consensus of this also we try to explore it, what are the adverse reaction reports that has been observed in the various adverse event reporting sites. So nearly around open whistle of more than 2 lakh reports have been observed. This is for only semaglutide we have conducted, because this is the molecule which we are trying to explore it. Semaglutide is the one which is having the high market rate as well as being more used because of its long duration of action. Once weekly subcutaneously it can be given compared to the other drugs. So this molecule is being explored, and in the VG axis, that is WHO episode, 83,000 reports have been seen, and we have so many case reports which are showing its adverse effects. But ultimately serious adverse effects, if we see, some of the disproportionate analysis, if we see, by that we got that ophthalmic artery aneurysm is one which serious concern seen with the semaglutide. So we need to, when we are doing any trial, we need to see that these conditions are not present in before starting these drugs in those patients. and gastric ischemia, because these are the drugs which slow the motility of the gut. And allodynia is also there, that increase in the pain. Maybe this may be either due to the disease or the drug, which is not clear. Because FAES, we get the information only whatever is being reported by the clinicians or the even the consumers also. So we could not able to directly relate the drug to the event from the FAES website. But these are the commonly related serious things which have been observed. I mean, the vitamin B12 deficiency also been seen in the website. And similar observations have been found in the one of the study of the systematic review by Roy et al. And they also reported some interesting findings that the males are having the more, they have the early onset of clinical adverse events compared to that of the females. And the adverse events rate will come down when they are being added with the other metformin group of drugs. So, because nowadays we are using only the metformin as such, I mean, these drugs are being used as a single drug rather than the dual therapy. So, this is one finding which have been observed. So, this also can be explored. In summary, GLP-1 analogs opens a wide therapeutic options with, however, associated with the serious adverse events. Hence, choosing the drug rationally, keeping in view of comorbidities as well as screening the risks associated. So, and they may show promising results in the new areas such as in neurodegenerative disease, as I told you, the AD, Alzheimer's disease and the Parkinson's disease. And sarcopenia may be of value. This we have to explore because there is some are showing the two types of results, pro as well as the cons are being there. And osteoporosis, they are also helping in improving the bone density also, because we don't have many drugs for the drugs which improve the bone density. Except the teriparatide, which increases the bone density, we don't have the drugs which increase the bone. Many of them only decrease or halt the progression of the bone resorption. So this area can be explored in the pulmonary fibrosis. And these two areas which I have told you, like muscle and the pulmonary fibrosis, which we are doing. The other area also, hope this molecule may give the good, because so many trials are going on, and I also explored the clinicaltrials.gov, where we have seen that 154 trials are going on as such among these areas, and most of the trials are on the pulmonary region, where the aspiration pneumonia and decreasing the COPD are going on. So these are some areas we can explore and get good results to translate the research out of the preclinical studies. At the outset, I would like to thank you, the IPS organizers and the team, for giving me the opportunity. Thank you. Any questions, sir? Sir. Hopefully, if it gets funded, sir, we are going to see, because it's the first repurposing drug molecule. We don't have any study on semaglutide on the pulmonary fibrosis. There are only two molecules that have been used in the pulmonary fibrosis, sir. One is an nintedanib and the other is a perfenadone. We don't have any other drug for use. So if these drugs get, or like we see the good results in the fibrosis, then it can be repurposed. Yes, sir.
Thank you so much.
Thank you, sir.
Most welcome. one. Dr. Richa Rathore. Madam, please welcome.
Thank you.
She is a PhD in biochemistry and farmer scientist, C at DRDO DePass. Farmer scientist. Madam, abhi kya kar rahe hain?
I'm scientist staff in DRDO, the past.
DRDO, okay.
I think there is some misprinting.
So, Dr. Richa Rathore focuses on high altitude physiology, muscle atrophy, redox biology, and molecular mechanism of hypoxia, with over 80 publications contributing to defense and high altitude health sciences. So, welcome, Dr. Richa. We would be very happy to listen to you and your insight.
Thank you, sir, for your kind introduction. So, after listening GLP, wound healing, and of course tuberculosis, this is little bit different topic, but very important topic. I hope you will make interest in this topic. That is high altitude induced skeletal muscle atrophy, a pivotal role of myokines. This is building of the past where the work has been conducted. As we know, DRDO is working for armed forces. So we are working for the armed forces who are deployed at extreme environment, like desert, high altitude, LIC, closed environment, sea. And basically my work is related to high altitude induced muscle atrophy. So before going to the topic, just I want to make a glimpse what is high altitude. High altitude is defined as the area of altitude equal to greater than 2,500 above the sea level. And we can differentiate high altitude into three parts. That is high altitude, very high altitude, and third is extreme high altitude. And the range is different, like 8,000, 1,200 is high altitude, very high altitude 12,000 to 18,000, and above that extreme high altitude. Where we go for visit like Manali, Mussoorie, these are not high altitude. This is normal altitude where we don't get any problem. So high altitude associated with number of environmental factors that we can't change, like solar radiations, UV radiation, humidity, temperature, hypobaric hypoxia, in which the main problem is hypobaric hypoxia. That leads to number of maladies like HAPE, haze, AMS. Acute mountain sickness usually start once you landed to high altitude, like headache, anorexia, insomnia, nausea, dizziness, vomiting, and except AMS, sleep disorder, decreased appetite, immune failure, and muscle atrophy. So I will concentrate on muscle atrophy that normally leads to the decremental physical performance. Why this is topic? Because physical performance is very important at high altitude, the soldier who is deployed at high altitude. If he is not performed well, the things will be different for the country. So these are some research area and international studies and national studies which says that high muscle atrophy occurs, decreased physical performance occurs at high altitude. And these are some Indian studies like the past studies that is started in 1987 who said that soldiers are having problem at high altitude. And this is the main study. This is done by Bharadwaj in 2000. And it is published by the past only, and they observed that 10-month stay at high altitude leads to muscle mass loss around 1.75 kg to 1.38 kg. So after these interesting studies, we have started work in vivo to establish the molecular mechanism that is responsible for high altitude induced muscle atrophy. As I said, hypobaric hypoxia is the main stress at high altitude. So we have exposed animals for hypobaric hypoxia, and we found that not a single pathway is responsible for hypobaric hypoxia induced muscle atrophy, but also it a collaborative pathways that is related to muscle atrophy at hypobaric hypoxia exposure. So we can see that chronic hypobaric hypoxia leads to protein modification as ROS is produced excessive at high altitude. That further lead to ER stress and inflammation. Now one of the interesting findings we found that muscle synthesis and muscle degradation is a normal pathway that balance muscle protein mass. But in the case of hypobaric hypoxia, we found that muscle synthesis increased in the initial stage, but protein degradation increased more than protein synthesis signaling pathway. Coming to the second topic, myokines. Why myokines? Because they are especially secreted by skeletal muscle. As we know that skeletal muscle is an endocrine organ that releases myokines during contraction and excitation coupling. When myokines regulate myokine differentiation, fiber type switching, and maintain muscle mass. In all the myokines, myostatin is the first myokine that is identified in the year of 1997. So if we want to establish which myokine is regulate hypobaric hypoxia-induced muscle atrophy. Then further, we have also wrote one review article in which we have established that muscle-secreted myokines not responsible for skeletal muscle health, but also it regulates other organs too. Like there is muscle-muscle crosstalk, muscle adipose crosstalk, muscle bone crosstalk, muscle brain crosstalk. So the myokines that is released for skeletal muscle that regulates other organs too. So this is very important for any human that skeletal muscle health should be good. So coming to the aim objective, we want to establish the role of myokines in skeletal muscle health during chronic hypobaric hypoxia. For that were exposed with hypobaric hypoxia for 1 day, 3 day, and 7 day. This is the chamber in which rat were exposed with hypobaric hypoxia. And as a result, first in the step 1, we have established the role of myokines that is secreted for skeletal muscle that is related to myogenesis and myodegradation. And we found that myosynthesis-related myokines like dacorin, irisin, and IL-15, they increased with the exposure of 1 day, 3 day, but they decreased after 7 day. And on the other hand, a myostatin that is negative regulator of muscle mass, it increased significantly from 1 to 7 day. So this hypoxia exposure alters myokine profile in the skeletal muscle. After that, in the second step, we established the down-regulated signaling pathway of myostatin and the hypobaric hypoxia. And we found that FOXO, SMAD3, SMAD4, MURF is increasing significantly from 1 day, 3 day, and 7 day. As in the signaling pathway, we can see that if myostatin is activated, it activates SMAD3, SMAD4, and it regulates AKT. It negatively controls AKT. So FOXO is released significantly, and all these significant molecules lead to regulation of MURF1 and atrosin that directly lead to muscle degradation. Behind myodegradation pathway, we also established the role of myosynthesis pathway. And we found that myosynthesis pathway, 1 day, 3 day, animal want to cope up with the thing. Means after hypobaric hypoxia, rat wants to cope up with the thing, so muscle synthesis increased, like myogenesis pathway is increased, but it is decreased after 7 day exposure. These are general morphological parameters and physical performance like body weight, muscle weight, grip strength, all are decreased as per exposure. After getting all the myosynthesis and myodegradation pathway related molecule, we have done string analysis, and we want to find a single molecule that regulates all the pathways. And interestingly, we found that myostatin is a molecule that regulates not only myodegradation pathway, but also myogenesis pathway. So it could be a marker for high altitude induced muscle atrophy. So as a conclusion, we can say that myogenesis play a prime role in regulating skeletal muscle mass under hypobaric hypoxia. Myostatin regulates, that is the molecule that regulates myogenesis pathway as well as myodegradation pathway. That it could be a biomarker for hypobaric hypoxia induced muscle atrophy. And these results also opens a new avenue to design the therapeutic strategies using myokines for skeletal muscle atrophy under high altitude conditions. Not a single person can do anything, but of course, teamwork can change the scenario. So this is the team. This is me, of course. These are scholars. This is Dr. Geeta Sirakumar. This is Navneet. Akansha is sitting behind. Thank you so much.
Any question? Anything? Yes.
Nice presentation, ma'am. My question is that you have discussed about skeletal muscle atrophy due to hypobaric hypoxia. That's for the stressed environment.
That's what the?
Stressed environment or higher altitude.
Huh.
But I am asking if any person developed that muscle atrophy living in the normal region, so that for that purpose, that pharmacological or pathological conditions will be the same or not? Or can we correlate with?
See, that is different from whatever the condition you are telling me, that is sort of sarcopenia at sea level. At this atmosphere, if we are facing muscle mass, muscle atrophy, that is called as sarcopenia. So the things are different. I have done one study to understand that just because of curiosity. This is not related to my work, my research topic, not for the institute work, but just for a curiosity. As you are asking me this question, I just want to get to know what will happen if sarcopenia is different from high altitude induced muscle atrophy. So we found in the case of aging or sarcopenia, the degradation pathway is high and muscle synthesis pathway is low. Means both things are low. That lead to how protein mass is in the balanced state. Once synthesis and degradation is a normal in a balanced state. But in the case of sarcopenia, synthesis is low, degradation is low. Then ultimately conclusion, protein mass is low. But in the case of hypobaric hypoxia induced muscle mass loss, what happened? Synthesis is little high, but degradation is more than high. Means if synthesis is 2% high, then degradation is 5% high. So there is again imbalance that lead to muscle protein loss. This work is also published in PLOS One. If you want to go for details, you can go for PLOS One. And another is Journal of Experimental Biology, in which I have published what is the difference between sarcopenia and hypobaric hypoxia-induced muscle protein loss. So you can go through, and if there any query, you can mail me. Thank you, sir.
Very hard.
Thank you, sir.
Brought a lot onto the surface, and it's a real problem.
Your appreciation means a lot, sir.
The way you are presenting, it seems that presenter is one.
Thank you, sir.
As you are not presenting, but you living the presentation. So very good presentation. I really appreciate it.
Thank you, sir.
Kudos to you.
Thank you, sir.
Thank you, ma'am. On behalf of organizing committee, we express our deep appreciation to our respected chair and the speaker also. Our respected chair, Dr. Rajen Dharna and Dr. O.P. Katare, please come on the dais. Now I request to Dr. Manish Jain, please. Kindly felicitate them with the token of appreciation. Dr. Rajendra Harna. For Dr. Rajendra. Speakers? Okay, no problem. Now,
Let us honor the speakers first.
Okay,
Job
sir.
is like labor work.
Okay, sir. कहाँ है? डॉक्टर प्रदीप कुमार. हाँ जी. मैम नेम क्या है? डॉक्टर करुणा श्री पौडीला. Now I request Dr. Manish Jain, please felicitate our chair with a token of appreciation. Dr. Rajendra Harna. To Dr. O.P. Katari. Thank you, sir. Sir, एक photo भेज सकते हैं?
अच्छा ऐसा है. चलो.
Speakers के साथ.
So all the presenters they can also come.
हाँ. हाँ. मैम.
आ जाएगा इसमें. हाई एल्टीट्यूड पे है. चलो हो गया, फोटो हो गया मतलब.
Thank you, sir. Thank you so much. Thank you, ma'am. Thank you, everyone.
Hello, 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 hello. Hello. Good afternoon, everyone, to all the distinguished delegates, esteemed faculty members, respected chairs, honored guests, and dear researchers. It is my privilege to formally commence Symposia 7, entitled Transforming Therapeutics and Medication Safety in India Using Artificial Intelligence and Digital Analytics. In an era where healthcare is shifting rapidly toward precision, automation, and predictive insight, this session brings together leaders who are shaping India's digital health revolution. This session is graciously chaired by Dr. Puneet Dhameja and Dr. Narendra Kumar. We are extending our sincere respect and gratitude to them for leading this important segment of today's program. I will introduce both our esteemed chair, Dr. Puneet Dhameja and Dr. Narendra Kumar. It's my honor to introduce Dr. Puneet Dhameja, professor at AIIMS Rishikesh. He's a leading authority in pharmacovigilance and drug safety with over 100 publications. He has made major contributions in ADR monitoring, regulatory collaborations, and training future pharmacologists. Our second chair is Dr. Narendra Kumar, an eminent hepatologist from PGI, Chandigarh, who specializes in liver disease and advanced hepatology research. He has significantly contributed to clinical practice. If I keep it far, there's no voice. And academic development while mentoring future medical professionals. We are privileged to have both Dr. Puneet Dhameja and Dr. Narendra Kumar as our chair for this session. We welcome both our distinguished speakers. Chairs, now I request Dr. Puneet Dhameja to introduce our first speaker, Mr. Tarun Gupta.
For this, I would like to invite first speaker, Mr. Tarun Gupta, who is founder, director, and CEO of Vaidya RS, which is a health technology platform which is dedicated to improving post-discharge care and medication adherence. He has almost more than 15 years of experience. He has more than 15 years of experience in AI and has received more than 40 national and international recognitions. And actively contributes towards policy making, and his focus work focuses on empowering patients to safer prescriptions. And we had a nice discussion about how these technologies and this platform can be utilized for the beneficiary schemes, health beneficiary schemes, including changing the outcome and or reporting adverse outcomes in the therapeutics. Dr. Tarun.
Okay, good afternoon, everyone, and good afternoon, esteemed speakers and organizers and chairpersons. Thank you so much for this opportunity and amazing introduction by Dr. Puneet, very generous and magnanimous. I am Tarun, and I would just like to correct, I'm not a doctor, I'm an engineer by background. And I've done engineering, I've done MBA, and I've worked in technology for many years. And Vaidya-Rx is my organization, or you can call it as a startup. We started at IIT Delhi, and now we work with various government and private hospitals. And for me, I'm a very passionate entrepreneur. And it all started with a real instance where I saw a lot of people, a lot of patients not understanding their prescription correctly or understanding it incompletely, where I have seen some people have lost their eye, some people have drank lactocalamine instead of putting on their hand, some people have misinterpreted medicines and have been admitted because of self-medication or in compliance or adherence. And that was where we started this organization called Vaidya-Rx, which was initially started with a concept called KYP, which is Know Your Prescription. It was 2.5 years back. And that is where my journey started, moving from as an engineer to solving a very significant, very prominent, and very profound problem in healthcare. And then we started building technology over it, and we built a software platform which connects patients, doctors, local pharmacists, and local diagnostics. We provide second opinion, we provide digital storage for prescriptions, and as well as a tool where a doctor can do a digital prescription. Now you must be wondering, that's software. So this is how the entire journey started. And I was very happy to have mentors like Dr. Puneet and many other senior pharmacologists who were guiding me through this. Now something significant happened while we were building this product. I realized I was sitting with few doctors at All India Institute of Medical Sciences, New Delhi, and they actually did a quiz with me. They said, can you guess how many prescriptions in one of the top national institutes are handwritten? I thought 50% are handwritten, 50% digital. He said, make another guess. And I was appalled, absolutely devastated to know in many of the hospitals, which are also the current state, almost 100% prescriptions are handwritten. And he actually pulled out that handwritten prescription and gave it to me. He said, Tarun, if you are a real engineer, if you are worth your salt, can you convert this prescription into gold, which can be used by doctors, physicians, clinicians, administrators to improve healthcare outcome, improve patient safety, improve patient confidence, as well as improve the way administrator runs their hospital. And I took it on us, and we worked with some of the senior professors at IIT Delhi, in fact, Stanford as well. And we built a platform where we started analyzing the handwritten prescriptions, which are very difficult to, and standardizing them and structurizing them and converting them into meaningful insights or the golden nuggets from them that can be used by doctors to actually make decisions which can be for better inventory management, for improving medical rationalization, improving therapeutic outcome, and also one of the leading things which people are talking about, AMR, is antimicrobial resistance, which was a part of G20 as well. We are also going to help in AMR surveillance. So this is a brief how, although AI, machine learning is a big topic, we chose to pick prescriptions and EMR, and then how we're going to utilize it. And the good news is that once we started this, it started as a small thought process, and fast forward to today, we are working with 4 private hospitals. We are working with AIIMS Bhopal, AIIMS Jammu, hopefully other AIIMS. We're almost on the verge of signing other AIIMS and private hospitals, as well as a couple of government hospitals. We recently signed an MOU with Steel Authority India Limited. And it's very surprising that they have got 53,000 employees and 5 hospitals, and they have no idea what's 700 crore worth of procurement is happening, and they have no idea. So they are using our tool to actually streamline it. So this is how the journey is, what we do. It was very important to tell because you need to know the context, and then moving on. So I'll cover the presentation as just for reference, but I will cover it because more or less I think we'll do question answers. Next one. So this is again, I would not like to highlight. This is a problem. I think as an entrepreneur, we always want to focus on solution. ICMR reports, my own personal experiences, various newspaper articles, various news reports are clearly indicating incomplete, illegible, improper writing on prescriptions, which we all should admit and work towards. But that solution always has an opportunity which we leverage with some senior doctors to create something as a golden insight tool for the doctors to make sense for various areas. Next one, please. Yeah, next one, please. This I've already covered. Next one, please. So this is the problems which we saw and I've already discussed. This slide will be shared for reference, and we've already published 5 to 6 papers in leading journals, and we are happy to announce that we are also presenting in international conferences as well, and we got good awards for what we are doing. So this is the app I was talking about. It started with the WhatsApp. Next one, please. Why I always show this slide, that entrepreneurial journey always start with something small, and why I give my example what I gave when I saw a father with an 8-year-old son who lost his cornea because he was putting the drop every single day. He felt it was getting corrected, but it had to be taken intermittently. I snatched the prescription and saw the doctor was correct to give the right dosage, right anti-inflammatory steroid, but the father had no idea of how it had to be given. And I saw the problem, and we started attacking it. So why I'm saying this? You have to start small. The problem is right in front of you, and healthcare has many opportunities that arise through a problem. So we started with WhatsApp. We started connecting 3 key stakeholders, and now in an ecosystem, we connect patient, doctor, local pharmacist, and local diagnostics. I give you a simple example. In AIIMS Bhopal, average time for a person to take a prescription from Amrit Pharmacy is 32 minutes. When we deployed our solution there, the time of taking the medicine got reduced to 7 minutes. A huge change, because they had a special counter for VaidyaRx, and the prescription would go from the app to the counter. This is how small things can change patient life, and we all should strive for improving patient outcome. Next one, please. This is our application. Next one, doctor application. Now this is where my heart lies, and I must admit openly in the forum, Dr. Puneet, who's sitting here, has been my mentor, has really helped us in analyzing some of the prescriptions. I do not come from a medical background, but if you ask for help, another entrepreneurship lesson, you get it. If you don't ask, no one is going to come there to help. So next one. We'll come directly to a solution because we're running out of time. This is the flow we follow. We take the prescription, which is handwritten. We structurize them. We put it into a standard form, and then we break them into 70 to 80 columns, which are proprietary, dose, duration, and then we give insights. Yeah, this is the process. We not like to cover it. Yeah, this is just go back. Yeah, so these are some of the elements of prescriptions which, if you look at it very minutely with a lot of attention, focus, you realize that, as I said, in a very wrong way, if you can convert that noise or garbage into gold, is where the real gold is. And when we started looking at each prescription with monomaniacal focus, laser focus, we realized that it has got all the information which a doctor might need over longitudinal, gives all the wealth for a better patient outcome or improving healthcare outcome. Next one. This is how we work. This is just a sample. There are tons and tons of elements we capture. Next one. And then we realized that any technology, and this is again for everyone's reference, because I see a lot of young people here as well, and people who want to understand AI. AI is nothing but simplifying things, and the output has to be very in a very simplified way given to the customer it is meant for. For example, Dr. Puneet, who's head of a prominent department at AIIMS Rishikesh, wants to look at how many medicines, how many antibiotics were prescribed in Dharma department last month. He will open the dashboard provided by us in collaboration with the hospital, and he can see which therapeutic category was number 1. Was it anti-ulcer, or what was the top antibiotic? How many prescriptions per medicine? How many were fixed drug composition? How many were not? How many were branded? How many were NLEM? What was the total cost of the prescriptions? What was the total medicine? And the questions, all the questions that a doctor might have with the data that we have, and the core engine that we have built, which is our proprietary, the answers you get sitting on the laptop on a daily basis. So this is what the power of converting unstructured, people who call it as redundant prescription, into something where Dr. Puneet sir can see and say, wow, this is what I needed to know to plan my inventory, to improve medical rationalization, therapeutic outcome, AMR, and so many other things that can come out of it. The best part is we worked with hospitals and we created papers. We already have written 5 papers accepted, and now we got it accepted in an international conference as well. Trust me, everyone is looking at India and the data, and if we can work together, nothing like it. This is OPD as well as IPD, and some people might say this is data reporting, this is data visualization, what's the analytics on it? Trust me, as we are getting more and more longitudinal data over a period of time for a particular department, for a particular hospital, we have started comparing hospital to hospital analysis, department to department analysis, drug to drug analysis, and there is no end to it. Some people say it could be a threat to data privacy. As an entrepreneur, you have to be very careful that data doesn't go out. We always take permission. And certainly it cannot go to a commercial organization, sorry to say, to any pharma company or anything who can misuse this data or take advantage of it to earn more money through us. But yes, this data is being asked by ICMR, NHA, and other organizations to help in policy framework. Next one, please. Yeah. This is the dashboards we have prepared. So we also look at antibiogram, and we compare which are sensitive, which are not. We have the antibiotics data, we compare, and as per the requirement of the doctor, we provide it back to them. Next one, please. This is the research papers, which are already published. And for example, some of the hospital, they ask very precise, okay, in diabetes, what was the top insulin given, which brand, what was the price as compared to generic. Whatever insights they want to know, for any reason, dubious reason, right reasons, whatever, we provide to the hospital as per them. Next one, please. Yeah, next one. So outcome achieved, we ensure that prescription is as per WHO. We are identifying the key trends, and we are identifying as per the norms, and many other reasons. Next one. That's it. Yeah, I think I already covered. We'll just end the presentation. I think we're already over time. Yeah. So this is some of the hospitals we are already at. Our services are at the prescription. Doctors are using it, patients, as well as the hospital. So we provide the software tool as well as the analytics. Everything has to go hand in hand. Always remember, analytics cannot be done without data, and data can only be done through a software tool, and software tool is built through technology. So technology and AI and its interpretation, everything is extremely important. If you want to, it's called completely the value chain. If you just only do one, it's very hard to crack business or hard to work with a particular client, because ultimately they would need everything. Our presence, I think, that's it, that's fine. So you can close the presentation. So I think I'll keep it short. My last submission is that AI as Dr. Puneet sir was rightly mentioning, AI, data analytics, machine learning, these are all tools. The first thing is to identify the right problem, which is a pain point. Then see how do we solve that problem, and then use AI to solve a real, genuine problem for a real customer to get real outcome, to create maximum impact for everyone and value for that. This is what we do, and this is how we have been working on AI. Thank you everyone. Thank you, Dr. Puneet. Thank you, Dr. Narendra. Yeah, I'm done here. Thank you.
I would like to thank you, Mr. Tarun, for a very informative and insightful session. Now, I will ask audience for any question. Dr. Puneet Dhameja also said, theoretically, artificial intelligence is not a very new concept. But in the recent years, since the development of very strong hardware, graphic cards, internet, and other facilities, including deep learning models and large language models, so the artificial intelligence has developed in the recent years. And it is common saying data is the fuel for the artificial intelligence. Sir, being a hospital administrator, I see everything through the eyes of hospital administration. My question to you is, how easily this platform can be integrated with existing hospital informatics systems? Mostly in India, government is using either e-hospital developed by NIC or CDEC, e-Sanjeevani developed by CDEC. So how easily this platform can be integrated with existing HIS system in India and electronic medical record system in all tiers of the medical primary, secondary, and tertiary care? Sir, please.
Thank you.
Sir, you can please.
Hello. Yeah, thank you so much for this question. In fact, this question shows and portrays that there is a serious intent for a government organization. As you said, you are part of hospital administration at a leading hospital. It shows that you have an intent to do something amazing, profound for the patient as well as for overall healthcare community. So answer to that question is, initially I thought it was difficult. Whenever as an entrepreneur you attack or you approach a problem, you always go with the pessimistic view, then you go to the realistic and the optimistic. At one half of my brain was optimistic, easy, and one half was always pessimistic. Why would they ever work? The good part is, if you have done the right hard work, where we did the pilot at few aims, where we did not integrate with, there are two organizations which we got to know. One is NIC, other is CDAC. CDAC is part of MITI, Ministry of Electronics and Telecommunication. And CDAC has presence in 13 states, and NIC is in AIIMS Delhi and AIIMS Rishikesh and others. And CDAC, somehow, they have an office in Noida. We realized that they have a lot of youngsters in their team and who are like doubling up as entrepreneurs. And part of the government policy, which is Ayushman Bharat Digital Health Mission, led by NHA, National Health Authority, they're all moving towards like ABHA ID, towards digitization. It is their target to have everything digitized. So when you, in management, it's called headwind. Headwind means when the policy or the intent of the leaders are there, then whatever you do will always add up. Provided your process is right, data security measures are right, your costing is right, and also you are not hurting their ego, because they should not feel that you are a competitor and you are trying to do everything which they are supposed to do. So answer to that thing, CDAC has done an amazing job in creating HIS, or Hospital Management Information System, which is called HMIS. And for a long time, now the data is getting entered into there, where doctors are willingly, intentionally putting the data, which was never ever there. So the first stage is getting cleared, which is the data is getting inputted. Where we come, sir, is at the second layer. We collect that data, churn that data, structurize the data, and convert into answers which doctors are looking. For example, if in a particular department, 5 antibiotics are given, how many are given for what dosage, duration, was it overdose, underdose, was it rational, etc., is the analytics part of it, where if someone comes with right intent and right specialized doctor, like we have mentors, many mentors from pharmacology department who are mentoring us because I'm an engineer, then organizations like CDAC who are IT focused are very keen to work. In fact, the good news is CDAC has itself written to us to sign an MOU with us for national level integration. That's a very, very big news. Might take 2 years, but the documents have also. This is how our existing government is now very, very proactive, especially with the Startup India and DPIIT promoting it without tendering, all that. So earlier it might be difficult, but it's difficult. You need mentors, no doubt, but it is getting easier. Yeah. So yeah, thank you so much.
Excuse me. Yeah. So how have you implemented with the Amrit pharmacy? Like you said, like if the prescription comes to the window of the Amrit pharmacy.
Yeah.
So what is the process after that?
So in our application, there are 4 stakeholders. So our application is live on Android, that's for public. But our application has 4 interfaces. One is for the patient, who can upload and put documents and do KYP, second opinion, or ordering medicines from nearby. Doctor, he can upload prescriptions. So in AIMS, what happens? The patient comes, doctor either takes a photo or does prescription through us, and with the consent of the patient sends it to the Amrit pharmacy. So there are 5 Amrit pharmacy in AIMS Bhopal, 1 Jan Aushadhi. Jan Aushadhi we have not integrated. In Amrit pharmacy in Bhopal and other SVS and SRM, there is no Amrit there. We have taken permission from their head of Amrit, not HLL, which is Hindustan Latex Limited, and their one person is having our app in their phone with a separate window, something like DigiYatra. So as soon as the doctor sends the prescription there, it hits their phone and they see the patient. The moment patient walks up to them, they are ready with the medicines. 30% there is error because some people even cannot pay 100 rupees. They haggle कि मुझे 5 नहीं 4 दे दो. मेरे पास 100 रुपए इसमें कौन सी मेडिसिन आ रही है? कुछ लोग बोलते हैं मुझे एक ही चाहिए. In 30% of cases, patients do not take the medicine. 70%, we were able to reduce the time by almost 80%. So we, in private, it's easy. It comes on the phone. We sign a contract because we don't take any commission or anything. We only take a due amount from the hospital. So everything is transparent. Yeah, that's the process. And they give the consent to us to analyze the prescription, which has only to be submitted to the project coordinator who's coordinating for the hospital. And then he has to distribute to HODs or director. That is their prerogative. We don't give it to anyone who asks. We only have one point of contact because this is sensitive information.
Nice initiative. I'd really like to congratulate on it. Being a pharmacologist, my one point to ask about is, do you calculate daily defined doses also? Because as I understood with your previous answer that it is not 100% capture. So are you aware of daily defined dose or not?
Actually, I'm not. It's like we get a data which is in a weekly or on a monthly basis and their dosage.
Yeah, actually daily defined dose would be for all the patients who are either attending the OPD or IPD, but because probably this is a partial thing and it is voluntary, so probably it's not possible at yours. Okay. No problem. Thank you.
Okay. Thank you, sir. Any other question from the audience? So I would thank you, Mr. Tarun Gupta, for the informative session as well as for the initiative in healthcare artificial intelligence. So our second speaker for the session is Dr. Sangeeta Yadav. She is Associate Professor in the Department of Pharmacology at Dr. Baba Saheb Ambedkar Medical College, New Delhi. She is deeply committed to medical education, guiding both undergraduate and postgraduate students while contributing to the research, curriculum development and departmental academic growth. Her expertise span is experimental and clinical pharmacology with a strong focus on mentorship and academic excellence. Now we welcome Dr. Sangeeta Yadav to enlighten us with her insight on the topic, Artificial Intelligence for Pharmacovigilance, Enhancing Patient Drug Safety Through the Advanced Technologies.
Good afternoon, all of you. Thank you for the introduction, sir. And good afternoon, all of you. My topic is to harnessing artificial intelligence for pharmacovigilance and enhancing safety through advanced technologies. So I'll be discussing about what is artificial intelligence, what are the challenges and gaps in pharmacovigilance, what is the role of AI in pharmacovigilance, and then what are the challenges with AI going forward. We are using artificial intelligence way before it came into the market. We have been setting our alarms using Siri from past 10, 15 years. So that was what I remember as the first artificial technology that I used. It is artificial intelligence is intelligence demonstrated by machines in contrast to natural intelligence displayed by humans and animals, and a broad term that encompasses any code, algorithm, or combined technology designed to mimic human intelligence or behavior. So we have artificial intelligence is an umbrella term which has multiple technologies, and in pharmacovigilance mainly we are using machine learning, deep learning, and natural language processing. So what is machine learning? Machine learning is simply algorithms are developed to help making decisions when new situations arise. Here the models are trained with the data which is available, and it will process the data and give you the result. It is useful when we are searching for information in a text, like in case narratives, in case of pharmacovigilance. Deep learning is again a type of machine learning only, but it is more advanced as compared to machine learning, and it is on the concept of the cerebral neurons of the humans. So in deep learning, artificial neuron networks are there, and there are layers of artificial neurons. Each neuron will receive information from the neuron in the previous layer and transmit it to the next neuron layer. And this is, the deep learning is utilized for natural language processing. So with deep learning, it can analyze newer situations also for which the model has not been trained, and going forward while processing, it will develop its own thinking, and it can come up with solutions of things for which it has not been trained. Natural language processing is ability of the computer program to understand, interpret, and generate human language, and it is useful for converting audio calls into text data. So what are the challenges as of now? What we are facing is, first thing is data overload to the regulatory authorities. And most of the data which is present is in unstructured form. Also, the manual pharmacovigilance process is very time consuming, and it is a repetitive process, and our associates, they are manually doing the repetitive process instead of utilizing the time in the analytical part. Then manual process has limited scalability. Another important challenge is underreporting of the ADRs from the physicians, from the society. And also untapped sources like apart from the spontaneous reports, which are the main source of ICSRs in our pharmacovigilance, there are other sources like electronic health records, insurance claims, blogs, forums. All these are not tapped as of now. So that is again one of the challenge. So in pharmacovigilance, case processing is the main thing which is happening, and it is a very structured process with case intake, its evaluation, follow-up, and then distribution as required. Now, how AI can be used for case detection? First, we have to identify the unreported cases. Now, what are the other sources apart from the spontaneous reporting? The cases can be found in the real-world data with the electronic health records, insurance claims, registry databases, medical literature, websites, papers, health blogs, mobile applications, wearable devices, social medias. These are the other sources apart from the spontaneous reporting where the cases can be found. And utilizing these untapped sources, US FDA in 2009 had come up with Sentinel process. So in the Sentinel process, what the US FDA was doing, it was looking for the ADRs in the electronic health records of the marketed drugs. And utilizing this process, they had come up with the intussusception risk by rotavirus, and it was reported by Ye et al. In 2019. But with the electronic health records also, the unstructured data, which is the rich source of the case reporting, is not very much extractable because it is the codes which are present in the electronic health records. Then comes the natural language processing technology. The natural language processing is the program that gives computers the ability to understand text and the spoken words similar to the human beings understand it. But the problem with NLP right now is that context matters. At times, the language in which the humans are speaking may not be taken up correctly by the AI. So the context is very much important. So for example, break a leg, we may say it normal conversation, it means something, but the AI may pick up it as a ADR, broken leg. So this is how the NLP is picking up the information from the spoken language and the text. It can pick up age group, gender, seriousness, severity, and adverse event. It can also classify the text. It can extract the entity, transform the unstructured data into structured data. It can synopsize the structured data. It can also interpret the tone and an emotion. And it can interpret the intent in the relevant answers. So these are how the NLP can be utilized in pharmacovigilance in all these areas. Then data can also be extracted from social medias like Facebook, Twitter, blogs, health forums, even before when they are spontaneously reported. So WebRadar was a project which was launched in 2014 in Europe, and it was developed to extract the ADRs from the Twitter. And with the help of that, a lot of ADRs were extracted. Now, India is also on a way for technology and artificial intelligence. In 2023, AIIMS Delhi, PGI Chandigarh, and AIIMS Rishikesh, they were declared as Center of Excellence for Artificial Intelligence. And the India is then coming up with our electronic health record with ABDM, Ayushman Digital Bharat Mission. So going forward, if India can also integrate the electronic health record along with the pharmacovigilance. So for example, this is a page of the electronic health record. Now in this health record, if one section of ADR can also be incorporated going forward in ABDM, so then the ADRs can directly be entered at the patient level. Of course, data privacy has to be taken care, and that can be integrated with the PVPI, so that can increase the reporting of ADRs. Then again, these technologies can also assist in literature monitoring, and they can search and extract information from scientific journals, from conference proceedings, and they can also identify the ADRs with specific drugs, medical devices, and they can track the changes in safety profile of a drug over time and identify emerging safety concerns as well. And these technologies can also prioritize the articles which are to be reviewed by the humans, expert. Then coming to case processing. In case processing, again, most of the data is present in unstructured written format, which cannot be, again, that becomes a very exhausting manual process to extract the data from there. So AI can be used to convert the unstructured data into structured data, and that can be, again, pick up, and then further technologies can be used to automate that data in the PV system. So OCR is one technology, that is optical character recognition, which can convert the written text into text It can also convert image with text into text, PDF, scannable, printable documents into text. It can also, the technology, NLP technology can be utilized to convert speech into text. So it can be utilized to extract the data from the calls which we receive in the PV centers. So audio calls, from audio calls, the unstructured data is collected, and using NLP that can be converted into text and structured data. This is to show how a phone call or an email or a letter, any information from these sources can be utilized to extract data for the system. Now using speech to text technology or OCR technology, the data can be collected from email and letters using OCR. From the phone call, again NLP can be utilized to extract the data, and then again further this digital data can be utilized to extract the information about main things, which is adverse event, reporter, patient details, cases, and that can be again automated in the PV system. Then again, the technology can also be utilized for automating the medical coding. So MEDRA coding can be directly given to the terms which are being extracted. Now this is what WHO has come with. WHO has come with WHO Drug Coda, which is again an automated coding engine which they are utilizing for automatic coding. Then seriousness prediction, neural networks can be used to predict the seriousness of the adverse events, and they can be trained with high quality data to learn about the patterns that are associated with serious events, and once trained, the model can be used to predict the seriousness, and this can help to identify and prioritize the serious adverse events. For causality assessment, we have different models which can be used, like decision tree, random forest, neural network, support vector machine. All these technologies can be utilized for causality assessment. But the thing to remember here is that these models have to be trained with very high quality data, representativeness, and well annotated data for performance. Then for a case, follow-up can be scheduled using AI. Automatic warnings can be sent, and the reminders can be scheduled. AI-assisted signal detection and monitoring, NLP algorithms can be used to extract the data from unstructured and to identify the patterns and trends that may indicate potential safety issue. So it can automatically analyze the reports. It can predict, the predictive modeling can be used to predict the likelihood of particular AI based on the patient's characteristic and their use of medication. Then data visualization can also be done, and graphs and maps can be made to analyze the patterns to indicate a safety issue. So these are all the areas in pharmacovigilance where these technologies can be utilized, from case intake to signal detection and risk mitigation. Now, what are the challenges with utilizing these technologies in pharmacovigilance? First thing is data quality. So with the available data, since all the in the pharmacovigilance, one thing is underreporting of cases. So the available data is not all representative of the data throughout the world also. And especially in case of India, there is a huge underreporting. So the available data quality is one issue. Legal and ethical considerations of data privacy and protection is the second thing. And then third thing is integration with the existing systems. So these three are the major issues. And now recently the upcoming issue is the black box AI, where the even the persons who have made the algorithm, they are not able to track from where the AI is giving the result. So that is another upcoming issue with AI. So for protection of data acts, there are certain rules and regulations. Convention 108 is for protection of individual regards to automated processing of the data, followed by US. And GDPR, that is General Data Protection Regulation, it applies to European countries. And coming to India in 2023, we have DPDP Act, which is Digital Personal Data Protection Act, and it is similar to GDPR, and it prioritizes consent and individual rights and privacy to the personal data. So summarizing and concluding, the use of AI in pharmacovigilance is huge, and from identifying the case reports to causality assessment to automation of individual case reports to automatic data entry and to risk mitigation at each and every step, artificial intelligence can be utilized with human oversight. So I'll conclude with that. Thank you so much. These are my references.
Thank you, ma'am. Thanks for the insights which you gave. Next speaker is going to talk on a similar topic, so we can reserve the questions after the second talk, because the topic is almost similar about AI in pharmacovigilance. So we will have common set of question-answer session in common. So I invite Dr. Sandeep Kaushal, who is Dean Academics and Professor and Head of Pharmacology at DMCH Ludhiana. He is a distinguished pharmacologist with major contributions to neuro and psychopharmacology, pharmacovigilance, drug utilization research, and clinical pharmacology. He is a recipient of honors including Dr. B.C. Roy National Award and international fellowships in pharmacogenomics. And he has over 50 scientific publications and extensive academic leadership. He also pioneered skin prick testing in children as DMCH first certified allergy specialist. And now I welcome Dr. Sandeep, and just not to forget that he was mentor during my post-graduation also, and his sessions on pharmacokinetics are very much valuable. So he has been into data management and data mining and since a long time.
Thank you, Puneet. Thank you for the kind words. At the outset, I would like to thank the organizers for giving me this opportunity to be with you. So many things will be common with my previous speaker because the topic is nearly similar, but I'll try to add some differences which need some elaboration. So we already are aware about that what is pharmacovigilance. It talks about not only detection, assessment, understanding, and the most important thing is prevention of ADRs. And prevention is the key. And in order to reach that key, it is very important that ADR gets reported. The seriousness of the issue is this, that the people who talk about pharmacovigilance are the ones who have to evaluate ADRs, not the ones who have to report ADRs. So reporting of ADRs as an individual, whether you are a pharmacologist or other, is our duty, we should do it. But more so, it is basically the clinicians who have to do it so that we can have a big data to look into. AI is now transforming pharmacovigilance by making it faster, more accurate, and in a cost-effective manner. So if you look at the beginning of pharmacovigilance was started with Dr. Y.K. Gupta. So he's fortunately here, and it's a proud moment for all of us. So see how we have improved in improving the reporting of ADRs. So now we are having approximately 2.5 to 3 million ICSRs getting reported into the system. So this is both a thing to be happy about. I'm not saying proud, because we are a population of 1.5 billion by now, and this is a very minuscule number. But still, if you see, there's a trend which is improving. So we all need to keep on continuing and doing it. And this brings into forth the challenge that such a large data set is available, and therefore doing it manually would have its own problems. As my previous speaker has already talked about the problems, which are being a large data set, time-consuming, error-prone, low scalability, delayed signal detection, and then most importantly, it is resource-intensive. So therefore, it creates a problem trying to find out a needle from this haystack, which is there in this large data set. And this is a problem not only for us, but it's a problem as a regulator, is a problem as a medical experts, pharmacovigilance experts, for industry, because it has a lot of regulatory issues associated with it, and also for the National Pharmacovigilance Program, which has taken a set up now. Now, this is just a representation of what my previous speaker talked about. We look at so many tools. We talked about machine learning. We talked about natural language processing, multimodal data fusion, knowledge gap constructions, deep learning architecture, statistical signal. So this is taking inputs from all the sources available. So presently, when we are talking about pharmacovigilance, it is not only looking at ICSRs, but it is also trying to get the data from all possible resources, because then it gives us a very rich source, and it helps us to extrapolate. And only by getting information and signals from elsewhere, we can make it a more scientific way of giving guidance to what things to watch for with which drugs, and also make sure that safer drugs are available for the population to use. So they use various types of tools, they use various types of data representations. Fortunately or unfortunately in India, presently the insurance data is not used that commonly for data mining and others, whereas in the West it is very common. And therefore it brings another challenge, because insurance again is going to be a business-driven model. And once this data is available, they would be deciding upon the premiums. If you are having ADR reported some to its drug, which carries a risk of maybe producing a Steven Johnson, which could be life-threatening, very expensive treatment, they might raise up the premiums. There could be genetic predilection to these ADRs, and then it could have an effect on the premiums which are going to be charged by these insurance companies. So as my speaker has talked about, that we now, when we use about pharmacovigilance, we're using sophisticated mining tools, which are helping us to analyze sizable databases, whether they're medical literature, adverse event reports, they are social media, and even electronic health records. Fortunately, the government of India is making a lot of initiatives to go in for electronic health records. But again, it's time consuming, and there are going to be presently, as ABHA ID has been made mandatory for all the hospital patients who get admitted. And there is a process in place that with this ABHA ID setting in, they want all the data in a format which is easy for the machine to read, to analyze it, because if you're going to generate data for the population whole of the country, it's going to be massive, massive database. And obviously there has to be some structure. So they are trying to look at some feasibilities, and there is insurance sector is putting a lot of pressure to get this data in an electric format so that it's easy for them to analyze, easy to them to take a decision tree to give insurance, or to give the whatever compensation has to be given, or whatever repayment of the charges has to be made. Then there is going to be, it's helpful by giving us the semantic analysis of adverse event reports which are getting collected in this databases. It's looking at real-time signal detection. That is very important, because what has happened in the past is that signals which have been there lying dormant because of lack of manpower, because of lack of understanding, because of lack of algorithmic tools, which could have predicted them earlier. So now with these tools availability, it's much easier to pick the signal at the earliest. And this really helps us to minimize the risk to the patients and to the population which is using these drugs right, left, and center, because they are available in the market. Then wearables is becoming a big important thing. So ma'am showed a very good example of Siri. And now Siri not only gives you time alarms, it also monitors your heart rate, it also monitors your respiratory rate, and there are softwares available which will even predict your ECGs for arrhythmias. So my former teacher and Dr. S.C. Chopra had an arrhythmia which was detected by his smartphone, and that is why he could start the treatment immediately. So see, this is the advancement which is happening. But how many people are aware that this is usable? So that is also a challenge. So once we are using these wearables, these bring a very good way of monitoring. But again, the importance of this, like for example, the treatment of arrhythmia or a treatment of hypoglycemia should happen in real time, because they are going to make a difference of life and death within seconds. If you're detecting it, you're getting treatment, you would be saved. If you're missing it, and you might be dead. Then also the population health surveillance. We recently had this COVID pandemic. So if we are able to have this information available, it would help us in planning for these future pandemics which might happen. Could be a part of a bioterrorism, could be a part of war, or could be a part of effect of nature on all of us. Then also it helps us to have an enhanced risk-benefit assessment. And remember, this risk-benefit is a very delicate balance. all the medication therapies which are happening, they're always based on low risk and more benefit which is there. But this dynamic can change as more and more information comes in, because when the drugs are approved at phase 3, information is very sketchy, but until and unless phase 4 happens and this continuous information coming in, it will give us a real parameter into that what is the paradigm shift between the risk and benefit analysis, and whether it is possible to or safe to continue with the medications which are available in the market now. Then there's going to be annotation and coding. My previous speaker talked about this coding which is there. So we talk about MEDRA, we talk about other methods which helps us in coding, so that making sure that we are able to categorize the data at the earliest. Then another important thing that we're looking forward now with the AI is predictive modeling. So pharmacogenomics is coming in a big way. In addition to this, the databases which are giving us, they are helpful in creating a phenotype of a patient which is with a particular disease and a particular drug regimen, which can help us to find out that which patient is at risk of developing an ADR or toxicity or any other manifestation which is not beneficial for the patient. And this can be used really with the advanced AI. Then we already have talked about integration of electronic health records. Then automated literature review. Obviously, every day or now, most of us have our advanced technologies on their phone, and just we have an article we load up and it gives you in a single paragraph that what is a summary or whole of it. The same thing is being done by creating the information from the literature which is available. And then natural language understanding, so taking into consideration patient narratives, social media posts. Though in India, social media posts does not talk that much about ADRs which are happening as such, but in the West it is very common. And there are many patient help groups which also keep on posting information, creating awareness, or advocacy groups which keep on adding information for the various medications which are available, what is their safety profile, what is the ADR profile with use of those medications for those patient age groups. Then obviously any procedure or any process we had, it is not possible to have a development until and unless we have an inbuilt quality improvement tool in it. So the pharmacovigilance, when we are using artificial tools, artificial intelligence, it would be having a continuous quality improvement processes which will help us to get information from all possible sources and make sure that the output which comes is improving with more and more knowledge which is getting in. Then adaptive surveillance tactics. So obviously, once we have these monitorings available, goals available, the data gathering techniques and risk assessment criteria, so they help us to prioritize which areas or which drugs to go in for focused pharmacovigilance, so that we can be sure about or we can generate data about their safety issues and efficacy issues. Then integration with regulatory reporting systems. So there are many people from industry here. So again, reporting an ADR is a mandatory from the industry point of view. And obviously it is a bit of a regulatory component duty, a bit of a pain point also. But again, it is mandatory, it has to be done. So if we are able to have AI tool which makes sure that all the indices or all the data points which are required, they get entered, and it's going to flag out if something misses out as such. Because most of the pharma industries have their own programs which are used for ADR reporting. And once they are on the same platform, it will make sure that this is going to be helpful to everybody. And because the regulatory agencies would be looking at the same points, whether it is from industry A, or a company A, company B, or company C. But it will make sure that there is a common data point which is captured from all these pharma industries which are required for the pharmacovigilance. Then obviously, once we have so much of data available, it helps us to improve the signal validation. The stronger the signal, more is going to be strong is going to be the recommendation. And then longitudinal safety monitoring. So obviously, when we're having these data points which are captured on a longitudinal way, so it will help us to have a more profound ORF data, which is more solid and more grounded with evidence. Then it also can be integrated with health information exchanges, which can give out alerts, which can give out information to the pharmacies, which can inform regulatory agencies, and as a result of which data safety or drug safety can be assured. Then we also, it can help to use drug interaction predictions. Already there are many softwares available which predict even when you're prescribing it on the app or you're prescribing it, electronic prescribing, it gives you a highlight that, okay, there's a possible drug interaction in this, and which could be overruled depending upon the patient need at times. And again, using this information can help us also to have a drug interaction prediction. Obviously, once this data set is available, it helps us to give out information as automated risk communication. So obviously, any patient started on a drug can get an automatic communication saying, highlighting what are the areas which are possible for this drug. Now, this has been used in a number of centers and all. There are a lot of publications which are talking about applicability of AI in pharmacovigilance. So I'm just referring to 2 or 3 bigger reviews which have proven that when we use these tools, not only the sensitivity and predictability increases, it is much better than the human. Okay? And then when we look at this comparisons which are in the predicting the needed dose for a patient characteristics, predicting clinical response from a pharmacological treatment, predicting occurrence or severity of adverse drug reactions during risk of drug inefficacy, predicting drug consumptions, predicting drug-induced length of stay in the hospital. So it has been much better as compared to other methods, and therefore it brings the point that these tools are really useful in pharmacovigilance. And similarly in this study, so it has shown that the identification of ADRs, which was there in 57.6% of the cases which they had taken up. Processing safety repository was there in 21.2%. They were able to extract drug-drug interactions in about 7.6% of the data that they used. So I'll be talking about 5 different systems that are being used very commonly. One is IBM Watson. So it is using both NLP and ML algorithms to analyze structured and unstructured data. The advantage is it enhances drug safety monitoring and decision making. But the important thing to remember is it is expensive, no doubt, and there is a potential for algorithm bias. But the limitation is it relies on data quality, because ultimately whatever software you use, whatever AI you use, it is going to feed on the data. If your data is not good, if your data is not proper, you will have wrong conclusions. And therefore, it can affect the accuracy. We talk about AstraZeneca AI-driven pharmacovigilance system. Again, it is using adverse event signal detection, identification, leveraging advanced machine learning analytics. Again, it has better AE detection and is following the regulatory compliances. However, it requires a lot of skilled personnel. So there is a training which is mandatory before we can start using this software. And obviously, it is again a cost-intensive thing. It sometimes overlooks rare AE. So if there is a weaker signal, it may not be picked up. And it also can give false positives. Then we talk about Advera Health Analytics Signal Mine. It streamlines adverse event monitoring and risk assessment processes, which improves efficacy and accuracy in AE monitoring. The disadvantage is there's a limited scalability and integration challenges, and it relies again basically on data availability and quality. The fourth one is Oracle Health Sciences. So it automates AE reporting and signal detection. It is again costly, it requires a lot of maintenance, and requires a continual algorithmic validation and monitoring. So it is a part of a process improvement or enhancement which it requires. And lastly, we talked about FDA Sentinel initiative. Madam talked about it, that it could pick up a rotavirus, rotavaccine induced interception, which is there. So it integrates data from various healthcare sources. It has a rapid detection and response rate to emerging safety signals. However, there are issues about privacy and data security, and again, it relies on data interoperability and standardization. So no longer that, as you can see, there are softwares available, companies are using it. There are issues regarding regulatory requirements. There are issues regarding data privacy. There are issues regarding ethical aspects. And obviously, the regulators are trying to evolve. And they are trying to make use of this information, making sure that whatever drawbacks there are, the ethical issues there are, the validity issues there are, they are taken care of. So when we look at, again, the same algorithm that we talked about in the beginning, so what are the challenges? The most important challenge is data. So can anybody tell me what are the things which are required for an ICSR to be called as a valid or complete? Anyone? Yes. So there are 4 basic things. And remember, there's so many other things which are missing out.
So at present with this, we are able to increase from a completeness ratio of 0.6 to 0.94. It is still better, but remember this is what is, we have kept a bar a bit low. But again, looking at that we are able to achieve it, it makes sure that there are going to be so many issues about the data which is getting captured. And then there are going to be issues about drug-drug interactions. We have a population, we are a diabetic capital of the world. We have so many diseases which are happening, and we have so many types of pathies being used right, left, and center, which have a potential of drug-drug interactions. They are again not taken care of at times. Then there are delays in the signal detection, looking at the enormity of data that we have with us. Then there are issues with the regulatory compliances which have to be met and they have to be mandated. Then there is issue about data integration, because as I said, each industry has their own software which does this data entry. We have this separate data entry system with the under Pharmacovigilance Program of India, which can be there. And obviously, there are tools being used. There's a manual review. Madam talked about black box, which is a big challenge. Remember, it is a tool. And when we talk about a tool, it has to be mastered by humans. So despite having these tools, despite giving out signals, there are certain issues when it has to be intelligently looked at from a medical personal point of view, whether that is relevant or not. So no matter whether we are using data mining algorithms or we're using traditional pharmacovigilance methods, we have to triage the outputs, make sure that signals are produced. These signals are taken forward, keeping in mind that how they change the safety and the efficacy ratio which is there, or risk-benefit ratio which is there. So despite these advantages, there are limitations. The most important being the data quality and completeness. Then obviously there are issues about ethical considerations, the name, the other things which are there. Then we have nuances about clinical judgment that I just talked about, that is the black box. And then we need more comprehensive and long-term research. It is not one time, it has to evolve over a period of time so that we are able to take care of all the constraints that we have. So no matter, AI holds a greater promise for improving efficacy and accuracy in pharmacovigilance, challenges related to data quality, ethics, interpretability, and limited research must be addressed to before large-scale deployment of this. To conclude, we are using AI to enhance the reporting by increasing the speed, accuracy, and efficacy of drug safety monitoring. We have to enable the collection processing and analysis of this vast data sets. Machine learning helps in streamlining workflows, reduce the manual workload. It augments, but remember, these are tools and they will never replace the human expertise. It is always required to. So one must not be fearing about it, that how it is going to change the pharmacovigilance industry. Industry is going to remain. There's going to be a role of people still in it, but the important thing is people who are enlightened, people who are trained in using these tools efficiently. It is a successful implementation requires careful attention to data quality, interpretability, ethical considerations, patient privacy has to be taken care of. There has to be an explainable and adaptive AI frameworks. It should be able to enhance the public health outcomes, foster innovation, ensure safer and more effective pharmaceutical products globally. Integration of AI with human expertise, it promotes a proactive, patient-focused, and collaborative approach for drug safety. So no matter, the ultimate aim that we have when we're using these AI tools is try to have a faster adverse event detection, enhancing safety monitoring for these patients, and please remember all these tools are going to cost us an arm and a leg. So that is very important, and obviously these are algorithms. You need to be trained to look at it. So these tools are here to stay. You must first understand what is the basis. If you understand the basis, you'll be able to use the tools much better. And in case there are any shortcomings or something, only then you'll be able to pick out the problems and try to correct them. So that is all that I wanted to say. Thank you. Thank you very much.
I would like to thank you, Dr. Sandeep and Dr. Sangeeta, for letting us know the nitty-gritty of artificial intelligence in pharmacovigilance, including the benefits, limitations, and what are the challenges. Sir, you may please have a seat here. We can discuss. So, as a hospital administrator, my understanding to pharmacovigilance, there is a statutory requirement of the National Medical Commission for all the medical colleges and tertiary care hospitals. There should be one committee or team of pharmacovigilance. The main role and responsibility of the pharmacovigilance committee is first is to record the ADRs, medication errors, to collect the data, manage the data, analyze the data, then to train the healthcare providers regarding the ADRs and medication errors. Third is to develop the policies related to the medication errors in the institute, and later to develop the SOPs and all. So I would like to ask one question. Suppose, considering the environment of the this India, how the infrastructure and other, we have some challenges, unique challenges of being a developing country. So if we integrate the artificial intelligence in pharmacovigilance, sir or madam, anyone can answer my question. What you think, what is the most important benefit we are going to get? Which will be which right now we are using the traditional method, as sir has said. So if we integrate the artificial intelligence in a tertiary care hospital, what will be the immediate benefit we are going to see?
Bayesian analogs are being used, or rhythms are being used for this, but these tools, if we have, so it will be able to give us much faster signals which are there. So it can also help us to focus on those are the drugs for which we need to do a focused pharmacovigilance. So obviously the time interval will reduce. So it's our the National Coordinating Center is doing a remarkable job. So they come up with signals, but obviously it will increase the pace at which we are able to pick up the signals. The only important consideration is that we need to improve the quality of the data which is there, because presently the only important source as of now is ICSRs. So if we want to have the data from social media, we want to take data from other inputs which are there. EHRs are not very common. It is only a few hospitals which are having, mostly private hospitals, some general hospitals, but the bulk is so large. So if we are going to take a data like that, it would be skewed, it would not be balanced. So we need to have more and more data coming in from different sources, which will help us to predict signals, which will help us to test these and try to give out guidelines about preventing or to using, to stop, or to come out with safety things which are there. And this is definitely going to cost us a bit. And what I can say, for patient safety, cost is not that important, but the most important thing other than cost is the data integrity and the data robustness which we are there. Training, our Indians are known to, again, rise to any occasion. So we rose up to create vaccines for whole of the world. We rose up to have so many startups. We had a first speaker talking about so many things. So rising to that is not an issue. But the most important thing is the raw data which is there, it needs to be more robust. It needs to be more strengthened. We need to have more inputs coming on that. Only then the strength of this would improve.
Thank you, sir.
I have been informed that the third speaker, Mr. Amit Verma, who is from National Health Authority, he is not here. He will not be able to come due to some emergency. So I would like to conclude this session with the overview of what you should think about after this session. The first speaker catered to how the basic challenge, how to get data into a system. The rest of the second and third speakers, Dr. Sandeep and Madam, they discussed about how to analyze that data. But the first major challenge is getting the data into the system. That has been dealt with a simple technology use of WhatsApp, which has been demonstrated by the first speaker from VaidyaRx. Now, once you have the data available of the prescription integrated with the reporting data, ADR reporting system, you will get real-time issue about the medication safety, the prescription data, the prescription analytics, and about the ADRs generating. Once this whole data is available in the right platform or in a common platform, it can be used for further analysis. We can share data with the PVPI program, and then we can generate signals. So the potential is immense. The only thing that as a young fellow, you are just starting your career, you are starting your professional lives, you can always be a part of that system where the data is integrated and it can be analyzed and give right results, which is actually AI. AI gives results. AI not just only gives the results manually using Excel. It gives you predictions. It gives you answers. So that can be your next area of expertise development or your area of interest in future. And I am hopeful that in next 10 years or so, we are going to see immense changes in this area. With VaidyaRx as just this startup, we are going to see, I see immense potential in that kind of work, which is integrating, which is putting the data first, analyzing it, and giving the results. Giving the results still is waiting, and it will depend only on the data which we have. So these are my concluding remarks, and I could see Dr. Y.K. Gupta is there, so my regards, sir. Any comments or questions?
No, I don't have any comments, but I just compliment. This is a wonderful session which you had. I just missed initial first talk and maybe half of balance, but it was wonderful presentation. Medicine safety is becoming more and more important, and Government of India is paying more and more attention to patient safety and medication error, and that's why a lot of importance on digital prescription, digitalization of prescription, and a lot of avoidable medication error, I would say. As a chairman of Signal Review Committee, what major challenge, one of the important challenge we find is what Sandeep has very rightly mentioned is the correctness of the data. Or see, sometimes we create a signal, but when we go into the detail of the case narration, we find it, oh, this is missing, this is missing, and therefore the decision on the signal review panel, we have to revert back. कि यार उसको बुला के पूछो कि यार वहाँ से इसने लिखा क्या है. So at the grassroots level, when the reporting happens, we have to be very vigilant that what they are reporting is correct, what they are reporting is they understand correct, and it is not just merely, because we have created more than 1,000 ADR centers, and when we give more pressure, then the report quality sometimes decreases, because वो उनको लगता है कि यार कुछ भेजना है. उसमें हमको AI can also help in finding out whether this report is adulterated, manipulated, or this is correct report. Uska bhi, because there are apps available which can easily find out duplication of the report. But if you can also create something which can create a pinpointed problem कि यार इस report में ये problem है, let us go back to the investigator or the reporting system. You have, I think both of you have given a lot of software programs starting from Oracle, and so we have to look for what is the Indian data set. वो हमको एक होना चाहिए rather than. I will just give you one example, because how we started Pharmacovigilance program of India in 2010 and AIMS when we started 10 centers. Before that, India used to pay $10,000 to Uppsala monitoring centers to put our data into their program. And then they used to give the mature data, means information out of it, sell it to Indian industry, sell it to healthcare professionals across the world at a heavy cost. Hamara data, then they came to us, we had a long discussion at AIMS. It was just like independence से पहले हम सूत भेजते थे और ब्रिटिश हमको कपड़ा बना के, टेरेलिन और कपड़ा बना के हमको 10 गुने दाम पे देता था. Then when we argued कि हमारा next target है, in 3 years we will reach 1 lakh data. जब उनको ये समझ में आया, तो within 3 months they said कि अब हम आपसे पैसा नहीं लेंगे $10,000. So that was our first victory of the. And after that they started calling us that we want to have a center at an IPC, when it shifted to IPC. So that is the strength of our data. At that time we used to take decision whether it is a rofecoxib, whether it is any other drug, whether it is a nimesulide, or based on the data which is available outside Scandinavia, Europe, anything country, for a country which is 140 crore population. So that was, in fact, I will just give you some narration background, because when there was some deaths occurred because of some vaccine and some drugs, so there was a parliament question, and Ghulam Nabi Azad, who was minister, called DCGI, and DCGI asked me to accompany, so we had to tell them till midnight what is the drug development process from phase 1, 2, 3, 4. He understood everything. डिनर के बाद में, फिर उसके बाद में कहते हैं फिर ये एडवर्स इफेक्ट क्यों हुआ, ये ऐसे डेथ क्यों हुई, जब इतना सब टेस्टिंग करते हैं रिगरस. Then I tell him that every time we say reasonable uncertainty, because we don't have variability in the clinical trial, but only certainty in reasonable uncertainty is uncertainty. So he said, Acha. And the movement was he sanctioned 1,000 crore for medicine safety. So that is what we sometimes say, adversary converting into opportunity this is how we need to learn communication skill. So, and since then, the government is asking, अब तो खैर कम हो गया, so what? या इतना पैसा दे दिया, इतने centers खोल दिए, so what? So we have to come out with artificial intelligence, not only what is the impact of the data which is coming up, signal generation, but what is the impact on saving life, saving admission, and converting this into tangible right or tangible return on investment. Government ने जितना investment किया तो उसको health care system में हमको इंडस्ट्री का तो ठीक है, इंडस्ट्री ने अपना रिटर्न ऑफ इन्वेस्टमेंट बता दिया, सोशल टर्म में, कंट्री के टर्म में रिटर्न ऑफ इन्वेस्टमेंट क्या हो रहा है, कितनी हमने हॉस्पिटल एडमिशन बताया. अभी भी अगर आप देखो 90% स्लाइड्स लोग दिखाते हैं क्या, एडवर्स ड्रग रिएक्शन से यूएस में इतने ह्यूमन लाइफ होती हैं, डेथ होती हैं, जितने की रोड एक्सीडेंट से ज़्यादा होती हैं. कैंसर की इंडिया का डेटा है ही नहीं अभी भी. We have no idea how many hospitalization, prolongation of hospitalization or is taking place because of this. So I think if this can also be integrated, particularly medical superintendent and you and all those, Sandeep is there, तो ये अगर हम convert करें, then I think policy makers will get more satisfied कि यार हमने इतना पैसा लगाया तो इसका इतना हमें फायदा हो रहा है. But I think tremendous, tomorrow is age for AI in pharmacovigilance. Congratulations to all three.
So, thank you speakers, thank you sir. And we wind up this symposium with the hope that in next 10 years we are going to see a lot of AI in this.
I want to put some industry perspective. So, you're mentioning about Oracle Argus Safety. You must remember that this was born in this city. It was called Relsys earlier. And then it was taken over by Oracle. And we have been using Oracle for the last 15 years. And industry has been using it for the last 25 years or so. What you call traditional is Jurassic for us. What we call traditional is manual processing. Now, a lot of it has changed from manual processing to what automation. There's a difference between automation and AI. Or automation is, for example, auto case narratives. But now, entire case processing is being done by AI. There are reduction, or better use of manpower. Other thing is there's a step called triaging of cases, where there is, you actually see whether, he was mentioning about the identifiable patient and identifiable report, the 4 minimum essential criteria. There's a 5th one also called batch number. So all that, we use at least 10 people. We go for 145 countries, for example, and we get about 100,000 cases a month. Now all that, and very little, as everybody has discussed, very little from India. All that triaging happened with at least 15 people. Now we are doing with 2 people. Example. Or literature cases. There is something called literature cases. Now every week the database scans 1 lakh cases, 1 lakh literature, out of which it comes about 15 cases. Now for this I am deploying a people of 25 people. Now one person does it. Because he doesn't even do anything. He actually reviews it and passes it to the case processing team. So that is a change that will happen. Why I'm telling all of this is because there has to be an academic industry collaboration. If that is there, a lot of knowledge transfer can happen, and it can be better utilized for enhancing the meeting which he was talking about, 2010. I happened to be a very junior person in the industry, and I was in that meeting, fortunately. And from there, we also started learning. Thing is, we are all running in parallel tracks. We don't know each other's world. We are on the same coin on the opposite side. So this is very necessary that they feed from each other. And to your further surprise, the two major databases, not just Argus, there's also RSG, LifeSphere. It is also born in India, in Bangalore, in Mysore. The third one is born in Chennai. And these have been taken over by multinationals. And as you're saying, हमसे data लिया, हमको ही बेच रहे. So it's very essential that this flavor needs to be put in any of this. And my role has been with sir is that we are trying to put in some industry flavor into academic conference. It should not be just the flavor. It should be a combined conference. We have not, हम आसमान से नहीं टपके, हम भी इनसे सीख के वहाँ गए. तो and we are trying to get academic people into the industry conference. The discussions that you will see is totally different. Then we realize where we can. Indian government has the infrastructure. It has 1,000 centers. Now, and us also in the industry, we practice like an isolated silos. मैं अपना केस प्रोसेसिंग कर रही हूँ, फाइजर अपना कर रहा है, एंड यूएमसी के पास कुछ नहीं जा रहा, सब एफडीए के पास जा रहा है, नहीं तो ईएमए के पास जा रहा है. And but to credit to FDA, EMA, they have come up with their guidelines. Indian pharmacovigilance guidelines also needs to upgrade to include the AI modules in the pharmacovigilance. Thank you. I'm sorry I took more time.
Thank you. Thank you.
Now I request our chairs to please grace the dais and felicitate our speakers. Mr. Tarun Gupta. Dr. Sangeeta Yadav. And Dr. Sandeep Kaushal. Now I request Dr. Manish, please felicitate our chairs. Dr. Puneet Dhamija. and Dr. Narendra Kumar. Yeah, all the speakers can please come up on the dais. With this, we bring Symposia 17, Transforming Therapeutics and Medication Safety in India Using Artificial Intelligence and Digital Analytics to a formal close. On the behalf of the organizing committee, we express our deep appreciation to our respected chairs, Dr. Puneet Dhamija and Dr. Narendra Kumar, our distinguished speakers for the exceptional scholarly contributions, and all the members of the audience for their gracious and attentive participation. We trust the deliberations of this session has enriched our collective understanding and inspired further commitment to challenges and considerations in preclinical pharmacology. next person, Govind. Good afternoon, everyone. To all the distinguished delegates, esteemed faculty members, respected chairs, honored guests, and dear researchers. It's my privilege to formally commence Symposia 19 entitled Advancements in Nutraceuticals and Herbal Therapeutic Technologies. This session highlights the remarkable scientific progress happening at the crossroad of herbal medicine, nutraceutical innovations, and modern therapeutic science. Today we have the honor of hearing from eminent experts whose contributions are shaping the future of chronic disease management across the world. This session is graciously chaired by Dr. P.V. Devan and Dr. Maninder Karan Vishisht. We extend our sincere respect and gratitude to them for leading this important segment of today's scientific program. Now I will briefly introduce both of our esteemed chairs, Dr. P.V. Devan and Dr. Maninder Karan Vishisht. We are honored to have Dr. P.V. Devan as distinguished pharmacologist and founder director of NIPER with over 300 plus publications, 17 patents, and mentorship of 36 PhD scholars. He is a leading figure in pharmacology, a fellow of Royal Society of Chemistry and the first Indian Chartered Scientist. Dr. Devan is also a recipient of the IPS Distinguished Pharmacology Award 2023, and he currently serves as the Research Director of Central Research Laboratory, Belgaum. We are privileged to have him as the chair for the sessions. We are also honored to have Dr. Maninder Karan Vishisht, Professor of Pharmacognosy at UIPS Punjab University. She brings 28 years of expertise in natural product isolations, phytopharmaceuticals, bioactive molecular research. She has guided numerous theses, filed international patents, discovered novel molecules, and authored over 130 publications. We are privileged to have her as a chair for this session. Now I request Dr. P.V. Devan to introduce our first speaker.
No, ठीक है. Good evening, everyone. Our first speaker is Dr. Madhusudanan Saraf, a distinguished pharmacology academic and institutional leader with over 4 decades of service to pharmaceutical sciences. He led Bombay College of Pharmacy as principal for more than 2 decades and later contributed to NMIMS and HA College of Pharmacy. With 90 plus publications, 22 PhD scholars, and major funded projects, he has significantly strengthened academia-industry collaboration. He is honored with awards including APTI Principal of the Year and Professor G.P. Srivastava Lifetime Achievement Award. Now I warmly invite Dr. M.N. Saraf to share his views on reverse pharmacology, transdisciplinary approach to rationalize herbal therapy. Welcome, Dr. Saraf.
Honorable Chairperson and Co-Chairperson of the scientific session, and distinguished members. No problem, I will finish in 10 minutes. At the outset, I would like to thank the organizers for giving me this opportunity to share some of my views on the topic of reverse pharmacology in regards to the herbal drug development discovery. Recently, all of you have seen what is happening in US and the overall sea change in terms of tariff, and pharma is also not untouched. And generics, our strong base, is really important, but over the years, the problem is going to be compounded when it comes to new drugs. In terms of self-reliance, what the government has been now talking about, there is a need to restructure our concept of drug discovery. Traditional approach of drug discovery, we all know, is an extremely difficult task, time-consuming, and economically not viable for a country like India, by and large. And our strength lies in our own systems. Unfortunately, the scientific base is lacking, and because of that, acceptance is poor. So although some well-established medications are there, we are not in really position to scientifically accept it. Therefore, the time has come of understanding a differential approach in this herbal drug discovery, development, and standardization concepts. I would like to share with you a few of my observations in this. May I have the next slide? Somebody is operating, or shall I do it myself? Thank you. Reverse pharmacology in no way new concept. It's well established way back in 1930s when first of all, there are some students, I'm sure that understanding is important, from the bench to the clinics. That has been our traditional approach of drug development discovery. But from the level of human just proving that the disease process to again the clinical side, that's the reverse pathways of drug discovery, is what is reverse pharmacology techniques. May I have the next slide, please? What basically is important in this process is documentation of clinical evidences and clubbed with the scientific basis of proving to bring it to the level of utilization. Now, as I was referring to, reverse pharmacology is no way a new concept. Sir Ramnath Chopra and Gananath Sen, they laid the foundations to reverse pharmacology. And what a wonderful work. The work pertaining to the reserpine, that is derived from Rauwolfia serpentina, a molecule of indigenous origin, what we should be proud of. That is the unique. A lot of good clinical work was done by Gananath Sen, and extremely proving not only the activity, but more particularly adverse effects. We know that Rauwolfia serpentina, reserpine, its utilization in the initial period as an antihypertensive drug. Of course, it was withdrawn later, but proved to be one of the important concept of the drug as far as at that period of time for hypertension. More particularly, the adverse effects like depression, gynecomastia, the characteristic gastric symptoms like peptic ulcerations, and Parkinson's-like manifestations. These were also very critically noted, and in fact, that led to the foundation of new drugs in all these series of depression, Parkinsonism, which has led to the concept of new drug discovery in real sense. I'm talking about the era 1930s. This is somewhere around 1930-31 that this demonstration was there by these people. And what basically is the concept of already proven drug, but on the scientific front, clinical evidences to bring it to the bench, from the patient to again back to the patient, the reverse pharmacology. Next slide, please. So many medicinal plants are described, and by and large, the concept of testing goes on hydroalcoholic extraction, purification, testing randomly in the country. Unfortunately, still there is no proper base in terms of what are they doing. Already known drug, again extracted, again activity, whole in the world, how many times this is happening, God knows. Next slide, please. So there has to be a base for whatever that has been done. Next slide, please. So I would like to take a few examples for the want of time as to how some basic things are happening. Sarcostemma brevistigma is a plant of Asclepiaceae family. Traditional name Rancher, Sher, Som, so many of these names. But interesting finding is that it was used by a Vaidya from Solapur region, Mr. Vaidya Bagewadiker, for the treatment of asthma. His observation was that when he used to give fresh juice of this plant, it gives the activity, very good activity. So essentially, he was scientifically driven mindset. He approached Hafkin Institute, and he told to the scientists over there that, look, this is what is there. Can something be done? It was taken up for further studies. Now that when it was progressed further, the conventional literature searches revealed no mention of any asthmatic activity per se. But conventional, I mean, this was the medical observation. But in the literature, traditional literature, Ayurvedic literature, no mention of that. Next slide, please. So, basic, again, approach of purification was followed in terms of most of the important constituents like alkaloid, purification, and fractionation led to one important constituent, fraction 2. Next slide, please. So now, based on the conventional methods of experimental pharmacology for anti-allergic activity, it was found to have a profound activity in chemically induced mediator release activity as anti-allergic, like in the compound 48/80, as well as in the active paw anaphylactic models, it proved to be anti-allergic agent. Further, on the respiratory resistance model of Conzett and Rossler, it showed a profound activity in a dose-dependent manner. Next slide, please. So this established is in vitro as well as in vivo activity. And conventionally, a drug like isoprenaline, like compounds, which are very good anti-asthmatic agent, but cannot be used because of profound effect on the heart. So we explored the effects and found that in the isolated guinea pig model, it does not have any effect, so a positive point again. But unfortunately, it did not have a very unique activity. It is on a non-specific smooth muscle relaxant activity, as could be seen, so a spasmolytic compound. So essentially, if looked into this perspective, from the reverse pharmacological perspective, there has been clinical evidence, there has been experimental evidence. The question is, has it been moved to the level where it can be used in the clinical practice? There is a question mark. So most of the work is held up for the want of proper clinical research in the clinical trials. If we see in the perspective of that it has gone through these clinical trials, is another molecule that is derived from Coleus forscola, from Labiati family, where we know that is the forskolin which has been derived from it. Excellent work in the Hex Research Center. Now it is no more. In the Mumbai, a few years back, in the Mulund area, a very important research center. This culminated into the active research in 10 years to derive this compound forskolin. Next slide, please. There are some good things that although compound was discovered from reverse pharmacological approach, the utility was limited because of more adverse reactions. And sometimes the old molecule having some unique activity, now that is what is the state of affairs now with respect to forskolin because of its unique activity of increase in the cyclic AMP concentration. Next slide, please. Now I come to a little differential approach in the phytopharmacology or reverse pharmacology, that it's not the drug discovery alone, but advancement of science as such is possible through this route. And very good example is that of Ami Visnaga, in which the common name is Khela. And next slide, please. It has the compounds like chromones, and disodium chromoglycate isolation was unique discovery. Why? Because there was no concept in pharmacology of a compound which has the ability to inhibit the release of the mediators. It was possible with the discovery of this compound. So essentially from the natural products through reverse pharmacology route, there has been a new approach in the understanding of the disease condition in a proper way. It is just not that blocking the beta receptors or phosphodiesterase inhibitor activity or some such models that only work for betterment of asthmatic activity, but more importantly, very inhibition and prophylactic approach could be possible to be known after the discovery of the compound like DSCG, and again through reverse pharmacology route. Next slide, please. Again, another compound I would like to say about this evolvulus alcinoides, which is Shankha pushpi. By and large, anybody can talk about it. Oh, that is for improving the memory, brain tonic. Everybody knows about it. But there was one article in the Lok Satta, Marathi Daily, which said that a lot of things are there, but it is good for asthma. And I had an opportunity to work on that, and we found that, next slide please, that again on this reverse pharmacological concept, this has basically anti-asthmatic activity. For the short of time, I have not given the details. Butea monosperma, again next slide please, has certain components which again has interesting such mechanisms of inhibition of the mediator release and the beta-2 stimulant activities, as could be seen from isolated tracheal chain preparations. Next slide, please. And the anti-anaphylactic activity of this compound. Next slide, please. Now I come to a last point, and that is about what to do. Isolation, purification, utilization, big question mark. Still not resolved. Both are good at their own ways, but then if you see in the perspective of reverse pharmacology, in perspective of Ayurvedic literature, the total constituent works much better. And if that philosophy is adopted, many of our problems are solved. Adhatoda vasica has two constituents, important constituents. May I have the next slide, please? Vasicine and vasicinone. And they have distinct and different activities. One is bronchoconstrictor and one is bronchodilator in the same plant. So basically, when you take in the total, in its form, rather than an isolated form, it has its own advantage. Next slide, please. So, I am coming to the end of this. The basic point is that oxidized product of viscosine produces dose-related bronchodilator activity and anti-anaphylactic activity. So, essentially, this has been possible from the scientific evidences as well as clinical evidences through the concept of reverse pharmacology. Next slide, please. So, to sum up, creation of the databases in a proper, organized scientific study to determine the mechanism of action, study of the impact of pharmacokinetics and pharmacodynamic factors in vivo, and evolving collaborative research and development network, quality, safety, efficacy of the plant product and active principle if we follow. I think it will have a tremendous impact in terms of this. I strongly believe that standardization concept doesn't work. You take some reference standard, you do it. In a long term, we want some proper evidences which has a base of strong science, Ayurveda, plus scientific basis. Ladies and gentlemen, there is an opportunity for us in this age to evolve some scientific proper base where we do have a great wealth, natural wealth, and that has to be properly exploited. Industry has to come forward along with the academics and take it to the level which will have clinical acceptance. Thank you very much for patiently listening. Yes. Yeah. Yes, a good question. Basically, we all know that medicinal plants is conglomeration of several metabolic products. And essentially, they are in a, we take when it is consumed in totality. We are not isolating anything. So when your question is how it works in its own way. When it works, when you take it, there is a balance that has been established. Essentially, if we see in the perspective of a disease condition, there is a hyperactivity of one of the things. Like if you talk about asthma, it is a more, dominance of cholinergic activity vis-a-vis adrenergic activity. And there is a need for balancing that. And precisely that is what it happens when you take it in total. So basically, it doesn't differentiate at the level when you take it inside, in vivo. And therefore, that is what is the importance of taking it in total, rather than isolation. So essentially, how it works is basically, I believe,
Thank you, Dr. Saraf. Our next speaker is Professor Sanjay Jajak, professor and former, he is not former, he is still head of natural products at NIPER SCS Nagar. He is recognized nationally for contributions to phytopharmaceuticals, ethnopharmacology, and natural product drug discovery. With over 30 years of experience, he has over 25 publications, 5 patents, and an H index of 41, along with major funded projects from CSIR, DBT, and Ayush. His work includes value-added herbal products like immunobooster herbal tea, and he has trained numerous PhD and master scholars. I welcome Professor Sanjay Jajak to enlighten us on nutraceuticals and phytopharmaceuticals, a paradigm shift in herbal drug technology.
Okay, very good afternoon to all of you. First of all, I like to thank Chair and Co-Chair, Professor Diwan and Professor Maninder Karan ma'am, as well as Professor Anil, very active HOD and Chairman of this department, to invite me here. So today I thought to speak on, due to shortage of time, I will try my best to tell you what I would like to say about these two translational research opportunities that we have when we work with natural products. And of course, the first speaker, Professor Saraf, mentioned reverse pharmacology also has a big role to play when we talk about the product development. So how it goes? So just this slide I have just shown as I think we listened to the approach of combination approach. Hello, am I audible? I think, yeah, so a combination approach, and this is reductionist approach, that we reach to NCE and find a new drug molecule from any natural product source, and that you see historically drug discovery from natural products has always been successful. As this slide says, all small molecule drugs that have come from natural products, 67% are actually of natural product origin, and 50% of all drugs definitely are coming from natural products. That tells us the drug discovery from natural products could be very rewarding, so we should be doing that. And I will not talk much about this slide. One example was already mentioned about Raulfia. These are the leads over the last 6 decades that have come from Indian medicinal plants. That shows the value of Indian medicinal plants. So with that, this is the research areas in my lab that we have. So basically today I'm just going to talk about the nutraceutical and phytopharmaceutical, but other areas do matter when we work in natural products, especially the new drug discovery for different therapeutic areas, and the very important aspect of quality raw materials, standardization of herbal drugs and polyherbal formulation. So this I will skip actually. This is the work we did with IMTech, find 2 preclinical candidates of microbial origin that actually proved very effective flux pump inhibitors, especially in MRSA, methicillin resistant strain. So that I will not go towards this escape pathogen. These are actually having the tendency of escaping the biocidal and the cell, you can say microbial cell, and they are ineffective. So on that aspect we have worked. But let us say nutraceutical, this is today's topic. We know the nutraceutical has become a very important segment now of the natural product research. Many pharma companies are actually into nutraceuticals. So this is the term introduced way long back in 1989, and we continue to work in this, and there is a paradigm shift, I think, from cure to prevention, and that is what Ayurveda also relies on. Prevention is better than cure, and on that system, it's a holistic system where we use the traditional medicine formulas that have been described in our traditional medicine texts like Rig Veda, Atharva Veda, Charaka Samhita, and Sushruta Samhita. So with this, very important is that the age of individual and the season of consumption. You get the best health benefit when you consume a particular food source or a medicinal food plant when it is consumed with the right season. So this is very important as the Ayurvedic concept, and the linkage of Ayurahar and nutraceutical. very much, I think we have the guidelines by FSSAI on emphasizing the linkage between the Ayurahar and nutraceutical product development. So we know that ROS has a very important role in many diseases, cardiovascular, lifestyle diseases especially. And you can see all these diseases, their origin somewhere you find in this reactive oxygen species. So to find antioxidant activities is very important in the medicinal food plants and their extracts. So this is the approach. In a nutshell, you have to first recognize or identify what is your therapeutic segment and also the food matrix. And from that, you can go towards the identification of bioactive components, their preclinical, clinical development, and all that should start with the market potential first, and then define the food matrix and the therapeutic segment, and go ahead with developing, follow this step, a nutraceutical product. And the guidelines are relatively quite simple if you compare with the conventional pharmaceutical product regulatory guidelines. The regulatory space here is not that stringent. You can get the license very easily from FSSAI, that is the agency, and we have this regulation 2016-2021, where we can make use of these, and you can prepare as a pharmaceutical scientist. All of us, we can very well manufacture a phytopharmaceutical product in these different dosage forms. I will not read out, but very important, this Schedule 4 of this Act enlists 200 medicinal food plants. So if you select a plant from this and then mix with other nutrients, you can very well form a new formulas under this nutraceutical, and there is a lot of demand, and you can easily actually formulate such products and bring into the market, as that is need of an hour that we should do. But today I want to more give emphasis on phytopharmaceutical. Now it is lot of discussion is going on since last 10 years, since the regulation was notified, but still we haven't come up with any product as on date. There are a lot of challenges, but let me tell you, now several products from CSIR with some industry linkages, they are actually developing the phytopharmaceutical, which will definitely see day of the light. So what is important here? As Dr. was saying, no, that extraction, purification, yes, all this is required. And here, what is required in best natural product chemistry lab, that we should learn how to prepare enriched fractions. Like you say, polyphenol enriched fraction, flavonoid enriched fraction, alkaloid enriched fraction, terpenoid enriched fraction, and so on. Because these are the active secondary metabolites that are present in several medicinal food plants as well as the medicinal plants, and we need to target that, because this regulation says you have to prepare an enriched fraction where you have to show the bioactivity of that fraction correlate to 4 biomarkers, which can be qualitatively and quantitatively estimated. And if you do that, definitely you are in the driver's seat to push this product further. As here we say, active phytopharmaceutical ingredient, APPI, or phytopharmaceutical ingredient, PPI. So that is the term we use when we use API, that is active pharmaceutical ingredient. That way, this is the regulation, and you see these are the comparison. You can read also in the articles, but what is important, last, you see entry here, last row, not developed yet. That is what I mentioned, but several are under clinical trial. There are a lot of challenges when we say this, but you see these are some examples. In COVID-19, I remember in August 2020, DCGI gave express approval for this first one, Cocculus hirsutus, called as Patal Garudi, to treat mild COVID-19 cases. This is a phytopharmaceutical from this plant, but later on they withdrawn it, but at least that showed us the path, how we can proceed. And you see now, for stroke and neuroprotection, we have ashwagandha. Type 2 diabetes, Agel marmelos, that is Bell, and then asthma, Boswellia serrata, and combination of Agel marmelos. So these are 4 under advanced, you can say, stage of approval from DCGI. And you see these are the things what we require. Now, as we see the today's status, in IP, Indian Pharmacopoeia 2022, 7 phytopharmaceutical ingredient monographs are already there. In 24, they will include 3 new. They actually constitute licorice, then Adatoda vesica, the example you have seen, and Gingiber officinale, as well as the other more examples are being added, and this regulation is in line with the regulation in USA and China, Europe as well. So we definitely have a very good regulation to push this phytopharmaceutical product development from a single medicinal plant, and that is the beauty of that, where we can reduce the complexity and go ahead with that. But having said that, we have a lot of challenges in the path. Like here, batch to batch variation, and you know that when we talk about even fraction, there could be variation of phytoconstituent profile from season to season, even age of the plant, even one season, April, let's say, to November, the phytochemical composition will be same, but their content composition will vary. And that is real challenge, and if that varies, your bioactivity is going to vary. So if you actually find the balance here, what is the range, limits, that is what I think you can see now. New guideline have just few days back, they were published on Pharmacopoeial Commission of India website. You check that on this phytopharmaceutical that I'm speaking, and here lies a lot of opportunities. With that, I show this example. Yeah, so I will. The work what we started 10 years back, we had several leads in our lab. When we talk about anti-obesity and anti-diabetic natural products with licorice and Muraya konegi, these were the best leads, so we took that lead further to work on this curry leaf that you know it's very commonly available throughout India. It can be easily cultivated, so sourcing of raw material is not a problem here. That's why we thought of developing this, and it is also listed under FSSAI medicinal food plant list. So from that we thought of working on this. We compiled the literature and published in the form of review, as well as we prepared alkaloid enriched fraction from the curry leaf original methanol extract that contains almost 100 compounds. From that we have come down to around 20, and you will see these 4 as per the regulation phytopharmaceutical. You need 4 markers. These are the markers, and we know these are all compounds of closely related structures. When we talk about the phytoconstituents here, 22 carbazole alkaloids are there in curry leaves. Out of that, we have identified by LCMS, and these 4, like mahanimbin, girinimbin, coinimbin, conigicine, you see very thin structural difference among these compounds, but these are the bioactive compounds present there. So we have done their quantitative analysis, and you can see the enrichment that our 2 students are sitting here. They did a lot of work. Akash, Kirti, Mrudula, they actually spent last 5 years. We are doing work on this plant, and now we reached to a stage where we can license this product to industry. So you can see the left-hand side, there are several peaks here, so which you are not seeing. So this is the original extract. More than 100 peaks could be there, but we have identified around 11 to 12, and that is enriched. You can see on the right-hand side, and this is the enrichment, what we have in our fraction, and that is what you need to do. This enrichment is really required when you go for the product development, because you have to have a very good quality standard and maintain the quality of your fraction, also batch to batch variation. So we published this and also suggested what is the best season for collection of this curry leaf, where you will have optimum content of these carbazole alkaloids. And this HPTLC chromatogram shows here that this is the original extract, and these are the 4 trials, and we have done several trials, even from 100 gram to 1 kg scale in our lab, and we are now doing it on pilot scale studies, and we see consistent composition. All these red color bands are chlorophylls. In also HPLC chromatogram, they were small at the base peak, but here we have seen that they are there on the HPTLC, and what we see, these are the consistent result of the alkaloid content in this fraction. And we have already filed Indian patent, waiting for final grant, so that will be useful when we talk to industry. Already we are talking to industry, and these are the in vitro studies in the adipocytes that we have 3T3L1. So just it will take another 5 minutes and I will finish. So please allow me that. So this is what I mean to say. Dr. Saab also said the combination is better. You can see individual compounds are less active than the fraction. So it is better to push the work with fraction. That is the AEF that we are talking about. And this is the assay that we followed, 13-week study protocol, where we have fed the animals with first high-fat diet, And you see the 25 gram or 20 to 22 gram mouse become bulgy, getting 36 to 38 gram weight. And then we have continuously gave the treatment with all these our treatment groups, including the alkaloid enriched fraction 100 mg per kg per oral, as well as 50 mg per kg per oral treatment. And also we have observed the body weight, what is the change, whether it is decreased or increased. So we found as compared to disease control, there is a decrease in body weight, and the 36 gram mouse became 22 gram after the end of 13-week study. And we have also seen all the anthropometric parameters, as well as we also conducted, as per OECD, 407 28 days repeat dose acute toxicity study. And what we find, this fraction is safe up to 1 gram per kg per oral. Our effective dose is 100 mg per kg per oral. So that gives us the preclinical safety indication. And this is what you see here. As compared to disease control, this is the actually the fat what you see, and that has been totally you can say reduced in this 50 as well as 100. We hardly see that. So this is where you see even liver you can see as compared to normal control and AEF. It has again the almost same size. So that is what we can see. This treatment is definitely reducing the weight and also all the biochemical parameters that I am showing here. So you can see the total triglyceride, cholesterol, low density lipoproteins were decreased, but what we saw at HDL is increased. And this is what very exciting observation we had, which I haven't seen any drug in the market for obesity that increases HDL. So that is why industries got attracted. I can tell you, Ashok is here. So this is what we presented to you also, some of this data, you know that. So that is the thing. And you see this histopathology, you can see this fraction at 100 mg per kg showed the closest resemblance to normal control, NC versus AF 100. You can see it is coming to normalcy as compared to the disease control. So this also tells us the safety profile. This is the mechanism that we have given, that all these parameters, including free fatty acid synthase, PPR gamma, even inflammation, NF-kappa B, then SR, EBP, 1C, as well as leptin resistin. So you see that all these in that as well as in liver, all these parameters were down-regulated. We have done the gene expression studies and given this mechanistic approach for the anti-obesity effect of alkaloid enriched fraction. This, part of this we have published in Journal of Pharmacology. This is what the stage that we are having. This is, we think now, as after talking to industry, we already have the technology readiness level between 4 and 5. That is what industry asks, what is your TRL? As academician, having all these facilities, we can distill here. Next, all industry has to come forward and take this innovation further. And we are right now doing already clinical study. And the results are quite positive, which I'm not going to tell, but this is what the clinical study is going on. After that, I think we'll be very much sure that it is working as we saw in preclinical in animal model. With this, I thank you all, and last slide. So we are organizing, like we see many conferences happening. We are also organizing one at Naipur Mohali with Society for Ethnopharmacology from 26 to 28 February 2026. So all of you are invited to attend this conference, and we look forward to meet you at Naipur Mohali. Not far from here, 10, 11 kilometers from PU, Chandigarh. We are located near Mohali Cricket Stadium. Naipur is Mohali. So thank you all. If you have any questions, I'll be happy to answer. So this is my research group, PhD students who are sitting here, and many more. They are doing very good work right now in my lab. So thank you so much. This is our natural product field laboratory at Naipur Mohali. So thank you so much, and I'll be happy to take any questions. Thank you. So I had to go very fast, but excuse me if there will be any questions due to shortage of time. Anybody, students? Yeah, please. Yes. Yes. Yeah, that's good question. All you people should see that you have to use different resins where we can selectively separate the interfering compounds. Like I'm talking about chlorophylls. There are a lot of, when we deal with leaf drugs, so we have to find a protocol, like we have used HP20 diene resin. So where we can very easily separate the chlorophyll from our compounds of interest. So there are lot of resins, that these are polymeric resins which are available either on, you can say, the exchange property, that is anion exchange or cation exchange. Based on that, so cation exchange resins sometimes work better for alkaloids. Even for polyphenols, we go for the anion exchange resins, and several other resins are available where we can do this enrichment. Or even by the precipitation methods also, there using specific reagent, we can precipitate out tannins, and these are another interfering compounds in plant extract that we need to separate. So can be done, and you need to see different publications that come in Phytochemistry, Journal of Natural Products, where many such protocols are published from time to time. So also look at that, and this is the way we do this actually, from original extract, the enrichment fraction, preparation of enrichment fractions. Okay. Yeah. Yeah. Exactly. Yeah. Yeah, actually, charcoal, no? Charcoal is, you should only try at the last resort, because that will adsorb your compound of interest. Please don't go for that. That's why I said resin. Resin and precipitation, other techniques, we can very well do this preparation of enriched fractions. So that is what I think the resin. So try HP20 diene resin. If you have, you can do it very well. Yes. Yeah, please. Yeah. Yeah, actually, yes, because you know that I mentioned the reductionist approach. So there the idea is to find a new chemical entity with bioactivity. But I tell you, I mean, 2 out of 10 times you may be successful. 8 out of 10 times you will not, because there is always, I was talking to Diwan sir, that there is definitely a concept of synergism. You know, in medicinal plant, 1 plus 1 is not 2, but 3. And that is, we have to prove. And it is there, I think, in many examples are there in literature like this, where we have to see that, even in our case, I have shown AEF is more active than the individual compounds. So this is where I think synergism, but we have to still prove it, that it works. But first thing, you show the efficacy as a combination, but it is a simple extract, simplified extract compared to the original methanol extract, which has more than 100 compounds. We have actually made it to 20. And then it becomes easy to work with that. So that is the idea actually. Yeah, thank you. Yeah.
Our next speaker is Dr. Govind Singh, a neuropharmacology researcher with academic credentials from GJU and AIIMS New Delhi, and also from KGMU. His work focuses on stroke, Parkinson's disease, Alzheimer's disease, traumatic brain injury, drug repurposing, and herbal therapeutics. He developed an in vitro stroke model using oxygen glucose deprivation validated through in vivo Macau studies, and has guided over 40 postgraduate scholars while leading DST-sponsored projects. I invite Dr. Govind Singh to share his experiences in unraveling the neurotherapeutic potential of unexplored Himalayan folk medicinal plants. Welcome, Dr. Govind.
Thank you, madam. Respected chairpersons and delegates, good evening to all. First of all, I would like to convey my sincere thanks to Professor Anil Kumar, the organizing secretary of this event, for providing me this opportunity to share findings of our lab. And at the same time, I like to acknowledge the sincere efforts of my PhD scholar, Mr. Karan Vadhwa, to conduct this study, and the PPT also. The basic concept behind this is we selected an Himalayan medicinal plant that was basically used as a folk medicine for different neuroprotective potentials. We selected that plant on the basis of different other properties also. And then we explored that plant in different models of cerebral stroke. One model of cerebral stroke is already well established, that is middle cerebral artery occlusion model in rats. We also tried to develop different model in zebrafish that is more mimicking the pathophysiology of human. So I'm showing these all. This is the basic introduction. The cerebral stroke is of two types. When the basic concept is reduced blood supply to the brain. If this blood supply is reduced due to the blockage, then it is ischemic stroke. If it is reduced due to the rupture of the artery, then it is hemorrhagic stroke. In most cases, more than the 85% of cases, it happens the ischemic stroke. This is the basic epidemiology. Is the second largest cause of death worldwide and major cause of morbidity. This is the basic pathophysiology. Just I'm going fast. When blood supply is reduced, there is basically energy failure and excitotoxicity and different cascades of oxidative stress, mitochondrial damage, inflammation, and finally cell death. Basically, what happens when blood supply is reduced? The cells at an instant they die, but the surrounding area they are under stress. At this time, all the drugs available are thrombolytics, which dissolve the thrombus and increase the blood supply to the brain. This increase in blood supply further cause oxidative stress and further cause damage or death of the cells, and this core region going to increase, and increase of the core region further severity of the disease. These are the limitations. Why we selected folk medicines? As I already told, there are so many advantages. Already explored so many medicinal plants. Now, zebrafish. It's our sincere efforts of different scholars, because this model was not previously reported. We first time developed this model of zebrafish in adult zebrafish. In larva, it was available. The beauty of this model, in MCA model, we create cerebral stroke mechanically by inducing suture through the middle cerebral artery to the brain. And through this procedure, another trauma we are causing to the rat. In zebrafish, we developed this model with chemical, and it's creating the similar pathophysiology as to the human. These are different advantages. Now we are coming to the how we selected. In zebrafish, hypoxia model was available. This was the mortality in hypoxia model, and this is the mortality with our chemical. This is the ponatinib, different concentration of ponatinib. And TTC is the dye which shows the infarction area in the brain. When we measure the TTC infarction area, see, this concentration is causing similar infarction as compared to hypoxia. And different behavioral changes are very similar to the hypoxic model. This is the main determination of the concentration of our plant. RI is the research investigation plant. I'm hiding this name because of confidentiality. We are going to do something further. So from these cytotoxicity studies, we determined the exact dose of RI to evaluate its neuroprotective potential. So this is the lethal concentration. We found it, and after decreasing 10 times, we selected 3 doses. And the cytotoxicity, 25.5 something, it was the LC50. So our doses was 2.5, 1.25, and further half of that dose. And this is the cytotoxicity and the absorbance of the infarction. When we observed different oxidative stress parameters, we see the restoration of all the parameters of oxidative stress because due to our treatment at day 1 and at day 7. At both days, all these oxidative stress parameters are restored. Similarly, the enzyme activity of different mitochondrial complexes, all parameters, we can see clearly that our drug restored these parameters in comparison to control. And we can see also the parameters are very comparable to the control at day 7. Cytokines, our drug also restored all the cytokine parameters at day 1 and day 7. And different neurotransmitters, all the neurotransmitters, dopamine, serotonin, acetylcholine, noradrenaline, glutamate, GABA, all neurotransmitters are restored. And see the beauty that drug did not reduce GABA, it maintained the balance of GABA and glutamate because balance is required. And all these findings further reflected in the behavioral changes, behavioral parameters. This is the control fish. This is the track of the fish that it moved. The control fish. This is the ischemic fish. And when we administered with the different concentrations of our experimental drug, see the fish is moving like the control. This is scototoxic behavior, light and dark chambers. Now, control fish, when ischemic fish is just moving in the dark chamber. With the increase in the concentration, the fish is just moving like the normal fish. This is the locomotor behavior. See, again, the highest concentration of our plant made the fish to the very normal, and the fish is exploring the central zone when ischemic fish didn't come to the center. Then the same study was revalidated in the MCA model because MCA model was well established and has been used since long. And we can see that infarction is very much reduced at this dose at day 7. This is the blood-brain barrier permeability, and we can see the permeability is again maintained with this dose at day 7, even at this dose also. So the drug is very much effective in reducing the infarction, maintaining the integrity of the blood-brain barrier permeability. Again, the oxidative stress parameters in rats, all the oxidative stress parameters are restored. Enzyme activity, just like in zebrafish, complex 1, 2, 4, and 5, all parameters were restored with the administration of our drug. Inflammatory cytokines further restored with the drug. Neurotransmitter levels, all the neurotransmitters, GABA, glutamate, all neurotransmitters were restored at day 1 and day 7. And at day 7, all parameters are very comparable to the control. mRNA expression, we can see the administration of our drug maintained all conditions of expressions. Further, behavioral changes, we can see all the observations after administration are very close to the control group. Learning, memory, and even we can see all this in trackings. In last track, we can see the movement of the rat is very similar to the control. And this is anxiety-like behavior in plasmids. We can see at this dose, the rat is moving like the control. In this, the rat didn't come in the open arm, but with the increase of the dose, the rat is moving like the control. This is the novel maze spatial recognition test, non-spatial recognition test. The familiar object, this is the novel object, and we can see this rat is just moving like the control. This is a Morris-Water maze test for spatial memory. In this also, with the administration of highest dose, the rat is showing the memory just comparable to the control. Further, in histopathology also, we found the improvement in neuroprotection. Now, we made the formulation, nasal formulation in spray type, and this formulation showed another more valuable results here, or more restoration in different parameters, or more neuroprotection. And all these neuroprotection is further validated through all these images. I think need not to repeat. So we did one Himalayan plant, analyzed its neuroprotective potential in zebrafish model, and we developed that zebrafish with a chemical, then revalidated all these neuroprotective potential in the MCA model. Then we prepared a formulation, nano formulation for nasal delivery to bypass the blood-brain barrier and first pass metabolism also. And all these parameters are further restored, well restored with the formulation. Thank you. Any question, please? Nano formulation of our extract. Ji. Actually, cytotoxicity evaluation is just pending, but we reduced the dose as comparable to the dose that we were administering previously. The dose was adjusted. Size 182 nanometer. Actually, sir, we can see that if there will be toxicity of nanoparticles, a nano formulation, then further damage will be. 180 nanometer बता रहे हैं सर. 180 ना 180 nanometer. For better delivery to the brain, because we didn't check bioavailability, we observed the results only. With the formulation, because we have to bypass the blood-brain barrier for fast delivery and for bypassing the will not cross. That's why we are bypassing the blood-brain barrier. Yes, sir. It's a nasal formulation through. It's a nasal formulation through trigeminal nerve and olfactory nerve. It's going to the brain directly. Yes, sir. सर ये तो ऑलरेडी एस्टेब्लिश्ड चीजें हैं सर, नर्व से कैसे जाता है? क्या सर? ट्राइजेमिनल नर्व और ऑल्फैक्टरी नर्व के जो टाइट गैप जंक्शंस हैं उनके थ्रू प्री जंक्शंस उसके थ्रू डायरेक्ट ब्रेन में जाता है। ये ऑलरेडी एस्टेब्लिश्ड मेथड हैं। और उसके थ्रू हमने फाइंडिंग्स भी देखी हैं। जब रिजल्ट्स आ रहे हैं इम्प्रूव्ड, ये जितनी भी ट्रेसिंग्स हैं वो उससे बहुत ज़्यादा इम्प्रूव्ड हैं। क्योंकि सर नेजल म्यूकोसा में ही हम उसको ड्रग को एडमिनिस्टर कर रहे हैं विद द स्पेशल दैट एडमिनिस्ट्रेशन प्रोसीजर जो ऑलरेडी हाँ वो अभी करेंगे मैडम। जी फॉर विच जी the zebrafish model we developed only, it was not already available. A standard drug. Okay, that we can think of. Thank you.
Last. So our last speaker of the day is Dr. Ashok, Senior Manager, Ayush and Scientific Affairs from Himalaya Wellness Company, Bengaluru. Dr. Ashok is an Ayurvedic physician and he is PhD in Medicinal Plants Pharmacology from Gujarat Ayurveda University, Jamnagar. With a research experience of over 15 years, he has co-authored 2 books, 90 plus publications, and he is a co-investigator for as many as 15 projects. He is proficient in product designing, standardization and regulatory aspects of Ayurveda, classical and patent proprietary products, herbal personal care and veterinary nutraceuticals and dietary supplements. A recipient of many awards including Young Scientist Award in Drug Research, Star Award and Ayurveda 2002 by the Himalaya Wellness Company, and an work experience of a discovery scientist with a work profile of a 360-degree product innovation. We invite you, Dr. Akash. Please, you're welcome.
Thank you so much, Madam, for that kind introduction. Respected Chair Prakash Diwan sir, who is always an inspiration for us to do his enthusiastic research approach, etc. is always inspiration for us. Respected Co-Chair Maninder Madam, and Jachak sir, Saraf sir, and all the learned dignitaries and friends. So actually, Dr. Rajesh Kumar, who is heading medical services and clinical operations, is supposed to come, but because of some other urgent this thing here not come. So I just play this role to share few of our learnings, experiences, and what kind of solution we adopt to produce in a globally compliant herbal or Ayurveda products. So my previous speakers, especially Saraf sir and Jachak sir, made my job easy. Majority of the points I think he have covered. So few of the points to share here with this topic. We like it or dislike it, or we accept it or not accept it. So-called traditional system of medicine, Ayurveda system of medicine, or complementary alternative system of medicines are part and parcel of our life. So as per WHO, you might be knowing more than 50% of the population still uses this as a option to take care of their health. But the problem is irrational use of these medicines, especially natural products, is bringing lot of negativeness to the Ayurveda system of medicine and all other systems, because of it is not scientifically proven. So to continue with that fact, in many countries it is more than even 60, 70, even 90% of the people are still rely on the natural products. And if you see the thanks to the COVID, recent surge of interest in the natural products, because of it taken care a lot of improving the quality of life during the COVID. So people again coming back to the natural products. So if you can see the business this thing that I am not going to deal in the detail. So tremendous opportunities across the globe is rising to work on the natural products. And also as in the lack of APIs, now preference is ABI, active botanical ingredients. So that's why lots of money has been spent, including Ministry of Ayush, to work on this particular aspect. But unfortunately, under this heading, so many people are playing in name of Ayurveda. This is actually very unfortunate thing, and it is bringing a very bad reputation to entire system. So the starting from the who sells Jadi Booty roadside, including the highly qualified Ayurveda physician or any natural etc. So there is no differences. So everything is branded as a Ayurveda. That's why, because of this kind of approach, lot of negativity has been developed towards our system. So that's why this is the now reality. There are so many buyers are directly going to the, even it is very much true in the modern medicine or allopathy. So over the counter, people are going and taking medicine. It may be Ayurveda or any other medicines, instead of going to the physician with the advice. So this is the behavior. That's why even AMR is continuously happening across the country. So the another major important thing is, whenever we visit a physician, the first question with the report of elevated SGOT, SGPT and or creatinine, the first question that comes to our mind is whether he is on herbal medicine or herbo-mineral metallic preparations. It's very much true and important also, because of inherently many herbal ingredients comes with the heavy metals, pesticides, aflatoxins, and there are several reports stating that these are contaminated with such a factor unless and until you establish its safety aspects in terms of these free from contaminants. Definitely that will continue to give to hepatotoxicity, nephrotoxicity, etc. And also surge of natural product popularity during the COVID leads to self-prescription or self-medication. It a lot of negative reports. Recently published, one in Mumbai Medical College, they have reported that the Guduchi, which is promoted as one of the best immunomodulator, since 5,000 years people are taking, till now it was not reported as hepatotoxic and other any organ toxicity. But recently, it was reported and it made lot of havoc in the herbal sector. Finally, it was concluded that instead of Tinospora cardifolia, it is actually, which mentioned, Ayurveda mentions, it is safest medicine when it is used in the prescribed dose. But unfortunately, the other species, Tinospora crispa, it is reported to be hepatotoxic. So, for the common people, it is very difficult. Even the expert also, it is very, very difficult to differentiate which is Tinospora cardifolia, and it's a very complex, a lot of complexity exists in the herbal medicine. So, and also recently in Denmark, ashwagandha has been banned. Why it is happening? So, unfortunately, I could see many institutes, it may be medical colleges, it may be pharmacy colleges, it may be Ayurveda colleges, many people have started taking the, as a research work, herbal medicine. But unfortunately, the source of herbs, correct source of herbs, from where they are getting. Whatever available either in the market or even directly if they are procuring also, who are there to correctly identify. So many other factors are involved in that, because it is so complex that in coming slide I can explain few of the things. See, it's the evening hours, I think everyone will get, watery mouth if you say, after seeing this. The simple Golgappa, where we use, everybody like this, right? You might have perceived the Golgappa which you eat in Chandigarh is entirely different from Mumbai or Bangalore or even Delhi. Why? What might be the reason? What might be the reason? The pani which you prepared, see there is a difference, which Diwan sir also asked. One is, one is inducing contraction, the same, how can it is relaxation? Sir, this is one of the best example. More than 500, reported or named varieties of Pudina mint. That's why sometime we prepare any dishes out of this. Sometime you'll get a very tasty. Sometime in the same they prepare, we complain, no, it is not good. This is the reason why. The one which is Menthas picita, it is the best carminative and the analgesic. Why the same another Mentha piperata, it is reverse action, gastroprotective. That's why sometime when we have the Golgappa, it is giving, very good digestion, etc. The same time somewhere it will be gastric irritant. So this is actually complexity that unless and until we understand, especially when you are taking the topic for the research, there if you don't understand this, we will land in either false positive or false negative. And the sake of academic achievement, we will report it. Okay, we will report it the same, but unfortunately the same will be quoted by some of the regulators saying that even the good plants is having toxicity. That might be the one of the reason. Yeah, completely different. Why? How many will understand that? Yeah, that how many? As you are the expert, you can understand. But at the research level, nobody is bothered to go and check whether it is a correct species. Very complicated. I will show the case study of Mucuna pruriens. Okay, velvet beans. It is recently, popular for to treat the Parkinsonism. So I'll show the one case study. Then you will understand the complexity of herbal medicine. See in the market now, ashwagandha is so popular. So many companies are making. So days are gone. Previously, so-called Vaidya, doctor himself used to procure, prepare and give. That time even in the folklore practices, people are there. That's why they are getting consistent, good outcome. But now it is not possible because of lot of transformation. So that's why we always I'll come to that, sir. I have that slide. I'll come to that. So, yeah. Whatever question you ask, we have the experience because last 95 years Himalayas, researching the nature and enriching the life through herbal medicine. So we can have lot of experience and examples we can give. So that's why always we make the statement that my ashwagandha is not your ashwagandha. Why? Because the company across the globe, Dolo 650 means Dolo 650 only. There is no chance of differentiation in that. But when it comes to the herbal medicine, lot of differences will come. So also there are several reports stating that drug interactions, not only drug interaction, there are many reports herb interaction. How many of us to taken care of that or bothered to take that? So when I am taking the ashwagandha, whether can I is it okay to take other medicinal plants as well? Because when it has an action, definitely there should be some reaction through the drug or drug-herb interactions. So these are some of the examples Guduchi I have explained. In want of time, I will just run through this thing. See, the dry form, who can identify this? This is the main culprit why Guduchi or Tinospora cardifolia has reported to be hepatotoxic. Instead of using the cardifolia, people might have taken this, so that led to the hepatotoxicity. Cinnamon, simple, the very popular. The herbal sector is so unorganized. When you procure it, very difficult to differentiate whether it is Cinnamon xylonicum or Cinnamon cassia. So both have different, pharmacological activity profile. Same with the Gugulu, one of the best anti-inflammatory analgesic. But in the name of Gugulu, many resins, oleogum resins has been adulterated, so you will not get the consistent result. So this is a everyday we use. This is asafoetida. So whatever the asafoetida or hing we are getting, so it is so much adulterated that it is only, I think, fit to use in the kitchen. I don't think any of the, for the medicine, it may not suitable. The black pepper, heavily adulterated with the papaya seeds. Papaya seeds comes in the 50 rupees. This black pepper is 1,000 rupees. But for to make the money, supply will just simply mix it and get. How we are going to, unless and until histochemical studies we do. Yes. Yes, sir. Correct. Yes. So for that, actually we have the solution. What should be that solution? That also we will discuss. That honey already I think you have given the explanation. And also Kesher. We generally consider the Kashmir Kesher as the best. Actually, if you see the pharmacological profile with the highest authenticity, which comes from Iran is the highest quality. So like that, these are some of the, I don't want to go in detail, we can discuss hours together. All these factors matters a lot in getting the expression of pharmacological activity. The simple soil pH will alter the quality of the plant material. Sometimes the haldi, when you used to make the pickle, when you cut it, you will get the orange color, sometimes it will be the yellow color. So there is a simple change of pH of soil itself alter the expression of our pharmacological activity and the phytochemical this thing. So these are some of the example which I am not going to explain in detail. So each and every factor that will definitely affect the outcome of things. That's why the solution is quality by design. The evidence-based research, if you subject any herbal ingredient to develop any product or translate from tradition to product, seed to shelf, you need to subject into the thorough QBD, quality by design. Then only you can prevent the even variability as well. Safety profile, efficacy profile, everything you can establish it through quality by design. So this is the overall picture how you can establish any good globally competent herbal products through the evidence-based research. Again, 2-3 hours we can debate on this. It's a very looks very simple, but it's a very complex. Each and every batch should be subjected to lab scale, pilot scale, commercial scale. If you establish, that's why it takes not less than 3 to 4 years. It's just like how modern drug discovery happens. Same way, if you develop any products by following all GACP, morning Gupta sir also highlighted why GACP, GAFCP and GMP all are important. So if you process or follow this process, definitely the outcome will be the best. So this is the Velvet bean or Mucuna pruriensis. Recently it got in the name of Mucuna pruriensis. When we try to procure across India, not less than 15 seeds has been provided to us. So when it comes in 1 kg, 2 kg, it's very easy to this thing. In the commercial batch, it comes in tons together. For us, for example, ashwagandha requirement is 200 tons. Neem is 8,000 tons per year we are using. Okay. So then how you can do the quality control? Otherwise, lot of batch to batch variation will come. So in one batch which you use the product may not give the same result as in the successive this thing. I'll show, sir. These all are, the outcome is, morphologically same, If you do the microscopically, almost same, histochemically same, because L-DOPA is a very unique this thing. The only technique we can utilize is RAPD, DNA fingerprinting. The one which shows 258 kb is the correct source of mucinopruriens. So this, if you establish this kind of authenticity and the purity, etc., definitely your product or your research outcome will be the best. So that's why we have the practice of, regenerative agriculture, the promotion of cultivation of medicine plant across India, its natural habitat. That's how you can get the Himalaya product always consistent, because ashwagandha, see the, sorry, I think it's moving somewhere. See the quality of ashwagandha. So 200 tons means, if you are, it is also very important for the farmers to improve their economy as well. See, we promote as a border crops, intercrops, like that. So rotation of crops. So like that, we have a project called Sampurna. So across the India, we have that program. We give every plant material, etc. We guide them how to this thing, and we will buy back. At least 10 years, we will have the contract. So that kind of approach we have. That's how we. This is a complete center to the. We have the SOP for that. How much should be cut and process and collect. Another 2 minutes, I will conclude. So that's why it is very, very important to establish each and every parameter, because for the consumer's delight as well as prescriber delight, all these should be properly established. And also, very important is to prevent the batch-to-batch variation. So that's why 3 lab scale batch and pilot scale batch and commercial batch. If you establish it properly, every process, you will get consistent product. So this is no need to explain. So the same should be subjected to all the efficacy aspects, including toxicity and OECD guideline, all the toxicity tests. And finally, as per global standards, we need to establish the clinical phase 1, 2, 3, etc. So finally, whatever the outcome, if it fulfill all these aspects, safe, pure, authentic, and efficacious, and the stability as per ICH guideline, if you establish, definitely it will give the best result without any fear. So this is actually what we want to share. So because of want of time, I just rushed this. So we have a stall with almost all these information displayed. We have our enthusiastic team with Dr. Azim, Dr. Eliza, and Munish with us. So anytime you can come, there we can have especially students. We expect our researchers rather than students, who the researchers, you can come and interact. Whatever the experience or little knowledge we have, we can definitely share with you. So thank you so much for the organizers and everybody for this thing. Thank you.
extract, and that created actually hepatotoxicity. And this is what has been proved already. So that is what the reason. This is the reason.
In fact, there is a issue in terms of aerial parts versus roots. The aerial parts are not used and the roots are used essentially in some of the pharma industry with this natural products.
Leaves is banned. Ayurveda don't recommend to replace roots with leaves, and that is the reason we don't have safety data on leaves. So it cannot be done. That is what we can do.
Ayurveda doctors, the first answer what we have given is ashwagandha, aerial part, you might have mixed. This is known for that. Inospora, crispa, known for toxicity. Ayurveda never
Crispa is
recommends.
also-
But bad name come to Ayurveda and herbal because of this kind of this
Mischievous way of doing it.
Thank you, Dr. Ashok. So, we are privileged to have so many learned experts over here. So with this, I also wish to thank my chairperson, Dr. Diwan, and for his esteemed presence and motivating the speakers as well as the audience. Thank you, everyone.
Now I request our chair to please grace the dais and felicitate our speakers. Our first speaker is Dr. M.N. Saraf. Dr. Sanjay Jachak. Dr. Sanjay Jachak. Dr. Govind Singh. And Dr. Ashok. Now I'll request Dr. Manish to please felicitate our chairs, Dr. Diwan and Dr. Maninder Karan Vashisht. Dr. Manendra Karan Vashish. Yeah. Yes, I will request all the speakers come on the dais for a group photo. With this, we bring Symposia 19, Advancements in Nutraceuticals and Herbal Therapeutic Technologies, to a formal close. On the behalf of the organizing committee, we express our deep appreciation to our respected chairs, distinguished speakers, and all the members of the audience for their gracious and attentive participation. Good evening, everyone. Distinguished guests, colleagues, delegates, and dear audience. Welcome to Symposia on Alternatives to Animal Testing in Pharmacology, Ethical and Translational Perspectives. This symposium brings together researchers and advocates working at the interface of ethics, basic science, translational pharmacology to discuss humane, scientifically robust alternatives to traditional animal models and the opportunities they present for drug discovery and safety testing. This session is under the distinguished chairmanship of Dr. Ashish Kakkar and Dr. Poonam Pipalani, whose scholarly leadership greatly elevates today's proceedings. Now I'll introduce our esteemed chairs. Dr. Ashish Kakkar is a faculty in pharmacology at PGI Chandigarh with expertise in clinical trial. drug development, infectious disease, neuropharmacology, and drug safety. He has over 42 publications with over 1,000. It's my pleasure to introduce Dr. Poonam Piplani, the professor at University Institute of Pharmaceutical Sciences, Punjab University. A specialist in pharmaceutical chemistry, she has guided numerous MPharm and PhD students and has contributed significantly to the discovery of novel drug-like molecules. As a senior faculty member, she plays a key role in research, curriculum development, and shaping the next generation of pharmaceutical scientists. Now I request Dr. Poonam Piplani to please introduce our first speaker, Dr. Aarti Singh. I would like to introduce our the second chair, Dr. Ashish Kakkar, faculty member in pharmacology at PGI with expertise in clinical trials, drug development, infectious disease, neuropharmacology, and drug safety. He has over 42 publications with over 1,000 citations and was elected a fellow of British Pharmacological Society in 2024. He contributes to antimicrobial stewardship, supports evidence-based medicine, writes on drug pricing and health policy. As an active reviewer, mentor, and educator, he guides students, researchers, while strengthening clinical pharmacology and capacity at PGI. We welcome both of our chairs. Okay.
Good evening, everyone. It's my privilege to introduce our first speaker, Dr. Aarti Singh, a neuroscientist specializing in mitochondrial dysfunction, chemobrain, zebrafish models, and nanoneurotherapeutics. She has over 50 publications, major DST SERB funding, IBRO fellowships, and serves as a review editor for Frontiers in Clinical Diabetes. I welcome Dr. Aarti Singh to please share her views on zebrafish as a 21st century model for ethical biomedical research. Thank you, Dr. Aarti. Please. The speakers are requested to kindly follow the time limit. It should be not more than 15 to 20 minutes, please.
It's CUIPS. Just open this. Yeah. From this now?
Yes, ma'am. You can create
Just click on it.
a.
Okay.
You just have to click on it.
So first of all, very good evening to all, and thank you very much, ma'am, for introducing and for introducing me. First of all, the topic which I'm discussing is zebrafish as a 21st century model for ethical biomedical research. So I will be broadly talking about zebrafish, but as you all know, or you all are aware of, that alternatives are more into research these times. So because most of the research we have done on zebrafish, that's why I'll be talking more about zebrafish. So obviously, when we talk about the initial screening of the novel molecules, drug discovery and development, it may be divided in two types. One is the target-based screening methods, another is phenotypic-based methods. But we all know that only small fraction of molecules, they enter into clinical trial and finally into the market. So what is the rate limiting step when we talk about screening of those novel molecules or moieties? So it is those models which mimic the human disease conditions. So when I say zebrafish, it is one of the model which is having potential in translating all those human disease conditions, and also screening of all those novel molecules into the clinical trial. So the studies which assess the bioavailability, safety and efficacy, these are major concerns which are briefly talked about, and they are briefly tested for the novel molecules in drug discovery and development. So the crucial step during this particular phase where the molecule it is tested, initially it is the target which needs to be discovered first, and then target it needs to be validated then. After that, the molecule it is screened for its potential. So those novel molecules, they then travel the process of identification and optimization, where so many things are come up with the advancement, including the in silico techniques, right? Then we enter into preclinical development, where the in vitro assays, they plays very important role, followed by in vivo testing of those molecules, where these animal models plays very important role. So we need to have some definite animal model which mimic the human disease condition, 100% mimic, but it is not feasible that any model can mimic a 100% disease condition. So as far as the ethics in biomedical research is concerned, we are more into addressing all those concerns which are related to ethics. And nowadays, it is more stringent as compared to the past scenarios. Regulatory pressure, it is more nowadays, which limits the use of the higher vertebrate for basically the toxicity assessment in drug discovery. And also, as far as the new approaches in testing, drug testing is concerned, the reducing reliance on the animal models. And also the 3R principle, which is the ethical principle, talks about replacement, reduction, refinement. They basically talk about the alternatives. What is the alternative? And here comes the role of those alternatives. What could be the alternative? It can be the stem cells, it can be these cell cultures, which can mimic the conditions. It can be 3 tissue models, which mimic the microenvironment of diseased tissues. It can be organ on chips. We are more like researchers, they are working on it. In silico prediction, before going into like the in vitro testing, in silico studies are available nowadays. We are doing docking, we are doing network pharmacology, where those molecules can be screened before they finally enter into some biological assays or cell-based assays. And finally, the in vivo studies. Humanized mouse models, the further advancement in terms of mimicking more human-like models and the data, and then finally the non-mammalian models, the lower vertebrates, where C. elegans, Drosophila, zebrafish, they plays very important role. And at UPES, Dehradun, we are having facility, the scientists, they are working on all 3 areas of alternatives. So why zebrafish? And before that, it was very first used in the research in the field of developmental biology by George Trestinger, and he utilized this model for his PhD research in 1960s, and later on he was considered as a father of zebrafish research, and he used it to simulate the human disease conditions later on, including the behavioral biology, drug testing, genetics, immunology, metabolism, oncology, and furthermore. Later on, the genetic similarities means on the basis, because we are talking more about the involvement of gene in a disease condition, and their genotype plays very important role. So genetic disease, obviously involvement of gene, it leads to prediction of early biomarkers in drug discovery and development. That's why they are very important. Another important advantage that this particular model, which is zebrafish, it is cost effective. It is also genetically tractable, because human genes and zebrafish genes are more than 70% similar, and ethically it is acceptable model. After the notification of CCSCA in 2021, they have considered it to be used in the research for developmental biology. And it also serves as a connecting bridge or a link between the in vitro assays and mammalian in vivo studies. So, and past 20 years, the studies which related to zebrafish, they greatly advanced our understanding in terms of disease, how how the development occurs in case of pathophysiology and physiologically, also the toxicity assessment. And this is the beauty of this model, that all 3 different stages, whether it is egg, whether it is larva, or adult zebrafish, all three of them, they have been utilized in the research for different purposes. For example, if it is egg, you can use it for studying the different developmental aspect of the environmental pollutants. Or if it is a drug or a novel molecule, we can check the toxicity aspect of that novel molecule on the developmental phase of the zebrafish. Similarly, larva and adult zebrafish. Another important advantage is transparency of embryo. We can see each and every phase of the embryo or the larva within the egg because it is transparent in nature and it is easily visible. And rapid development, the egg usually hatch within 3 days, and it took around 48 to 72 hours for the major organs to develop. Within this hour, all the major organs just get developed within the zebrafish. High fecundity and the property which indicates that the laying of egg, the property which defines it, it's like 200 to 700 egg in a single day a zebrafish can lay, right? And small size, obviously because of its small size, easy in handling, low maintenance cost. Compound screening, which is a part of a major early drug discovery phase where high throughput screening is done on the novel molecules. So this model, it considered to be a best model for screening the n number of molecules within a limited period of a time. As far as ethical advantage is concerned, although as per CCSEA, IAC approval is required for doing research on zebrafish, but if it is less than 5 days, means 5 days post fertilization, no ethical approval is required. Then comes in pharmacology, what kind of areas researchers they are working on. It is the toxicological aspect, including cardiotoxicity, hepatotoxicity, neurotoxicity is the one area in which we are working. And in drug discovery, high throughput screening, I have already discussed about. Another important thing is pharmacokinetic and metabolism aspect, where the enzyme, which is the metabolic enzyme, CYP, cytochrome P450, it is just comparable. The genetic aspect is comparable to that of mammals. Neuropharmacology, where the molecules for its antidepressant, anxiolytic, and cognitive enhancing properties, they have been tested. Cardiovascular pharmacology, the heart of zebrafish, it is having regenerative potential. And we can, take multiple times. The blood can be taken up for the pharmacokinetic analysis multiple times from the zebrafish herd. And cancer pharmacology, the xenograft models can be easily developed in zebrafish. Obviously, 3R principle, it is equally followed in zebrafish. Regulatory acceptability, it is equally, because it is approved by CCSEA and it is acceptable and as per the guidelines, CCSE and IAC. Previously it is CPCSEA, now it is CCSEA. And obviously it is ethically and economically sustainable model. And it goes with the line that One Health, Sustainable Science, it's the best model that fits in this particular goal. Education and skill development potential, the model is having enormous potential as far as the skill development of the students who are undergrads. So for that, introducing zebrafish research for young pharmacologists to just encourage them for ethical research, it really plays an important role. As far as industrial perspective is concerned, in early 2000, the pharma companies, they have utilized zebrafish, the larval zebrafish, for screening the novel molecules. And the companies like AstraZeneca, Novartis, Pfizer, they are utilizing this model for screening the molecules indirectly. The molecule, it is currently in a clinical trial. They have validated the potential of those molecules in zebrafish, and now it is in clinical phases. Other partner companies like academia, they are also participating nowadays. More of the zebrafish lab, they are established. The CROs are also participating in zebrafish research. And from 2006 to 2013, 24 zebrafish studies, they were co-authored by pharma researchers or pharma scientists. And a survey, it has shown that 59% of pharma researchers, they are utilizing zebrafish as a model, mainly for CNS, cardiovascular, auditory, visual system, retinal regeneration research. It is one of the most researched area in terms of model, because the retinal regeneration potential is also very good in terms of the model as far as this particular model is concerned. So zebrafish, as far as Viksit Bharat vision is concerned, this can be considered as a best model. if it is Viksit Bharat, then indigenous zebrafish research facility. Nowadays, there is a facility called, it is in Navi Mumbai, and they are now participating in developing all those infrastructure which is related to zebrafish, whether it is the behavioral apparatuses, whether it is the NMA's tracking system, right? So all of things, they are now scientists or the researchers, they are participating in it. Cost-effective alternative. It is one of the cost-effective alternative to rodent models, which are considered as a best model, the past model, or we can say that. Training young scientists in 21st century ethical pharmacology. It is one of the mission of the Viksit Bharat. It is also playing a role in bridging the academia-industrial gap, because more academic institutes, they are participating in zebrafish research, so that can enhance the potential in screening some novel molecule, and that the technology can be transferred to the industries. Current research, as you can see in the flow chart, how the research, it is increasing from 2009 to 2019. So this is how it shows its potential, that yes. And there is a database which is called a zebrafish database and zebrafish information network. It is available, and the website is this, where the database in terms of genetic or genome, database in terms of publication, it is available. If you want to refer, if you want to if you want to study for any specific model, you can search these databases. They are available. We are also submitting our studies on these databases so that it will be helpful for other researchers. And the brain atlas, as you all are aware of rat brain atlas, that was given by George Paxinos. So this zebrafish brain atlas, it is University College of London who developed the zebrafish brain atlas. You can explore all the areas, their functionalities, and it's very easy to locate all the areas of zebrafish brain. So this is how the drugs which come up in market, or not market, but they are currently in different phases of clinical trial, and it is tested on the zebrafish as a model for these specific conditions. So this is how the pharma research industries, they are participating in it. So major challenges and future direction is, the major limitation which I found in this particular model is the pharmacokinetic differences as far as the pure genetic is concerned, because I'm saying 70% is similar, but again, if it is 30 to 25%, if it is not similar, from that aspect, a pure pharmacokinetic studies, it is very difficult to perform on this model. Because for that, we need to have some blood-related parameters or studies, which is very difficult to isolate from zebrafish. Apart from that, there is a need for standardization or validation, right? And capacity building, obviously, the zebrafish research, more and more zebrafish research-oriented center need to be developed in the India itself. And also integration of AI. What is AI? It is artificial intelligence, and also the network pharmacology, how they are advancing our research, means before going into in vitro or in vivo studies, we can screen the molecules, we can screen the molecular pathways with the help of these softwares, right? So these are some of our publications where we have established the model, and this is the model of Huntington disease, and we have assessed the potential of casein measles, and in this the piperine, it is incorporated in these models, this particular formulation. Another model is anti-chemobrain activity of amino acid assisted ferulic acid solid dispersion, and here also we have utilized ferulic acid and we have assisted it with amino acid which is leucine, and the model here which we have used is a chemotherapy induced cognitive dysfunction in zebrafish. Next model is the LPS-induced inflammation in the zebrafish, and here the QCT-loaded TPGS-coated solid lipid nanoparticle, they have been assessed for its potential. Then this is one of the best paper where we have established the model of hypoxia in zebrafish, and we have tested the effect of intermittent fasting along with the herbal agent which is hydroalcoholic extract of Setella asiatica. Then we have also developed a model, similar model, which is chemotherapy-induced cognitive dysfunction, and here piperine-loaded metal organic framework. So recently, the Nobel Prize, it has been given to the metal organic framework, and this framework we have utilized to incorporate with the piperine to enhance the potential of piperine. And these are some few papers, including epilepsy. Yeah, sure. This is last slide.
Because already we are running short of time.
Some of the patents, and finally concluding, so yeah, from tank to translation, zebrafish, it plays very important role, and it is having immense potential in translating all those results into clinics.
No doubt.
Thank you.
We would like you to continue, but it's a very interesting topic. And even in this conference, I think people are really working on this zebrafish because we had so many sessions and workshops on this.
Yes.
How reliable is this, the model, the results, in comparison to the other in vitro results?
As far as other models are concerned, first of all, it is the literature search, literature data that we have, that it is supporting. Another thing, when we reproduce same thing in our lab, we found the result. The one study which is on hypoxia, we were like the hypoxia model where the oxygen level, it was reduced to approx 1.6% in water, then it produced that kind of ischemia on the brain. And we have tested it, we have validated it. Most of these methods, they have been reported in literature, and we have reproduced that.
Thank you very much. Any questions? One question from your audience.
It was a nice talk.
Thank you, sir.
I would like to know what is the most common and suitable route of administration of investigational drug, especially in the larval zebrafish stage.
Okay.
And if it is just the immersion, just immersing into the media, then how and how much does do you have any information on how and how much the drug enters into this zebrafish?
So there are 3 different stages, as I have said: egg, larva, and adult.
Right.
So in egg, we can directly micro-inject that particular component inside that. But in case of larva, the same thing can be given to water or the media in which the larva it is there.
Right.
But for that, we need to quantify.
Yes.
Right, with the help of HPLC, GCMS, LCMS, we need to quantify how much the component it enters into the system.
Okay. But how does it enter? Do you have any information on how the drug enters from the water
Because
into the
in case of
zebrafish?
larva, gills are almost developed, skin is almost developed, orally also, so there is not a single route for administration via water method. In case of adult, it is orally, the well-established method. It is the drills, which is Dr. Reddy's laboratory, they have established this method. And intraperitoneally also we have given that drug, then intramuscular also.
Yes.
These are the routes which are explored in adult zebrafish.
Okay, thanks. Thank you so much.
Thank you. Thank you, ma'am.
Thank you. Okay, now I have the privilege to invite Dr. Rohit Bisht, who is a scientist of regulatory toxicology working with PETA India. He did his PhD from University of Auckland, New Zealand, and his postdoc from BITS Pilani. He has completed numerous projects from AIIMS New Delhi and University of Kansas, USA. His research work has been recognized by American Association of Pharmaceutical Scientists. And now I invite Dr. Rohit Bisht to enlighten us on humane relevant methods in drug development and safety testing, where we are and what's next. Dr. Rohit, over to you.
Sure. Good evening, everyone. So in today's talk, I will share the information about non-animal methods and what significant changes is happening on global regulatory bodies, what PETA India science team is doing globally and in India for the advancement of non-animal methods, what Indian regulatory bodies are changing in regulatory guidelines regarding the non-animal method acceptance, and what type of programs or initiatives we are working or running in India for promoting non-animal method, and of course at the end, the most important, the challenges and opportunities which relates everyone, industry also, academia also, because with non-animal method, it looks good when we talk, but when we do it in practically establishing any technology in our lab, there are some regulatory changes, there are some technical challenges like hands-on training, expertise on handling these, development of these, because if you search any non-animal method, whether it is organoids or organ-on-chips or tissue culture 3D models, regulatory bodies approve them, they are integrated in OECD test guidelines, but most of the models are commercially available in abroad. in Western countries. In India, now we have started developing these organoids, organ chips, but they are at initial stage. We have to validate them. And for validation also, there are a lot of requirements from regulatory bodies. So these are in India. So in India, this first one is hands-on experiment challenges. Second is the funding, available of funding for the development of these alternative models in India. So let's begin with some introduction of non-animal method, then slide by slide we will talk about different aspects related to non-animal methods. So initially, I will just highlight about some limitation with animal model. Then these limitations are endorsed not only by the scientists, but by renowned scientific agencies like NIH. And as you know, like 90% of drug which shows the promising results in preclinical trials fails in the clinical trials. And it has been investigated, the results concluded that about 18 to 89% of research are irreproducible. So reproducibility is a lot of challenges there when we talk about academic research also. So this is also a concern with the animal testing. And according to some literature, has been found that only 10% of highly promising studies enters into the clinical trials. And even though the failure rate is more than 95% when we consider about some certain disease conditions like Alzheimer, still there is no alternative model. We have to use the animal models. Like cancer, HIV vaccine, strokes, and sepsis, these in vitro models are still in the R&D phases. So our ask with the regulators and with the scientists that whenever you are working with the animal model, if there is an alternative model which can be used, you should shift from the animal models to the in vitro models, or do a comparative study, and if it justify and you feel confidence on the results, you can transform from the animal model to non-animal model. And in the areas where there is no availability of in vitro models, then of course you have to use the animal model. There is no other options. Considering this, we have the PETA scientists have developed this document known as Research Modernization Now. This is a roadmap for how industry or academia and regulatory bodies can shift from the non-animal models to in vitro models. So in this, we have discussed about the different type of animal models, the limitations, different sections of biomedical research, cardiology, cancers, cell gene therapies, new upcoming therapeuticals categories, what type of animal models are available, what limitations are there, and what are the in vitro models which are available there, which you can use, and what the regulatory bodies are talking about these in vitro models. And then we have developed some strategies how you can shift from this animal models to in vitro, because this transition is not easy. It takes time. It will not happen in one day, for the regulators also and for the industry also. To change its whole quality management system in a small time is not possible. So we are starting from the ground, from the first step, spreading the awareness, hands-on trainings, and then slowly communicating with regulator and so on. But the main objective with this is that replacement of animal method with the non-animal method in the areas where animal models have already shown to a poor surrogates for humans. Critically reviewing the additional area of research to determine whether animal use can be phased out. So if you go to the website of Animal Controlling Authority in some countries, they have database. You know like how many animals has been used in 1 year for different projects. We are communicating with CCSE also, DBT, BIRAGs also, that we should have developed such type of database, like what type of categories of research there where the proposal is received, what type of animal models this year we have requested for, and ultimately after that we have to also enter the details about what is the output, because the next person who want to use that models will see what either this animal model is having some significance for the studies or not. So it will save a lot of animal lives. Then redirecting funds. So we are in communication because we work with the regulators, we work with the scientific agency, we work with the industry, we work with the academic and industrial scientists for the advancement. So we are continuously asking with the DBT and other scientific agencies to increase the funds or allocate a separate funds for the non-animal development. So recently DBT has announced a separate funding opportunity for development of in vitro disease models. And now we are also planning to develop a center of centralized facilities for the development of in vitro models. So introducing ethical harm benefit analysis, this is also in process. And education training, this is the most important part, which is we are doing with the industry also, with regulators also, and with the academic scientists. So last time we have sent about 7-8 Indian scientists to Institute of In Vitro Sciences, US, for hands-on training on different in vitros. And next 2026, we develop a collaboration with the National Institute of Biologicals, where we are organizing this hands-on training for industry and academic scientists in collaboration with Institute of In Vitro Sciences, US. So starting with the drug development, this is the project which I am leading in India, equine-based therapeutics. With the equine therapeutics, there are some risk of mild allergic reaction, serum sickness. There is a risk of transmitting of microbes, which is very important or critical when we are in pandemic conditions. And there is a batch-to-batch variability and short plasma half-life. That's why when we discuss about the equine-based therapeutics, we can't store them for a longer time. So if you see the latest government initiative about reaching, promoting the rabies vaccine or snake bites, the vaccine scarcity is there to, in urban and rural areas. So what WHO, because this project is requested from WHO and UK Health Agency, is that they want to shift from the equine-based therapeutics to the recombinant technologies for the old molecules. So we are in India, we are promoting recombinant technologies in collaboration with DBT. They have national biopharma missions where they organize different workshops with the stakeholders to promote recombinant technologies. We are also working with CCSA because they have the equine guidelines, which is 2001, which is not updated. Now they have constituted a new committee and reviewing these documents. We have also had an opportunity to talk with the DCGI, DBT Biraj, so that now you will see soon there will be a revised guidelines on this. We have a collaborations with AppCam for the development of monoclonal antibodies. We have a collaborations with AppCalis. We also working with US FDAs in developing the old molecules into new recombinant molecules. We do our research and developments. We have developed recombinant diphtheria antitoxins because the available old one is equine-based. So this is the project which is supported by UK Health Agency and WHO. So in animal testings, we have different type of non-animal methods, reconstructed human tissue models, spheroids, organoids, bioprinting, precision cut organ slice. We have cell-based assays. We have in silico models. We have now organ-on-chip models where we have a single organ, multiple organ models. Now we have combination models also, in vitro coupled in silico, organoids, inner organ chip models. So these are the models which are commercially available. These are the leading company, MedTech, IPSCON, which have regulatory approved model in vitro method which company and regulatory bodies are using. And also Indian companies are also right now using this, and they are integrated in OECD test guidelines. So I'm just giving you the slide highlights about, and then I will go with the main topic about regulatory significant changes. So this is spheroids. This is organoid, but the important thing with organoid is that in 2022, US FDA has granted first approval of the use of organoid and organ-on-chip technology for new drug approvals. 2025, US NIH announced a reward of 87 million in contracts over 3 years to standardize organizing modeling center. And this is the curator, they having their organized platform. So they have got the first US FDA IND approval for the anti-cancer drug, which is tested and on organoid models. So this is how the regulatory body is also accepting the alternative models. So this is just an example of an organic model by Cherry Biotech, how they have this, which can easily use in a lab, a small setup, and different areas where they can use. Biochem printing already installed in different academic institutes in India. We have in MIT, NIPERS, IITs, they are using. In this, you can use it with different type of cells. You can choose the organ. Then, depending upon the cell, you can develop these 3D tissue models. And depending on that, your project, you can, take a lot of information from that. So in different areas, they can be used. In silico models, we have developed a first regulatory approved QSR toolbox, which is known as OECD QSR toolbox. We have a collaboration with OECD on different projects. This is one of the projects, and this toolbox is already OECD approved and freely available for the use. US FDA also accepted this PBK simulators for the pharmacokinetic studies of the drug molecules. And this in silico models, we have different, but recently they have also used for reproposing of drugs which are very much used. Even in the COVID-19 cases, we have remdesivir, which is reproposed for its use. Organ of chip models, now we have organ chip models for each and every organs. And I would like to discuss about this liver on chip model, because this is the first model which is accepted by US FDA for its iStent pilot program. So US FDA regulatory bodies have their separate iStent program where they, depending upon the technology potentials, they accept this proposal and collaborate with the stakeholder for the advancement and whole study and approval of this model in a large scale. This is another models which you can use, precision tissue slice models from the diseased person, and then you can use this for the drug screening. Now, this is the important section I want to discuss about global regulatory shift. So first, I will discuss about the US FDA. So US FDA have from time to time, so I'm highlighting the recent changes. There are a lot of changes. If you go to the US FDA website and type non-animal testings or alternative testing, they have different changes, modification, guidelines for each and section. Monoclonal antibodies, vaccine, veterinary vaccines, pyrogen testings. So time to time they release different type of guidelines. Recently they have FDA Modernization Act where they have accepted these all in vitro methods for the preclinical testing of new investigation drug. Following this, India has also updated their new drug and clinical trial rules. Now this amendment has already approved by the government. They have included all in vitro models, in silico, in vitro, everything in the guidelines for the investigations, preclinical investigation of new drugs. So this is another recently happens. The FDA announced the plan to phase out the animal testing requirement for monoclonal antibodies. This is still open for the stakeholders. So we are also proposed some of the tests for the recombinant monoclonal antibodies are in vitro and in silico coupled method, which we have proposed for monoclonal antibodies for immunogenicity, specificity and tissue bindings. Because for the immunogenicity, the rabbit model which is used by the previous old guidelines, ICH, it is already known that it is not good model for immunogenicity investigation. FDA Advanced Drug Development Innovations, iStent, is a program for which, for medical devices also, if you go to the MDTT program, this is a separate program started by the FDA where they have accepted in vitro models for the investigations of the effects by using in vitro model. We are working with the FDA on the MDT. We have used monocyte activation test. We have validated this test for medical implant, tooth implant basically, and we submitted our validations data with the US FDA and it is in discussion. So these are the different guidelines for an alternative for their validation, qualification requirements. So a lot of changes is happening in US FDA. US FDA also do the collaboration work with the stakeholders. They have developed the comprehensive in vitro polyarrhythmia assay, which is, it's a big study concluded, and now these are included in ICH guidelines. They have also did the cross-cutting FDA applied research, liver microphalangeal systems. They have also working with the stakeholder for 3D model for lung airways. So inhalation toxicology, a lot of things is going on in inhalation toxicology in US FDA and European pharmacopoeia. There are different types of in vitro models which are already approved for inhalation toxicity. So this is about European pharmacopoeia. A lot of things are going on in European pharma. Even though for non-animal toxicology or non-animal testing, European Commission is ahead of all the regulators. So they have completely removed abnormal toxicity test. They have also removed the in vivo methods by in vitro methods for quality control of vaccines. They have replaced the histamine sensitization test for vaccines, toxin. Now they are decided to remove all the rabbit pyrogen tests from the monocarp. So I will go to the India. So what we do, we work with the regulator, policymakers, scientists and industry for advancement of non-animal methods. These are the different projects that we are using at different countries. We work with the regulators, policymakers on different areas, providing awareness, hands-on training, communications with the regulator, proposals, validations, supporting industry for OECD test integration programs. So these are the recent changes which has happened in India. These are small changes. Complete removal of, if you go to the Indian Pharmacopoeia, the regulatory books, they have completely removed the abnormal toxicity test. Target annual batch safety test is completely removed by the 13 veterinary vaccines. New drug clinical trial amendment, I'll already discuss. New revised guideline for biosimilars. So we work, biosimilar manufacturers of India contacted us on provide us the authority where we have communicated with DBT committee and CDSCO. Now this revised guideline where they have waived off the requirement of animal test for the market authorizations of biosimilars. And these are the different others. So other than this, these are our initiatives. We do non-animal method focused sessions. We provide hands-on trainings to the scientists. We also award in equipment or instruments and grants, research grants, and all this. Yeah. Thank you.
Thank you.
Yes, this is fine. Yeah. Yeah.
So first of all, thank you for giving us a very comprehensive overview of non-animal methods of testing, particularly toxicity testing. Leaving aside the ethical issue, what bothers me is the premise for shifting from animals to other models is that these animal models are useless anyway when you move to humans. Most of these candidate drugs which are showing success in animal models are not succeeding in humans. So where is the guarantee that if you use these alternative methods, they'll be successful in humans?
So for this,
one other thing is that many of these models are going to be proprietary, so they're going to be much more expensive than animals.
So answering your first question is that I'm giving an take example of pyrogen testing. So please, sir, you can have a seat. So in pyrogen testing, we are working with regulators and industry about monocyte activation test, which is in vitro test for pyrogen. Initially, in regulatory guidelines for pyrogen, we use rabbit pyrogen test. But where rabbit pyrogen test is not good test because it's having a variability, European Pharmacopoeia already removed. Indian Pharmacopoeia already removed rabbit pyrogen test from majority of the drug monographs. Now they have included bacteria endotoxin test, that is LAL assays. But the problem with the LAL assay is that first it is depend upon the blood of horseshoe crab. Second is that it only detect endotoxin pyrogens, it can't detect non-endotoxin pyrogens. Non-endotoxin pyrogens is very important when you consider the biological formulations. So now, currently we have only one test, monocyte activation test or either rapid pyrogen test for the detection of for the pyrogen, any type of pyrogen. Now, the technology and challenges in the term formations. What we are doing, we recently had a meetings with all the TMAT test provider, global TMAT test providers. MAT Research is that, MAT Biotech is that, CTL MAT is there, Immunospot is there, Indian providers also there. We take their views. Then we had a separate meetings with the regulators. CDSCO is there, IPC is there, DBT BIRAC is there. We invited CDTL, CRI, NIB is there. We take their views point, how best the way to take these projects so that the transition will be smooth for industry also and smooth for the regulator also. Then we had a meeting with the industries. All the leading bio manufacturers are contacted. Now what we are doing, we have developed a validation protocols. We are doing the validation of monocyte activation test kit. And we do the this validation in collaboration with National Institute of Biologicals. We are in collaboration with National Indian Pharmacopoeia Commission
that
also to show them how effective is as compared to the rabbit pyrogen test to build a confidence that, yeah.
No, that is specific for the pyrogen test. When you go on to move on higher models like organoids and 3D tissue models.
So for this,
only time will tell how successful.
No, because for this, because if you consider monocyte activation, just as an example, it is already validated, a lot of literature is there. But what regulator wants is that you have to do a validation, product-specific validation, again in India, in Indian specific condition. Then you have to review data. If I take example of organoid, it's already validated, everything's done. Again, we have to do a specific validation in collaboration with the industry, we have to do the comparative studies with the ongoing models and to show and convince the regulator that this models is giving the same results as compared to the old traditional methods. And then they are agreed to, because unless it comes into regulatory guidelines, nobody will, bother to use them. So most of them are in research base. Yeah.
Pharmacology at SGR DIMS Amritsar. She actively contributes to research, conferences and pharmacology education, and she is published in national and international journals, including her work on drug addiction awareness among medical students. I now welcome Dr. Preeti Sood to please share her insights on good clinical practices. I welcome you, ma'am.
Good evening, everyone. After so much enlightening and newer things, in the end of the day, I'm just telling about the good clinical practice. And as we all go for the clinical research, because I am from a medical college, so I will talk about the clinical research. What is the good clinical practice mean? It is we have to follow first of all certain ethics and principle for the human safety, data handling, and the effect which we are showing. It should be reproducible, right? So along with that, it is our responsibility to carry certain standard norms, follow certain standard norms like good clinical practice is a set of the internationally recognized ethical and scientific quality requirement which must be observed for designing, conducting, recording, reporting the clinical trials that involve the participation of human subject. Because along with the data procurement, the safety of the human which are enrolled, we call it as subject, that is the foremost thing. Along with the drug research, compliance with this good practice involves assurance that the rights, safety and well-being of the trial subjects are protected and the result of the clinical trials are credible. So we all know about this, that is the basic research, then is the disease discovery, there we don't want any regulation. Then come the drug recovery. In these categories we don't need any regulation. Then when we find this lead molecule and preclinical development is there in the laboratories, and there is a certain good lab practices. But when humans are involved and clinical trials are there, then the GCP comes into place and the trial 1, 2, 3 phases are there. Then the manufacturing, we have the good manufacturing practice. That is the phase 4 for the ADRs. Now, what is the key principle of the GCP? I will skip certain slides because of time constraint. Yeah, but the key principle of the GCP, integrity of the scientific data for the first thing, then in the subject protection, accurate and reliable data. That means that whatsoever data we have collected of the trial, that should be reproducible. It should not be like this, that we are showing certain result and when auditors come and they check the thing, it is not reproducible. They are showing certain different things. Then fourth point is transparency and accountability. Whatsoever effect the drug is showing, it should be little transparent. We should not break the code that we are showing everything to everybody, but if required, it should be transparent, and up to certain limit, the patient should be knowing what's going to happen with him or her and what can be the result. So these are the points. Now, what was the need for the GCP? In the initial era, that was the before 20s, there was no standard set mark like the Second World War, etc., all the episode. In those, these were the type of the research which was going on, like the Nazi medical war crime during the World War. They just took the prisoner and they will do the study. And there was no codes. I will show few slides like tuberculosis model or some infection model. They just cut the human, healthy, normal human, put the silica, put the glass, all these type of things. For tuberculosis, they themselves instilled the tuberculosis. And then comes for the high altitude model, and for the other things, they have done all. And then come the era when there were so many physicians, they were just did a trial in the camp prisoner, and all of them died. And then the need for the ethics for the humans come into play. And it was called as Nuremberg Code, which states that the voluntary participation is required, means they have the right to say yes or no. And then these GCP guidelines come into play. So Nuremberg Code in 1948 says that the voluntary participation and the informed consent. What is the informed consent? It is the other side of the voluntary participation, that the participant should have a right the participant should have the permission to say yes or no, and along with that there are the various methods like written consent, verbal consent. Nowadays there is the set boxes in the physician's room, they record like AI, that part has come, and you can record the consent whether the person really has said yes or no. Right? So all these comes into play over the time. Then is the Coffers Amendment, Belmont Report, all these things comes into play time to time. Then eventually Declaration of Helsinki, it comes into play, and there are so many, right now we are just following that, and there's so many amendments at the different time. Like 1970 till 2024, there are so many things. Nowadays, last amendment is a need for the e-consent on use of data and samples. Like if a person can't give that black box is e-consent sort of, let patient can't write, so controversy should not be there. And it is recorded at some other place, that is the current research organization. They put a box and they record over there, but we are examining the patient over here. So many trial, multi-center, unicenter are conducted in that way, right? So now what the present thing is, GCP means quality data along with the ethics, right? So now what are the Helsinki Declaration says nowadays? There are the certain important points, like one is the autonomy, then is the beneficence, maleficence, fidelity, truthfulness, confidentiality and justice. Most important is autonomy. Autonomy is like we admitted a patient and we want to do the treatment, like a patient is of the gallbladder problem, right? We can operate by this cholelithiasis, we can operate lab chole, we can do open procedure, we can do. We have to explain everything to the patient and look for his opinion. He want to do open, he want to do closed, right? So we should not decide. We should take the person into confidence. And he has every right to say no. It is the principle of the autonomy. Second is the beneficence and non-maleficence. They are nearly same. Beneficence is for the benefit of the patient. Non-maleficence is the do not do the harm. They are nearly same but not the same, right? We have to benefit the patient, other is the not to harm. Then comes the fidelity, truthfulness, confidence. These are also covered along with that. Last is the justice. Justice is the distributive justice is the term nowadays used after 24. It says that like we have got two groups and we have got a new drug and we found that it is found to be good. We should not discriminate that our near ones and our relatives, they should get the good treatment, others should get conventional. So that is the principle of the distributive justice. We should be justifying, we should be honest while doing the practice. So these are the 4 pillars of the GCP nowadays: autonomy, beneficence, non-maleficence and distributive justice, right? So all these things are there. Then is the ethics consideration in GCP, is the, I will skip all these because we're running. Now when we conduct a trial, there is the role of the sponsor, then is the investigator, then is the institutional ethics committee, and then is the subject. Everywhere human subjects are in, to be kept in the mind. And then is the auditor, then is the monitors. All these things I will discuss. First is the sponsor. Who is the sponsor? Like here we are talking about the drugs which are industry funded, right? And the sponsor is the person who just want to do a trial on some drug. It can be multicentric, it can be unicentric. So, but the trials we do in the institutes ourselves, there is no sponsor. May or may not be the sponsor is there, right? So, in the sponsor funded, designing of the protocol and everything is in the domain of the sponsor. He has to plan with the help of the other people, like there are so many regulation, people who deal with all these things. He should make a team and he should develop a suitable protocol, right? Then he will appoint an investigator. Then he will take the drug, how the drug should be kept and all other things, right? Second is the investigational product. He will make the inventory in the institute where he want to conduct the trial, so that a drug should be kept at a optimal temperature. Failing that drug is a chemical entity, it will be of no use conducting a trial, right? Then is the financial and administrative support. It is again with the sponsor. He should be bearing the charges of the investigator. If subjects are paid, how to pay them. If ADR happen, how to manage, who will pay the compensation. And then is the monitoring and auditing. Appointing a monitor and auditor, both are his responsibility. Now, what is the difference in monitor and auditor? Monitor is a person who is a part of that trial, but company has appointed him that you go to the site and see the things are running properly. While the auditor is a firm normally, that is a resource. We have just outsourced it. And they will come and they're like inspectors. And they will come and they will see the things are whether right or whatsoever written in the protocol is complied upon, or it is just they're doing in its own way. So auditors and monitors, they are not interchangeable. And monitors are the internal people who will come visit. If investigator wants something, they will provide. If something is not up to mark, they will do all the things. So this is the thing. Then institutional review board, when a sponsor has developed a protocol and selected the investigator, he or she will, the people will come to the institution and they will submit that protocol in that particular institution. And institution ethics committee, we know that it comprises of 7 to 8 people, like some are external, some are, most important is chairman should not be from the same institution. It should be external, because chances of the bias will be more, right? And other people, few clinician, few ethicists, few social worker, all these people comprise. They will look upon the protocol and then they will see whether it is feasible, whether it is up to mark, and सबसे important, they are not harming the human subject. Those are the patient over there. So these things are the institution review board. Then clinical protocol, what it is a document. It is a compilation of the so many things, and which in this case sponsor had constructed with the help of the other people who are expert in those, right? Regulators, government authorities, and newer guidelines, all other things. And no doubt, drug itself, biggest thing, right? So the protocol developed is there, reviewed is there, then submitted, approved, amendments. Now there is one term that is the protocol amendment and protocol deviation. Deviation is like investigator, who is a doctor over in this my task, and he himself can change it. May the laboratory technician changes, like maybe the person has forgotten, maybe something, maybe they don't like the process. So these things, they are the deviations. And while the amendments are, we are talking about a new drug. And in the globe, we don't know, though we have got very excellent internet and all the things over there, but everything is not open. So in the other part of the globe, the same drug can be worked upon by certain other set of the people. And during the process of our clinical trial, maybe some result has been found, right? And maybe that result is different, that dose, that everything is different from our one. And we think that we are little lagging behind and we want to change it. And that is the protocol amendment. We can amend the protocol at any stage, and only thing we have to inform the regulators, and we have to take the institution ethics committee into confidence. So paperwork should be complete, and it is normally done by the sponsors and investigators along with. But deviation is manipulation. of the protocol. We are just doing it according to our own will. Maybe the technician has missed some reading, we have introduced some more. These type of the thing deviating from the regular path. So these things should not be done because it will affect the ultimate data which we have collected. And it will be introducing the bias. Then come the investigational product management. It is very clear till date that we have to maintain a top optimal temperature, whatsoever is written in the papers. And all these sources are to be arranged by the sponsor, not by the investigator. Because sponsor is the whole soul of the thing. And he will arrange everything. Whatsoever is lacking, we have to just tell the person that we are lacking because of that. Then comes the investigator responsibility. Here he is a doctor or some research scholar, right? And till date it is clear, but protocol adherence is important. Whatsoever is finalized, data integrity, subject safety again and again. Then in the reporting requirement, right? Whatsoever data is maintained, it should be properly managed, software should be loaded, and the whosoever is involved, be the laboratory technician, be the data operator, they should be properly trained, so that they should not be like that. You are saying, put the data into software, and he say, I will do after taking the D, then forget. So it will lead to protocol deviation. We don't want that, because otherwise there is no fund of running $1 million or 1,000 rupees of a trial, right? So there is no fun of doing like that. So this is very, data management is very important. Then data integrity and management. Management, I have told you again, proper fitting. Just ma'am, 2 minutes, 2 more minutes, it's going to finish. Integrity is data should be reproducible. Means if we have followed the protocol properly and no deviation has been done, integrity, reproducibility and compliance, everything will come into play. There is no big thing to do. Then all these, then is the quality processes and quality procedure. Again, auditors come into play, they will visit, find the errors, meetings are there, so many things are there. And monitor is an internal thing. The auditor play more constructive role. Though they look very, inspector always look questionable ki why they have come. But they ultimately shape the clinical trial. This is the thing. Then informed consent process, we know importance of that. And ADR reporting is very important. In protocol, it should be properly mentioned what will they do if something happen. Who will pay the compensation? Every ADR should be documented and it should be aptly covered. And then we have to think whether the subject to be kept in the trial or excluded. But before that, it should not be done. It should not that like that. The patient is sick and you ask, we drop you out. Nothing like that. So these things should be properly managed. Then the amendment deviation I have talked about. Then document record keeping is good. Then is the GCP training and qualification, all the whole of the staff, whosoever is involved. And it should be certification. It should not be locally trained, and they should be skillful. So they should know what is the importance of the protocol again and again. So these are the things. Required authority, GCP compliance we have talked, inspections ho gaya. Then challenges and emerging technology again and again. AI is also. All these factors are affecting. So how we can overcome these? Repeatedly, how we can improve our GCP, good clinical practice improvement, regular audit, training the staff again and again, data management system, it should be robust. And then is the technology integration, right? So these things will improve our good clinical practice.
Thank you.
So, done.
Any questions from the audience? The sponsor.
They will just look for those things which you have deviated, how you have deviated. Monitor is the internal banda. Auditors, you have to
Okay, yeah. Yeah, it can be done by ethics committee as well as by the regulatory authority as well. So that will be like external audit. No, we do it audit in PGI. We go and audit the clinical trials. No, we do for our clinical trials. For the ongoing clinical trials, we do auditing, yes. So this is what has been enabled by the NDCT rules. The ethics committee, they have a lot of power now, and they have responsibilities as well. So now they have to be registered themselves, and plus they also have they can go and do auditing for clinical trials. It's not like finding faults, it's just making sure everything is in order. Yeah. Yeah. So that. Yes, yes. So that is done within the trial team. So like you are saying, the CRO, their monitor will go and so if the trial is going on at 10 different sites, so they can go at different sites and look into if the processes are in order. Right, exactly. Thank you. Thank you.
Thank you so much. I congratulate all the speakers. Very illuminating talks and very informative. I wish we had the time to understand more and gain more information. Maybe next time we have the links now. So I hand it over to the organizers now.
I would like our chairs to felicitate our speakers. Yeah, Dr. Ashish Kakkar and Dr. Poonam Pipalani. And our speakers, Dr. Aarti Singh. Dr. Rohit Bisht. And Dr. Preet Sood. I will request Dr. Manish Jain to felicitate our chairs, Dr. Ashish Kakkar. And Dr. Poonam Pipalani.
Speakers, group 4. speakers can all come for group photo.
On the behalf of the organizing committee, I thank our speakers for their excellent tasks and for advancing this vital discussion. I also thank the audience for your thoughtful attention. The symposium will now proceed to the next scheduled event. Thank you.